All right. I'm showing live. I'm going to trust it. Hello and welcome, my gentle and, of course, very modern apes. Welcome to Gutsy Gibbon. This is my channel. And this is the, very late part, ninth, some late episode of a series that myself and my guest have been working on here for quite some time, where we essentially work through why conventional science believes evolution to be the best explanation for modern biodiversity and why we think the Earth is very old. And this is of note because I'm an evolutionary biologist, asterisk. My background's in like primatology, and I study human evolution now. So I'll put that label on myself, but there are a lot of labels I think you could maybe slip in there that might be more appropriate. So this is what I study formally, and Will, my guest, is a young Earth creationist, but he's also a guy that loves science. So we're sort of working 1 month at a time through the entirety of an adapted version of a lecture that I taught at university as a graduate student, which I still am a graduate student. Adapted to be a little bit more general, but we're finally starting to get into the specific human evolution stuff today, and I'm super, super excited for it. Will, you want to give a little bit of background, like where your claim to fame, maybe?
Yeah, no problem. So my claim to fame, which is not that great, but I'll take it, is I took flat earthers to Antarctica about 18 months ago, and that was a pretty big deal. We're actually creating a documentary about our trip down there. It was called The Final Experiment, and that should be released this year, so within the next few months.
That is beyond exciting. So you guys know this, or if you've been, I guess, watching this series, you know that Will and I, when I was in Colorado this year, we met up for lunch. We both brought our spouses, and we ate at a Brazilian steakhouse, which was an absolute blast. Will treated us, and it was an expensive place. So I don't, I was kind of floored by the options on display. And as I've said before, I still to this day think about the honey-coated cheese that they serve, which seems like it wouldn't work, but it really, really works. And I got to ask Will a couple of questions about like, what was it like to go to Antarctica, and to especially go to Antarctica with people who are going there expecting to see some kind of proof that the Earth is flat, expecting to not see the 24-hour sun. So we got to talk a little bit about that. And I'm sure you're going to get into that in the documentary, so I won't spoil anything, but I'm really looking forward to it. Typically, the way that we structure these shows is Will begins with a presentation where he talks about sort of his main takeaways from the last lecture, things that we may agree on now, things that we may still disagree on. And today, if memory serves, he's going to sort of be talking about quite a few of the case studies, or at least referring back to some of the case studies over the past 4 lectures, talking a little bit about cetaceans, bird evolution, mammal evolution, and tetrapods. And I'm super excited to hear his perspective on that. After that, we usually go into my presentation, and it's a beast today, which is why we talked about this beforehand. We are going to change the super chat minimum just for today to $15. If you want us to read a super chat at the end, actually, you want to do 10 or 15? What do you think?
Yeah, it's tough. Probably 15.
Let's do 15. We're going to do a $15 minimum for super chats. You can still send them in, but we won't read them at the end unless it's for $15 or more. And the only reason we're saying that, I'm actually hoping that this is going to get us fewer super chats, not more $15 super chats. My hope is that we will not be here forever tonight, but I say that every time. And then the top comment on all of these is usually something along the lines of like, Erica at the beginning, this one's going to be short. 5.5 hours later. So my hope is that we're not going to be here for 5.5 hours, knock on wood. But if you really have a burning question for either of us, you can ask it for $15 or more, or you can save it for next time. Hopefully we'll be able to bring that back down for shorter presentations. There's just a lot to cover as we jump into human evolution, and we're not really even talking about everything that I want to talk about. With that, Will, are you ready to take the stage here, take the floor?
I'm ready.
Excellent. I've got your screen right here, and you are shared. I'm going to mute myself on my end and test to make sure you can't hear me.
Okay, sounds good. So let's see. This picture is probably my favorite picture that I've ever taken. The reason I'm proud of it is, for those of you that don't know, I take pictures only with my cell phone, and I don't edit them. So this picture has a cool story behind it. My daughter, my youngest, she came running into the house screaming. And it was one of those screams where you don't know if she's lost a limb or if she's just seen something incredible. And so luckily it was good news. And she said, "Dad, there's baby iguanas outside." So she took me outside. and I just had a field day with these baby iguanas. You can't tell size on this picture, but these baby iguanas would fit in the palm of my hand. So this was a picture that I took, and just everything was perfect, the color and everything. So it was really exciting. I spent hours just photographing these baby iguanas. They were a little skittish, but would be also curious, I think, because they were so new to the world. And so I would just take every opportunity that I could to get as close to them as I can, to where I wasn't spooking them, and get these really fun pictures. So anyway, those are the pictures I thought would be fitting for tetrapods. All right. Before we start, I want to give everyone some advice. And this advice is for those who want people who disagree with you to trust you, and your goal is to get them to change their mind on something you think they are wrong about. There's a very strange phenomenon that I've noticed in the modern day debate slash debunking world. This applies to flat Earth versus globe, creation versus evolution, old Earth versus young Earth, etc. I see it everywhere. And what I've noticed is a strange inability for people to admit when someone on the opposing side gets something right. It seems like simply admitting the opposition got something right is their kryptonite. Now, since this refusal to admit an opponent is right is so common, I don't think it really stands out or is even noticed. But what definitely stands out is when someone does the opposite of this. Now, a couple weeks ago, I got an email from a pretty prominent YouTuber, and in this email, he praised a young Earth creationist. Now, I can't say who this YouTuber is because I don't have his permission. I take private communications very seriously. I don't ever want to lose somebody's trust, but I can tell you that this YouTuber is definitely not a fan of young Earth creationism at all. Now, when something like this happens, it hits you like a ton of bricks because this is so rare in the debate and debunking world. More importantly, though, it helps you trust that person as they are showing they are willing to be objective. Now, only twice since we've started this endeavor back in November of last year, only twice have individuals praised someone on the opposing side and communicated to me personally that they were right or made a good point or do good work. Now, again, these two examples stood out to me because it so rarely happens. The first person who did this was John, who runs the YouTube channel Age of Rocks. Here's his channel. I wouldn't be surprised if he's actually watching right now and is in the comments. I can't see the comments. But he told me that Dr. Stephen A. Austin, who's a young Earth creationist, he told me that he's a good scientist, does good work, and publishes his work in the peer-reviewed literature, which is what you should do. Now, I cannot tell you how refreshing this was to hear from John. It caught me off guard because you almost never hear this in the polarized debate culture today. Most importantly, though, it made me immediately trust John. He showed me in that simple, honest acknowledgement that he can be objective and not caught up in the us versus them mentality, which we see in sports and politics. And I feel like we shouldn't see that in this pursuit of truth. Now, the second person this happened with was Rebecca Davis, who runs the YouTube channel Examining Origins. Here is her channel if you're not familiar with her. And Erica, you're going to, I think, appreciate this, but more than once, Rebecca has contacted me to tell me that you were right about something you said. And even though it was implied, she made it clear and verbalized to me that I was also wrong. Now, again, this was refreshing and made me trust Rebecca as she was willing to be objective and brutally honest with me, even at the risk of turning me away. So this brings me to the email I received a couple weeks ago from a prominent YouTuber about a young Earth creationist. The young Earth creationist he was praising was Dr. Jason Lisle. He shared a video with me praising Jason Lisle's presentation and explanation of something in mathematics known as the Mandelbrot set. That's not really tied to evolution or the age of the Earth. So if you haven't heard of the Mandelbrot set, that's okay. But I do want to share this. On January 21st of 2022, Jason Lisle published predictions about what the James Webb Space Telescope would detect And this was about 6 months before the first image from the JWST was released. And Jason Lyle's predictions were based on his belief that the Earth is young, and his predictions went against everyone in mainstream science. Here is his original article from January 21st, if you want to go look at it, which contained his original predictions. Now, what was mainstream science predicting? They were predicting galaxies to taper off to zero by around redshift 14. If you're not familiar with redshift, that's okay. I don't want to get too technical here, but you can definitely look that up. They were predicting galaxies in the earliest stages of formation, and they were predicting the first stars would have no heavy elements. These are known as population 3 stars or pop 3 stars. Now, what did Jason Lyle predict? He predicted galaxies at higher redshifts than expected due to the accepted age of the universe. He predicted galaxies would be fully formed and not in the process of assembly. And he predicted that the stars would contain heavy elements, so pop 2 stars, not pop 3 stars. So, how did these predictions go? Well, mainstream science got the first one wrong. Jason Lyle got it right. The second one, mainstream science got it wrong. Jason Lyle got it right. And the third one, mainstream science got it wrong. And Jason Lyle got it right. Now, if you don't believe me, here is NASA's website. This is currently there. Sometimes I do screenshots with dates and times so you can see when I took the screenshot. Also, you can see the URL. But look at the quote from this article. It says, with Webb, referring to the James Webb Space Telescope, we are able to see farther than humans ever have before, and it looks nothing like what we predicted, which is both challenging and exciting. That's on NASA's website right now. Here's an article from Scientific American. JWST's first glimpses of early galaxies could break cosmology. The James Webb Space Telescope's first images of the distant universe shocked astronomers. Is the discovery of unimaginably distant galaxies a mirage or a revolution? This is very significant. Now, Erica, I want to encourage you and others, Buzz Rana, Dr. Dan, Dr. Joel Duff, Professor Dave, Dapper Dinosaur, Grayson Hawk, all the other YouTubers that watch your channel, to make a video titled something like, or something along the lines of, Young Earth Creationists Got It Right, and give credit to Dr. Jason Lyle for his accurate predictions that went against the overwhelming scientific consensus. I'm confident this will help the other side trust you more and will also encourage them to watch more of your content. So again, if your goal is to change minds, I do believe this will go a very long way. All right, back to the presentation. So last week, this is a screenshot, not last week, last session, this is a screenshot where I got something wrong. And the people in the comments made it very clear to me that I got something wrong. So thank you for that. I put the comments here so that you guys know I do read the comments. I try to read as many of them as I can. I should mention that while I did get it wrong in the first paragraph, I did get it right in the second paragraph. But hey, a mistake is a mistake, and I'll own up to it. So thank you. I also created a syllogism in my last presentation. And some people in the comments who are clearly better than me at philosophy, because I'm not an expert in philosophy at all. I barely dabble in it. Told me that my syllogism was valid, but that it's not sound. I would bet money that you guys are absolutely right. I believe that you're right. And so I am more than happy to say thank you for correcting my error there as well. All right. Now, I will say this. It's honestly a bummer that this has been such a sticking point in this endeavor. I truly don't understand the importance in saying birds are dinosaurs as opposed to saying birds evolved from dinosaurs. It feels to me, just based on feedback, that it would not be enough if I ended up agreeing that birds evolved from dinosaurs if I don't also agree that birds are dinosaurs. And this is how it feels sitting in my position. I like memes, as you will learn if you haven't seen that already. All right. Now, my confusion has only increased after learning last session that snakes and whales are tetrapods. I've dabbled in Greek for many years, Koine Greek, and can clearly see the etymology of tetrapod as tetrapus, which means four-footed. So I went to the dictionary. The dictionary defines tetrapod as a vertebrate with two pairs of limbs. And so if a snake and a whale are a tetrapod, then the dictionary definition is no longer valid. I went to Wikipedia, though, and Wikipedia came through with a different definition, thankfully. And they define a tetrapod as any vertebrate animal of the clade Tetrapida. Now, this definition is circular. It's absolutely circular. And this is also what's called a tautological explanation. Something is tautological if it is redundant, circular, or true by definition, offering no new information. You'll notice the definition of a tetrapod currently on Wikipedia does not give us any new information of what a tetrapod is. We don't even know which vertebrate animals are tetrapods or not. So I think that this definition is completely invalid. Now, I love analogies. I think they help. And imagine if we did this. This is an analogy. Imagine if we did this with kids at school regarding shapes. So they go to class and you say, okay, we're going to teach you today what a quadrilateral is. And a quadrilateral is a four-sided shape. You teach them, they understand. Then they come back to class the next day and you tell them that triangles are quadrilaterals. They're not going to understand. Now, if any of you right now is giving me a hard time saying, Will, this is silly, a triangle is not a quadrilateral. Well, let me tell you something you may not know. There was actually a fusion event in the past where two sides joined together into one. And so that is the evolution of quadrilaterals and triangles. Now, speaking this way is super confusing to me still. And so I couldn't help but continue to think about it and what it would mean if applied to other areas of life. And it occurred to me that if these things are true as stated, birds are dinosaurs, snakes are tetrapods, that would mean some Americans are Europeans. This is a picture of my beautiful grandmother. She was born and raised in England, and she was born in 1925. And here's a picture of me with her at her 100th birthday party. Unfortunately, she just passed away this year, a few weeks shy of her 101st birthday. Now, is it better to say Americans descend from Europeans, or should we say Americans are Europeans? I've never considered myself a European, even though my direct ancestors originated in Europe. Now, it is often asked of creationists to define what a created kind is. And I think this is a perfectly valid question to ask someone who denies evolution. But I'm curious if you would accept the exact same kind of circular and tautological definitions that are given for clades, like we just saw. So how about this? A created kind is any organism that belongs to a kind originally created by God. Not good enough? How about this one? A created kind is any living thing or its descendants that was created as a distinct kind by God in the beginning. No? Okay, how about this one? A canine kind is any animal that belongs to the created canine kind. Still not good enough? How about this? A canine kind is any carnivorous mammal of the created canine kind. Now, if these definitions of created kind don't sit well with you, I don't blame you. My point with that exercise had just one purpose, and that was for you to know exactly how I feel being told that a tetrapod is any vertebrate animal of the clade Tetrapida. Now, I regret that we need to discuss nested hierarchies again, especially after Erica made an awesome presentation last time with Schmanimals showing how this works. But I will say that even though we are going to rehash this again, I'm glad that I took a deep dive on this. Spoiler alert, I finally get it. Now, I spent many hours looking into the history of taxonomy and the classification of living things, and I finally figured out why I've been struggling to understand and accept this. I'm excited to announce that I am ready to accept Erica's Schmanimals example and also the work of Linnaeus, but I am not yet sold on cladistics. This might sound really strange to some of you, but it was my discovery that classifying living things by physical traits, morphology, and classifying living things cladistically, phylogeny, it was my discovery that these are actually two very different things that helped everything finally click for me. So let's look at phonetics versus cladistics. This is what clicked for me. According to phonetics, they look at what's called numerical taxonomy, but cladistics looks at phylogenetic systematics. Phonetics looks at observable traits, whereas cladistics looks at shared derived traits. Phonetics is quantitative, whereas cladistics is parsimonious. These are actually very different. Now, quick history. John Ray coined the term species, and he was the first to classify animals systematically based on a combination of internal and external structures. Then we have Michael Adamson. He argued against artificial systems that relied on one or a few traits, and he promoted using all available traits, morphological, anatomical, etc., without prioritizing any to reflect true natural relationships. Then we have Carl Linnaeus, who we've been talking about so much. He created the system of binomial nomenclature that we still use today, and he also organized the natural world into a hierarchical classification. Then comes Sokal, Robert Sokal and Peter Sneath. And this is important. They wrote a book called Principles of Numerical Taxonomy. And this was, again, their explanation as to why we should use phonetics to classify living creatures and to create nested hierarchies. But at the same time came a guy named Willy Hennig. He wrote a book called Phylogenetic Systematics, arguing, no, we shouldn't use phonetics. Instead, we should use something called, which is now called cladistics. Then came along a guy named Gareth Nelson. He wrote a book called Systematics and Biogeography, Cladistics and Vicariance. And it was those two guys and their books against these two guys, Sokal and Sneath, and their book that created what is now known as the systematics wars that took place in the 60s, 70s, and 80s. The systematics wars took place in the literature, conferences, and in articles and books like I just showed you, where these main four, two on each side, were arguing that their way should be used versus the other. The light bulb finally went off for me when I realized that phonetics and cladistics are different methodologies. This is why I would follow the Linnaean hierarchies and even the Schmanimal classifications, but then get stuck on birds are dinosaurs. Because according to phonetics, birds are not dinosaurs. According to cladistics, they are. According to phonetics, humans are not fish, but according to cladistics, they are. And according to phonetics, snakes are not tetrapods, but according to cladistics, they are. And finally, according to phonetics, turtles are anapsids, but according to cladistics, turtles are diapsids. This is a big one in our discussion about tetrapods, which I will return to shortly. So when I got this presentation here on how we classify animals into nested hierarchies, that is phonetics. And the Schmanimals example is also phonetics. Since the last session, I took my kids to the Denver Museum of Nature and Science, and I noticed that they say that turtles are anapsids. That means that is also phonetics. Cladistics says birds are dinosaurs, snakes are tetrapods, and turtles are diapsids. Now, some of you might be thinking right now, Will, these are two sides of the same coin. They are just two different methods of arriving at the same conclusion. I mentioned last session that I have noticed the many comments telling me that genetics is the final experiment for evolution, but I asked two questions that have gone unanswered. The first was, what does evolution predict regarding genetics? Meaning, once genetics is discovered and before we sequence any genomes, what does evolution predict we will find when we sequence those genomes? The second question was, what could be found in genetics that would falsify evolution? Falsifiability is a key principle of science, and thus far, I'm not really hearing many ways evolution could be falsified. So I didn't get any answers that I could find. So I'm guessing on these. So these could be wrong. So is this evolution's genetic prediction? Is it organisms with similarities in phenotype, observable traits, will have similarities in genotype in their genetics? For example, bonobos and chimps, they look similar. And so is the prediction that they will have similarities in their genotype? Is another prediction from evolution that organisms without similarities in phenotype will not have similarities in genotype. For example, a jellyfish looks nothing like a fruit fly, and so therefore they won't have similarities, significant similarities in their genetics. If those are the predictions that evolution is making in regards to genetics, then what does it mean when we find the opposite, the exact opposite? So the exact opposite of number one is organisms with similarities in phenotype that don't have similarities in genotype. An example of that is falcons and hawks. And if that's shocking to you, join the club. I just learned this and it's shocking to me too. The opposite of the second prediction on the left is this: organisms without similarities in phenotype that have similarities in genotype. A good example of that would be hippos and whales. So again, what does it mean when we find the opposite of what evolution predicts? Now, we recently had a bird of prey event at our house. We hosted it. We had nearly 30 kids, which was so cool. Get to experience birds of prey in person. And so here's a beautiful hawk that we had in our house. We had an owl as well, which is just beautiful. I love owls. We had a peregrine falcon, which is amazing. They're incredible birds that almost went extinct. And we actually had a bald eagle in our house as well. Don't let the non-bald head fool you. This is just a juvenile bald eagle. So I am in a Facebook group for Colorado birds, and it's really a fascinating group. Any bird experience that we have in Colorado, you can post about it and get a lot of feedback. And here is the last post that I made in the group, which will give you a little insight into my sense of humor. My wife does not really enjoy my sense of humor, but I submit this as evidence to her that other people do. Now, someone in this group posted that falcons are not related to hawks and eagles as once thought, but are actually nested with parrots and songbirds. I about fell out of my chair when I read that. I didn't even believe it. Now, this was a complete shock to me, and I still struggle to believe it. One of the most frequent questions asked in the world of birds is whether a bird of prey is a hawk or a falcon. They are very hard to tell apart. And upon further inquiry, the similarities in physical characteristics between falcons and hawks is independent according to evolution. So they now believe that what you're looking at here is convergent evolution. This throws a pretty big stumbling block for me on the topic of fossil similarity and evolutionary ancestry. If two animals that look this similar to where they are routinely confused are not related, but actually evolved independently, convergently, then the explanatory power of similar fossil structures is essentially lost. If falcons and hawks were extinct and we only had their fossil record, their morphological similarity would be used, without a doubt, as evidence of transition and close evolutionary relationship. We remember from last time the tarsal bones of the colugo, the flying lemur, and the tree shrew. It was originally believed that the flying lemur was nested with tree shrews because of the similarities of their bones. These look like dead ringers to me, and as we recall, genetics said no, they are not related to tree shrews. They're actually more closely related to us, or primates. Now, turtles. One of my favorite things to do is to sneak up on turtles. This was a turtle I snuck up on in the Caribbean. Now, turtles, are they anapsid or diapsid? Here is a turtle skull. I have to say that's one of the coolest skulls I've ever seen. I would put that in my house for sure. Now, turtles are anapsid. You can clearly see what an anapsid skull is here versus a synapsid or a diapsid. It just, whether or not you have holes or not behind the eye cavity, anapsid has none, synapsid has one, diapsid has two behind each eye. Now, here's a paper that actually says genetically turtles are diapsid. Okay, so because of genetics, turtles are now considered diapsids, even though they are anatomically anapsids. So it is now believed that somewhere on the evolutionary timeline, the fenestra, which is the openings behind the eye sockets, were lost. I believe this is known as evolutionary reduction. I'm very confused. Snakes are tetrapods, turtles are diapsids. And so it's very fascinating to me and confusing that we make such a big deal about these physical characteristics, but then the physical characteristics don't really seem to matter once genetics come into play. Now, the message I've been hearing in the presentations and from the comments is that the nested hierarchies created by phonetics, Linnaeus, etc., has essentially been confirmed by genetics. Here's a comment from last session. Yes, when you do it with genetics, it makes basically the same tree as physical traits. I now have serious doubts that this is true. From what I'm seeing on my side, genetics is producing very different nested hierarchies than those created by phonetic classifications. I was curious what the scientific literature had to say about nested hierarchies, specifically if the hierarchies originally created by morphology, phenotypes, matched the hierarchies created by genetics and genomics. I wasn't sure who would be best to reach out to in order to find out what the scientific literature says on nested hierarchies, so I reached out to the only two people I know who are routinely quoting peer-reviewed papers. Before you guys give me a hard time, Erica is excluded from this. She's incredibly busy and she doesn't need to help me do my homework. The first person is Dr. Cornelius Hunter. I've mentioned him before. And then Rebecca Davis, who I also mentioned at the beginning of this presentation. They sent me a ton of papers. This one is about the octopus, and it says morphometric analysis fails to separate the two New Zealand species from one another and from T. peninsulae, and it is unlikely that such data would prove phylogenetically useful, even if they provided separation among species, since there was no congruence between morphological and molecular matrices for the Southern Ocean species. Here's one on sloths. Here we utilize collagen sequence information, both separately and in combination with published mitochondrial DNA evidence, to assess the relationships of tree sloths and their extinct relatives. Results from phylogenetic analysis of these datasets differ substantially from morphology-based concepts. Here's one on pigs. The current molecular evidence has overturned hypotheses of evolutionary relationships based solely on comparison of morphology between species of sub-Saharan African suids and has provided additional insights into the relationships within Eurasian pigs. Here's one on snakes. Another challenge is the ongoing disagreement between what morphology and molecules suggest regarding several higher-level snake relationships, specifically several clades that were identified on the basis of morphological variation do not appear to be monophyletic when viewed through the lens of molecular systematics. Here's one on gharials, crocodilia. To explore the causes of discrepancy between molecular and morphological estimates of crocodilian phylogeny, we examine puzzling features of the morphological data using a priori partitions of the data based on anatomical regions and investigated the effects of different coding schemes for two obvious morphological similarities of the two gharials. Here's one on birds, ratites. Several morphological studies strongly support ratite monophyly in conflict with our nuclear genetic data. Simply collecting more data or combining molecular and morphological data are unlikely to resolve the impasse generated by these incongruent signals. Now, this is just the tip of the iceberg. I was overwhelmed with how many papers I received from Cornelius and Rebecca, and in addition to those I just covered, I left out the papers showing a similar pattern that we just saw with bats, badgers, bears, cats, Chelicerata, crabs, Mustelidae, rodents, skunks, and turtles. Now, here's a paper called The Universal Ancestor, and this says phylogenetic incongruities can be seen everywhere in the universal tree, from its root to the major branchings within and among the various taxa to the makeup of the primary groupings themselves. Here's a paper that says often consistency index values as low as 0.3 were observed. As a result, we began to question whether there was any useful phylogenetic information in particular data sets, as evidenced by the number of steps on the minimum length tree or by the magnitude of the consistency index. Here was the most important paper I looked at: consistency indices and random data. Here is a chart from that paper, and I'm going to do my best to explain it, and I hope I don't get this wrong, but I don't think I will. So this chart, a consistency index of 1.0, which is that red line at the top, is essentially that the morphological data and the genetic data match. That's kind of like a perfect match. Now, these two lines that you see going down, those are two random data sets that they used for purposes of looking at what is predicted is the red line, and then what's just purely random data. And you'll notice the number of taxa that they looked at, which goes here along the x-axis. The more taxa that they looked at and compared, the more it showed closer to random versus the top line of 1.0. So only when they used 5 taxa did they get a result that matched. As soon as they increased that, all the results more closely aligned with the random data set versus the prediction. So, conclusion: phonetics classifies organisms strictly by overall similarity, using as many characters as possible and without distinguishing ancestral from derived traits or requiring monophily. And as our mutual friend Clint Laidlaw says, monophily is king. So, since I now understand and accept phonetic classifications, such as those created by Linnaeus and those presented with Schmanimals, where do we go from here if they are seemingly incongruent with what we are finding genetically?
Sweet. All right. I have some notes. I
Awesome.
loved your presentation. Can you hear me okay?
Yeah, loud and clear.
Awesome. I think this is my favorite response that you've done. And I think the reason for it is that a lot of the papers that you talked about, I haven't read, right? Like I have not read most of those papers. I wouldn't say some, I would say I haven't read most of them. And part of the reason for that is that I specialize on primates. And as you're probably going to see, primates don't have this problem. And primates, phylogeny and morphology, or the genetics and the morphology, they actually map quite well with one another, with a couple of very interesting exceptions. And I'm going to go through today why genetics is the winner. Why does genetics beat morphology? Because it's a fundamental concept that's kind of, I wouldn't even say it's fundamental to evolution. I would say it's fundamental to biology. And so that's why we're going to have to take sort of a roundabout way back to genetics. What I will say is that hopefully, remind me if I don't, but we need to come back to the specific taxa that your papers covered. Some of the ones that I want to see that I'd like for you to send to me are the pig one, the rodent one. I think you mentioned an artiodactyl group there at some point. I tried to write, you said like 4 badgers. That's what it was. Because these are groups that, at least according to the, what's the word I'm looking for? At least according to literature reviews that I've done, these are some of the groups that also map quite well. So I'm curious to see when these were published. I wholeheartedly agree that octopus, sloths, snakes, gharials, ratites, and there was one more that you said, but I don't have it here. Some of the birds of prey. That I'm unsurprised that these don't map. I'm unsurprised that the morphology doesn't map with the phylogeny there, especially given the completeness of the genomes that are involved, but we'll talk about that a little bit later. 1, I don't know anything about Jason Lyle's predictions regarding the James Webb Space Telescope, other than that he made them and that I've heard people say, and I don't know if this is true or not, so that's why I wrote this down to ask you. I've heard people in this space talk about how Jason Lyle, I was going to say James Lyle, that his predictions did not preclude an old Earth, because I think that's kind of important. Like, are his predictions, like, it's kind of like a Humphreys thing, right? I mentioned this last time, but are the predictions being made in that they preclude an ancient universe? Or are they only consistent with a young Earth is I guess the summary of it, because discrimination in hypothesis testing is kind of the backbone of this. And I don't know the answer to it. So if you don't know the answer, I'll probably just look into it and bring it up next time, because I'm not sure. I have heard whisperings, but that doesn't mean they're correct. So I want to look into this myself. I agree on building bridges. I try to do that myself. I'm happy to talk to any creationist that wants to talk to me. I read your screen grabs and I put them into my PowerPoint. So we'll kind of come back around on that, like with tetrapoda and things like that. Loved your bit on phonetics versus cladistics. I would argue cladistics can, and in fact often is, quantitative. Quantitative things can be reliant on parsimony because it has to do with how many given genes, if you're looking at a gene comparison, or how much of the entire genome, if you're doing sequence identity, or alignment rather. And then if you're looking at aligned sections, how much of that aligned section matches base pair by base pair. That's numeric. I mean, that's quantitative for sure. But parsimony is in tree building important. I agree. Morphology is still important. Now I'm biased because I work with fossils. So of course I think morphology is important. And we're at something of a disadvantage with fossils because most of the time we don't have any DNA, right? Like fossils don't tend to preserve DNA. Every now and again they do, but not beyond a certain point, unfortunately for us. I think you make a great point on cladistics versus phonetics, and specifically I don't know very much about turtle, anapsid, diapsid, synapsid, but I have heard before that it's proposed to be an anapsid, or a diapsid rather, that's lost that fenestra, both of those fenestra. But I don't know enough about it to say. I can't really speak to that. What I will say is that it wouldn't be the first time, even within groups that we would both accept as related, it wouldn't be the first time that a trait has been lost in organisms. So I don't personally see that as problematic for reasons that I'm going to talk about here in a second as we loop back around. You talked about the final experiment. We're going to talk about that today. The final experiment for genetics. All these people are saying, genetics is what's going to do it. Genetics already did it. And I agree. And I think that I've got some good slides that are going to be helpful with that as we get into this. Falcons and hawks being related. Are they related?
Unrelated.
Yeah. Are they related though?
In what way? I mean, according to evolution, everything is.
Yeah, but do you think they're related?
Oh, definitely. Yeah.
Why?
Physical similarities.
Yeah, because they're overall really physically similar, right?
Yeah.
That's the same answer that I give when somebody says, usually creationists, actually always creationists, who say, what if we found dog breeds throughout the fossil record? We would think they were transitional species. Yes, and they are related, right? Like we would think they were transitional because they show a transition. And in fact, all dog breeds are related. So I agree. Convergent evolution, or another word for it is homoplasy, can confound specific relationships, but it doesn't confound whether or not they're related. You see what I mean? Like evolution doesn't propose and has never proposed that there are two things that are completely, absolutely 100% physically different, or at least very physically different, that those two organisms are on opposite sides of the tree of life. So for example, superficially, there are moles, moles like that dig in the ground. And then there's marsupial moles, and they look really, really similar. And part of the reason for that is because of convergent evolution. Marsupial moles evolved from a marsupial on Australia, and nothing was digging in the ground and exploiting the resources there. And so these animals took that niche, right? It's adaptive radiation. We see it all the time when islands get colonized today. When nothing is exploiting a specific resource, species that are there will tend to do it, or at least they'll try. But if you take a marsupial mole and a placental mole, like a regular mole, and you cut them open, it's going to become immediately obvious that one of them is a marsupial and the other one isn't, because one of them has a pouch and it has a specific type of uterus and it has a specific type of reproductive system. So while it is true that homology can confound the ends of the branches, what it doesn't do is preclude the idea that these things are related at all. So I hope I'm going to get into that a little bit now. Okay. Is it okay if I jump in?
Yeah, please.
Sweet. All right. This thing's a beast, so we'll try our best to get through this in a timely manner. Also, I'm going to do my best to stop and chat more frequently this time, because I think that would be better. All right, 1 second. Let me find what I'm looking for here. I tried to split this PowerPoint in two because I thought that might make it a little bit more comprehensible for my computer, because my computer was like lagging so badly. but it didn't help. So I ended up just keeping it as 1. All right, 1 second. Also, I had this in my notes too. I loved those baby iguana pictures. That's insane. I love that you can see, what did I write down? I wrote, love that you can see the hand through the leaf. In that first, the way that the light was coming through.
Yeah.
That was awesome.
So fun.
Also, I had no idea that you did those with your phone. That's pretty sweet. My phone's good, but I don't know that it's that good.
I buy cell phones specifically for camera quality, nothing else. I have a reputation. So people are like, if you want to know what phone has the best camera, go talk to Will.
There you go. I'm going to be bugging you next time. All right. Why is it doing? Oh, I remember what I have to do. Okay. Window. There we go. Okay. Can you see this okay?
Yes, it's not in presentation mode yet.
How about now? Oh, I'm a stupid idiot. Hold on. Okay. Share screen. And then you would think that I would know how to do this after all this time. Okay. And then now presentation mode. We good now?
Yes. Perfect.
Excellent. Okay. I love this picture. In fact, when I looked at this picture, I was reminded about how much I hate AI generated images, because this is not an AI generated image. This photo was taken in 2016 by Sergei Uladnikov. And this is a baby bonobo looking at his or her hand. And I find it very striking because, it's easy to anthropomorphize, but you can almost see this little character contemplating, like, what is this? What am I looking at here? As they look at their hand, you can see human babies do the same thing when they're little, and they're like figuring out that they've got hands and feet and like what it means to move them around. I found this very familiar. And in addition, there's a hypothesis that we're going to talk about a little bit later next time when we enter into the hominins, that part of why humans look different from chimpanzees is because we retain some of our neonatal or baby features into adulthood. Because as you look at this youngster here, that head and muzzle and face looks a lot more human than an adult bonobo or an adult chimpanzee does. It's got a rounder head, it's got a flatter face, the brow ridge isn't very prominent. And so we'll discuss that sort of when we come to it, because that's not a great genetic change. It is persisting characteristics from childhood into adulthood. And maybe, as we'll talk about, that has some interesting consequences down the line. Now, as always, a little bit about me. Here's my background. You can stop and look at it if you want. I'm a current 6th year, going to 6th-year PhD candidate in biological anthropology. So I study human evolution, and that's why I'm excited for today, because we're talking about human evolution. And this is a series where we are going to be talking about evolution, why the scientific community accepts it as the consensus, and I would say to why people find it compelling in general outside of the scientific community. This is not meant to be a creationist and debunking series, but we're going to talk a little bit about creationism today, as you'll see here shortly. Will knows which bits hopefully we're going to mention, but yeah. We have a roadmap here. Today we're talking about apes, and I have here apes and the first hominins. I wish, as I was saying to Will beforehand, there's just too much information about apes. I know, Erica has too much to say about apes. Isn't that shocking? So we're going to boot our first hominins to Australopithecus and kin, and I actually do think that it probably makes more sense to do it that way anyways. So I added this while Will was talking, quick addendum. This is a slide from the tetrapod conversation last time. I sort of defined what tetrapods were in a scientific sense and said simplistically, tetrapods are vertebrates with 4 limbs and identifiable digits. You would correctly notice that whales and snakes don't have, at least hind in the case of cetaceans, but in the case of a snake either, they don't have 4 or hind limbs with digits. So why would a snake be classified as a tetrapod? Well, as I noted here in the last presentation, legless animals like snakes, caecilians, which are a type of amphibian, and cetaceans are included because, number one, they have the genes for these traits and or immense genetic similarity to animals with them. So we're going to talk about that, sort of leads into, okay, so what? Why should genetics trump morphology? We'll talk about that in a second. Number two, developmentally, they do have these traits. One thing that I really hope we touch on or circle back around to is the fact that cetaceans grow hind legs in the womb. They start to grow them. And then that section of development terminates, and those leg those leg buds are resorbed back into the body for the rest of development, except when they aren't, in which case sometimes cetaceans are born with hind flippers. And you can recognize in those hind flippers, in that hind leg, the homologous bones that map developmentally and genetically to legs in terrestrial animals. I don't know why they would have those characteristics if they didn't descend from terrestrial animals. But more importantly, that's partially why they're considered tetrapods, because developmentally, they have hind legs with 5 digits. Same with snakes. And snakes can be born with these little spurs sometimes that are basically just a femur attached to their little pelvic. They're going to call them pelvic spurs, but I don't think they're actually bits of the pelvis. It might be bits of the pelvis rather than the femur. Not a snake person. And then this last definition here is the one that you mentioned that feels very circular, tautological, that they're overwhelmingly most similar to other legged tetrapods, that they descend from legged tetrapods. That's the cladistic definition. And I understand why that's frustrating. Same thing with birds and dinosaurs, or turtles and their anapsid diapsid status. I understand that. I'm really hoping that the conversation about genetics today is going to clear that up. So I just want to be clear. There are other reasons why those animals are classified as tetrapods other than just they descend from tetrapods. Those are located in their genetics, their development, and in sometimes their abnormalities, what they're actually born with. I took a screen grab of this because you nailed it. Yes, when you say it occurred to me that if these things are true, as stated, birds are dinosaurs, snakes are tetrapods, then that would mean some Americans are Europeans. Yes, in a cladistic sense, Americans descended from Europeans. So in the same way that you would say that like English is a Latin language, Americans are of European descent. And so you nailed it. That is absolutely exactly what the concept is. And that's why cladistics can be so counterintuitive. But the reason why we use it as a means of organizing life is because it can be applied backwards into time. And it allows us to handle things like the fossil record. And it also makes sense of the genetic patterns that we see. Okay, so far so good?
Yes, definitely.
Okay. So last time we talked about tetrapods, and I talked about these concepts that we've been hammering home, repeating over and over again throughout this series, the nature of biology, what we actually need to support evolutionary theory in a given lineage. With regard to biology, that would be mechanisms that need to exist to explain the emergence of new variations of traits. So how do you get variation in the first place? Mutation, right? Natural selection acts on variation within a population, and that's how populations change over time in the face of an environment that's always changing. We talked about taxonomy a little bit, and we're going to come back around to that today, especially with regard to functional versus non-functional regions. We talked about development and anatomy, like why those characteristics support evolutionary relationships. Let me ask you just here, what do you make of the hind limbs that start to develop in whales and dolphins?
Yeah, I was thinking about it when you said it, and I'm a little bit interested in how we know exactly what that is, that is what it is and not something else.
Because when they
Yeah.
continue to develop, they have those hind flippers. So if you go back to the cetacean presentation that from a couple of months ago, there's a picture of that dolphin named Haruka. She was a Japanese dolphin. I say that like she's from Japan. She's from the waters around Japan, and then the Japanese researchers captured her and had her in like an enclosure or whatever, like a research aquarium, I guess you would call it. But she has hind flippers, right? So in her, those limb buds continued to develop into adulthood, and they became those actual functional hind flippers that she could move. They were attached to her body, like she could use them to pivot as rudders and everything. So that's in part how we know, because we've watched them continue to develop, but also they have the same genetic code for the legs that develop in other animals. Does that make sense?
I don't understand what it means that they have the same genetic code, but it does make sense what you're saying.
So like, think of it this way. When we talked about Hox genes last time, we talked about how you could take the Hox genes for leg development in a fruit fly, and you can put them in a mouse, and the mouse will develop normal legs. The genetic language between all life, all animals at the very least is universal. They speak the same language. And so genes that do one thing in one animal can, especially if they're body plan genes, do the same thing when they're transplanted, literally plucked out of the organism and placed into something else. So the code is the same. So when you look at these genes, these body plan genes, and you isolate that limb bud in those cetaceans, the code is for hind limb development in every other animal that we find it in.
So why doesn't it work?
Because it turns off. They've had cetacean-specific mutations that silence it before it can finish development. And the reason is because why would you have hind limbs if you're committed to the sea? This is why every animal that lives in the water full time, none of them have hind limbs. Like, it's just a burden. If they get, like, they don't help you do anything, and they drag on the body, they make you less hydrodynamic. And if they get injured, they can cause an infection, which is kind of useless or harmful, I would even say dangerous, if you're not using them for anything. You see what I mean? So this is why you lose structures. This is why an animal would ever lose a structure, if it's unhelpful and if having it can be a danger. It's also why, like, when people are born with extra limbs or digits or things like that, sometimes people, they'll have surgeries to get rid of them because they're an encumbrance. Not often, but sometimes.
Okay, yeah. So are you suggesting that a single mutation at some point in the past made offspring go from no legs, or from legs to no legs, and then that continued?
It wouldn't be a single mutation, although in some cases it can be, right? So we've talked before, like in some of the previous lectures, how a single mutation, especially on body plan genes, can drastically change everything downstream. So one of the examples we talked about is how you get human-looking hips from like ape-looking hips, which they're very different in how they're structured. Ape hips, like non-human ape hips, to be scientifically, cladistically, let's say, accurate, are very tall, and the blades of the hips don't run front to back, they run side to side. So this question in human evolution is, okay, how do you get the human configuration and the human morphology, even though all the bones are the same, right? Humans are shorter and squatter, and they're wider instead of taller. So how do you actually get that? And the genetic pathway to get there has been outlined by scientists, and it's relatively few actual genetic steps, and none of them in and of themselves are lethal. What's cool about that is it means you can accumulate these mutations in the background, and they don't cause any harm, but it's only after they've accumulated in their entirety that they cause a morphologic change because of how these genes interact with one another. And when it comes to these hips, in that case, which I'm most familiar with it because it's apes, the development of those hips when they become human-like impacts the development of the muscles that attach to those hips, right? So you don't have to have a separate mutation that changes the musculature. The musculature is already developing alongside that ilium, or the blade of the hip. And so when the hip rotates into what you would call the sagittal plane, front to back, when it rotates during development, those muscles come with it. And so you end up impacting multiple organ systems, the skeletal system, the muscular system, the cardiovascular system, because all of the veins and arteries have to change slightly their direction with it, with a single set of mutations. And so in the case of whales, right, if you'll go back and watch the cetacean stuff again, or just like flip through the slides, you'll see that we've managed to isolate the genes that are responsible for leglessness in whales and in dolphins. And so it's like, to me, from an evolutionary perspective, it's like, okay, the prediction is that if whales were once on land, if their ancestors were on land, we should be able to find traces of that everywhere. And it's like, well, we do. They have the genes for legs, they're just turned off. And when accidentally, because of how development works and how mutation works, when they're not turned off, they develop hind flippers, which is like, I can't think of why that would happen unless they descended from something that had hind flippers. And so then you can look to the fossil record, and what do you see? You see the loss of legs in things that go from being like non-whalish looking things to whalish looking things. And yet the last whalish looking things have the same ankle bones as the terrestrial things that are proposed to be their ancestor. So it's like a nice little sanity check, I guess.
Yeah, I'll definitely review it again. It's hard for me to try to visualize this process, but
yeah.
But yeah, I'll look at it again.
Sweet. And try to keep in mind too, when we talked about mutations at the very beginning, like actual mutations and the impact that they have in living populations that we're watching right now, right? in a lot of these experiments, we're looking at changes in structures that already exist. So if you'll remember those lizards or anoles that had an increase in the size of their finger pads, right, and and a decrease in the size of their bodies, or an increase rather in the size of their bodies because of hurricanes that had swept through their area. All adaptations that make them better for holding on to trees, so they're not swept away by hurricanes, which is an experiment that was run by scientists collecting a million lizards, anoles, taking a bunch of measurements of them before and after an actual literal hurricane swept through the area. And then they came back, they were like, who survived, right? And then they looked, and the ones who survived were the ones with these finger pad adaptations, which is musculoskeletal. So it's a change in something that's already there, which is important because when we're talking about something like humans and chimps today, humans and chimps have all the same bones and all the same muscles and all the same organs. So it's like when we're looking at a change between a human and a chimpanzee, what is actually changing? Same thing if we look at a cetacean versus these little hoofed land mammals in the past. What is actually arriving, or what is actually disappearing? Does that make sense?
Mm-hmm.
Okay, cool. So we talked about all of this with regard to tetrapods last time. We also rehashed a couple of major concepts, but when we did, a couple of new questions emerged, and I'm going to try to answer them today along with some older ones that we haven't really addressed yet. The first one is, how do we falsify evolution? That's really important. The second is, are there any out-of-place fossils? And this is, Will sent me the paper for the out-of-place fossil that he mentioned last time, so we're going to go through that because I'm going to be honest with you, I don't really understand what's going on here. So I'm gonna tell you where my problems are, and since you know the author, maybe you can reach out to him and get some more information. I'm going to talk about how we date fossils on the surface because that's really important. It is true that if we're saying we've got morphologic change over geologic time, like the emergence of the whale traits from non-whales, that has to happen through the geologic column in order to propose that one sort of begets the other, right? So how do we do that when a lot of the fossils that we find are on the surface of the ground, right? Or on the surface of the earth, like sitting on the ground, like you walk up to them. How do you date those? Why does genetics beat morphology? How does the molecular clock work? And we're going to touch a little bit on that today, and we're going to go a lot more in depth on it next time as we talk about hominins, because it's important for that. But we are going to mention it today. Okay. It's really important to talk about what science is. I'm thinking a lot about final experiments and about scientific progress and about paradigm shifts when and where they occur. And I think I want to retouch on this. So science is a way of acquiring knowledge. We defined it at the beginning as this, and we talked about the criteria that something has to meet in order to be considered science. One, it has to seek natural causal explanations for observable phenomena. So it can't speak to supernatural or unobservable processes or like one-off events. Two, it searches for observable regularity. So you can't test something that has only happened once, right? Like how do you actually understand what has to be repeatable? Claims about it have to be repeatable. They have to be consistent. And when it comes to historical sciences for this is done by making predictions based off of assumptions about the past. So if you assume something about the past and you consistently get accurate predictions, that's repeatable support for the notion that those assumptions were warranted. And in order to say that those assumptions weren't warranted, there has to be a better explanation that makes more accurate predictions, a superior model. This is how this applies to historical sciences like geology or paleontology or astronomy. And lastly, it relies on empirical observation by independent observers. This is really, really important. And I think you get a lot of pushback for this because I feel like you as an individual are very open-minded to the idea that anyone can come in and overturn a paradigm. And I think, have you seen Ratatouille? The Pixar movie?
No.
Well, you are killing me with this. Okay. Well, in Ratatouille, there's a line, anyone can cook, right? This famous chef writes this book called Anyone Can Cook, and the main character of the movie is a rat who wants to learn how to cook, right? It's a Pixar movie. And so, Remy is like, anyone can cook. What does that mean? And the answer that the movie gives you is not that literally anybody can cook, but it's that a good cook can come from anyone. So I feel like I agree with you in that overturning a paradigm can come from anyone. but it doesn't mean that anyone can overturn a paradigm. I'm not going to walk into a chemistry lab or chemistry symposium and overturn the basics of, inorganic chemistry, because I don't know anything about chemistry. So you have to have some kind of background, some kind of understanding of the subject in order to overturn it. And the more people that come to the same conclusion, the more reliable an idea is. And I sort of predicate that on the idea that these people are coming to these conclusions by testing and making accurate predictions via those tests, right? So they make a prediction, they test it, they're right. They make a prediction, they test it, they're right. Someone does that in Brazil about the same subject as somebody in Alaska, as somebody in Kenya, as somebody in Norway, as somebody in Papua New Guinea. You can probably be pretty sure that they're on to something, right? If they're all testing on E. coli and they're getting the same results, it's like, okay, well, they're probably on to something about E. coli because all these people are getting the same result every time. And lastly is that things have to be falsifiable. If it's supposed to be scientific, you have to be able to prove it false. And I've been racking my brain about this. I said this last time to Will. I've been racking my brain about the falsifiability of evolution because I've always proposed in this series that the Cambrian rabbit as the falsifiability. But I'm not so sure about that now, and I want to explain why. Evolutionary theory is a theory, right? So theories are specifically hypotheses that have been rigorously tested across space and time by tons of different people. And they consistently are showing these predictions are coming to fruition. They're consistently making accurate predictions. Hypotheses are always provisional, and models and theories are always revised, but hypotheses can sort of graduate to becoming a theory if they're describing, why something is happening, and they're doing so accurately by multiple people, across a century and a half. This is a nice little graphic that I use with my students that sort of helps you differentiate between an observation, a hypothesis, a law, and a theory. Theories don't graduate to become laws. Laws and theories are the same level of powerful, but they're just describing different things. A law is usually an equation. It's describing what's happening, and a theory explains why things happen. So a couple of well-known theories outside of evolution are like heliocentric theory, cell theory, the germ theory of disease, gravitational theory or relativity, plate tectonic theory. And so when I was thinking, how would you overturn evolutionary theory right now? My immediate next question was, how would you overturn any of these other almost universally or universally accepted theories? How would you do it, right? Can they be falsified, right? Heliocentrism could be falsified when it was first proposed, right? We started making observations about the solar system, and if it turned out that all of the other planets and the sun indeed orbited Earth, the observations would have borne that out, but they didn't. Consistently, scientists found that no, actually in our solar system, things are orbiting the sun, the Earth, Mercury, Mars, Jupiter, whatever, everything is going around the sun. So that's how you could have falsified it in the past. How would you do it now? Could you? I guess if you found out there was some kind of like quantum gravity well or something that simultaneously managed to overturn all our previous observations and then provide a better one, maybe. But does that mean that heliocentric theory isn't scientific? What about cell theory, right? There were people that argued that cells were composed of essentially these little particulates that came together, and that's how sort of development and division happened. And then we found out that no, cells are these individual units and they make up all living things, with no exception that we've been able to find. Either something is a cell, it's a single cellular organism, or they make up larger organisms and it's a multicellular organism. It can only be broken down to those cells. Otherwise, the organelles aren't going to be able to survive, at least mostly free living. So we discovered this when Robert Hooke invented the microscope. But how would you falsify that now? I don't know that you could. Does that mean that cell theory isn't scientific? No, because it could have been falsified. There was a chance to falsify it. Again, in theory, you could still falsify cell theory, but I don't know what it would look like. Okay, what about germ theory of disease? In the past, people believed in miasma theory. They believed bad smells caused sickness. We proved this wrong when we found out that actually what makes you sick are tiny particulates, microorganisms that are in and of themselves alive or close to alive in the form of viruses that hijack your body and make you sick by destroying your tissues or attacking your tissues or colonizing your tissues. Okay, how would you disprove germ theory of disease now? It could have been disproven. It could have been falsified. But how would you do it now? I don't know. You would have to show that there's a violation to germ theory of disease, that some smells can make you sick. And they weren't making you sick because they were carrying germs, right? They made you sick because of some kind of essence that hasn't been categorized yet. Same thing with gravitational theory, right? How would we disprove it at this time? So many of our observations about gravity and how it works have borne out. We literally, our planes rely on how gravity works. The way we build structures rely on our understanding of how gravity works. Our understanding of sending rockets to the moon relies on our understanding of how gravity works. So could you falsify it? Again, there was a time when we could have, but I don't know that you could do it now. Does that mean it's not scientific? Also, for that matter, does it mean that if I drop my pen that I'm holding right here on the floor and it suddenly flies up, does that mean that gravity is disproven? Would that one observation do it? What do you think? Asking, actually.
No, that one observation wouldn't disprove gravity.
It has to be a pattern, right?
Yeah, I'm not familiar enough with these four theories to tell you off the cuff here how they would be falsified. So I'm not ready to say they can't.
Yeah.
But you're giving me something to think about.
Yeah, me either. And this is why I'm asking. Because and chat, you and people who watch this later in the comment section, if you've got an idea as to what kind of observation or observations we could make that would falsify these theories now, I'm all ears. Because I sat and I was like, okay, what would it look like, right? The last one is plate tectonic theory, right? The idea that the tectonic plates that we're on top of right now are moving. And this is something that we observe every single year. We watch them moving now. So how would you falsify it? There was a time when it could be falsified. Before we knew plate tectonics, the tectonic plates that we're standing on were actual things. Then the people who proposed this were like, okay, this is what I think. We could go out and test it. We could see if these plates are real. And they are. So how do you falsify it now? I don't know. I don't know if you can. But I want to be clear. Just because I can't think of a way to falsify these things, one, it doesn't mean that they can't be falsified. And two, the difficulty I'm having in coming up with how you would falsify them doesn't make them less scientific. Mm-hmm. And you can see where this is going.
So how could evolution have been falsified in the past?
We're going to talk about that today.
Okay.
My argument, the TLDR, is that it boils down to genetics and the fossil record. I think both of those two things are what distinguish evolutionary theory and specifically common descent. Because I don't think, and actually, I want to ask you this right now. Do you have a problem with mutation and natural selection?
What do you mean by a problem?
Do you think they happen?
Well, mutations definitely happen.
And can populations change in the face of a changing environment?
Yes.
Okay.
I think of the moths when you say that.
Yeah. Well, I'm actually, now that I'm thinking about it, I feel like I remember you saying I'm down with natural selection. So I
do have issues. Yeah, I do have issues with two things regarding mutations.
Okay.
I didn't have time to put this into my presentation, but in your last presentation, you had a slide on mutations, and you had bad at the top, good at the bottom. Yes. And I think your best one at the bottom was a body plan change.
Just the most extreme, not the best, just the most extreme. That's the biggest change that I can think of.
Okay. The picture right next to it was a fly with 4 wings or 2 sets of wings. I don't consider that good. And so I'm not yet sold on mutations providing a significant benefit. That makes sense. The examples that I've been given, like lactose intolerance, being able to hold your breath, sickle cell anemia, all of those examples I've been given don't change us physically, to where, we see a difference, a body plan change, so to speak. So that is something that I still struggle with.
Can I ask what is your impression when I say a mutation is good? What in your head, what does that mean? Because I want to make sure that I've explained something properly.
Sure. I hear a good mutation is something that would confer a survival advantage.
Perfect. So can you conceive? That's exactly right. I wasn't trying to like
Yeah.
gotcha. What I realized and what I have realized as we've been doing this is that sometimes I think I assume that we agree on something, but I haven't actually checked. Like I assume that if you haven't mentioned it, we agree on it, but I haven't actually touched base. So I'm trying to do a better job about that. And I should have been doing that this whole time, but live and learn. Do you think that if a second pair of wings in flies conferred a survival benefit to them, made them better flyers, would then those wings be selected for?
Hypothetically, yes.
Would it be a good mutation?
Yeah, I think so.
Okay, cool. Awesome. So I would propose then that if you go back and look at some of the mutations that we discussed, these genetic studies that we do in the present are mostly to show that these mutations can happen. That if you tweak this Hox gene, you can get a body plan change. Or when we were talking about the zebrafish or the spider with 10 legs or the 4-winged flies for that matter, that they can happen naturally on their own. So that's providing a natural body plan change, as in, like, I mean, I get that you're saying it's not a body plan change. I don't know what else would count though. Like, you know what I mean? Like going from 2 wings to 4, to me, that's a body plan change in the same way that going from fins without digits to fins with digits.
Sorry, I might have misspoke. Going from 2 wings to 4 is a body plan change. I have no disagreements there.
No problem. Okay, cool.
I just don't consider that example a good mutation. I consider that a deleterious mutation.
Yeah. Well, and it would be if it didn't confer survival benefit. Because my understanding is that flies with 4 wings, fruit flies with 4 wings, they're not better flyers, right? But if they were, it would cease being a deleterious mutation, right? Like the context is what decides whether or not, I say, whether or not a mutation is beneficial. The reason I'm checking on this is because I'm sort of cataloging a list of the mutations that have to occur with humans and chimps, but I just want to make sure. Okay, are we going to move?
Yes.
Okay, cool. So each of the ideas that we just talked about help form the foundation of their respective fields. And I encourage you to go look into heliocentrism, germ theory of disease, cell theory, whatever, which just pick one that interests you and see just how foundational it is to the field that it's in, right? I mean, like medicine without germ theory of disease, where are we? Microbiology without cell theory, where are we? Astronomy without heliocentrism, where are we? So I would propose here that falsifying any of those ideas, let's just say we could falsify them, right? If we could show that they were false. In science, that's not necessarily good enough. It's not necessarily falsified them so much as is shown a flaw in the model that exists. In order to truly falsify something, you have to provide an alternative model that explains the data better. So miasma theory, the idea that smells make you sick, was a real theory, right? It was an idea put forward by, medicine men of the time to explain why people got sick, right? I should say it's a hypothesis, but they would consider their lines of evidence probably enough to be a theory by the standards of the time. But then germ theory comes along and it doesn't just say, hey, miasma theory is wrong. It provides a different, better model that says it's not just that smells don't make you sick. It's actually that if you take like tissue from a sick person and put it under the microscope, you can see the microorganisms that are making them sick. And that's what's actually causing all of this. So in science, ideally, if you want to falsify a theory, especially, you need to have something that explains reality better, waiting in the wings, ideally.
Can I push back on that?
Please.
Okay. It seems to me that falsifying something is way easier than proving something.
It is. And you can't prove anything in science, right? It's all provisional, always. So even our best theories of today, even though to my knowledge, gravity has never been violated on planet Earth, it's still a theory. And we could tomorrow make a series of observations that start pushing us towards rejecting it. right? So falsifying something is typically relegated to individual hypotheses. So you wouldn't say, I've made a single observation that falsifies cell theory or something like that. In the same way that my pen falling up wouldn't falsify gravity overnight, right? Like, you're right, falsification has a lot of steps to it. And then you want to have something waiting in the wings, not to prove, not to say, these observations actually prove my hypothesis, but to say that my alternative model actually makes more accurate predictions about reality, which is how cell theory worked and heliocentrism worked and plate tectonics worked.
Yeah, I personally, I can think about this more and possibly change my mind, but where I'm at right now is that it's possible to falsify something without a replacement model. An analogy I would,
It
okay.
is.
An analogy that I would give, which of course is just an analogy, is that I'm thinking about as you're saying this is in a courtroom where some guy's on trial for murder, and the defense attorney says, "Well, Your Honor, my client couldn't have committed the murder because he was on vacation in Paris when this murder took place, and we have the proof of that." And the judge says, "Well, who did commit the murder?" And the defense attorney's like, I have no idea." And he's like, "Well, then your client is guilty." So
I'm with you, but counterpoint, someone could also point out that while the client was in Paris, they paid to have an assassin kill the victim,
yeah.
right? So that single observation seems like it does preclude him being guilty, but it might not, right? So look, this is why single observations don't always do it. It's got to be a pattern. And I agree that to falsify something, you don't have to have another model waiting in the wings. It's just good practice, because otherwise you're left with these situations like with germ theory or heliocentrism, where you're saying, okay, well, I falsified this aspect of it. It doesn't seem to work in this particular scenario, but I don't know that would actually falsify the theory. That might just falsify the observation. This is why hypotheses versus theories, like, I can't think of the last time a theory was falsified. You know what I mean? Like,
Yeah.
And again, the reason I'm pitching it to you like this is because I don't have the answer. I was noodling over this and thinking, like, okay, like what would work? What would do it?
Yeah, I appreciate what you're saying.
Yeah.
I do appreciate it, and like in your previous slide, not the one before this, but a couple of them before, you were talking about how one observation wouldn't do it, and I'm glad you said that. Therefore, we have to throw out the rabbit in the Cambrian, because I was going to tell you that anyone could find a rabbit in the Cambrian.
Yeah.
So.
I mean, that's the thing, right? Like, if you found, and I want to be clear, like, a rabbit in the Cambrian, to me, it's the observation wouldn't disprove evolutionary theory in deep time. But if you showed that it was actually a rabbit in the Cambrian, like if you did checked off all of the boxes, that would put you a long way towards, if not dismantling deep time, dismantling all of our understanding of evolution, because you'd have a rabbit, a modern mammal, living 550 million years ago. I don't know how else you would explain it other than we've gotten something seriously wrong. But I would propose, as somebody who's interested in out of place fossils, and I try to track them down whenever I can, because when they're given to me, at least, because I think they're interesting and I want to know what's going on. I've never seen one that's compelling, and what's really weird is I've never seen one that's actually, even as it's proposed, to be that out of place. And I'll talk about I have a slide about that, so I'll mention that in a second. But
Okay.
okay, so here are my 3 falsification criteria that could have disproved evolution at any given point over the past 150 years at these major discoveries. I think that if natural selection was shown not to work in the 1920s by the series of experiments put forward by all of these early geneticists and zoologists who were doing work actually in the field, this is actually when the peppered moth study was first put forward. I think if natural selection didn't work, evolution's dead in the water. And they said the same thing. In fact, you can find some pretty crazy headlines from the time where they're like, look, we validated Darwin's theory of evolution, because natural selection is at play in these populations. And when we run the math, it makes predictions that then bear out in those populations, whether we're talking about fruit flies in the lab or the peppered moths in the wild. So I think that would have done it, because no mechanism, no change over time. Number two would have been if we discovered there was discontinuity in the genomes. And this is going to be the reigning theme for the talk today, is the unity of all organisms genetically and the pattern that they form, that nested hierarchy that they form with their genomes. Not one gene, not a few genes, but their full end-to-end genome. And what I think is especially important is that they form these hierarchies not just in the functional regions of the genome, but in the non-functional regions too. And everyone agrees, at least to my knowledge, that there is some portion of the genome that's non-functional. Usually creationists will say, okay, that's what's happened since the fall, right? That you have the fall of Adam, there's genetic entropy, whatever. And then that's what explains the non-functional regions of the genome. And when I say we know there are non-functional regions of the genome, I say that because we can knock out portions of the genome and organisms will develop just fine, right? They're not huge portions of the genome, but that does show us that the genome is not 100% functional. And these hierarchies that I'm going to pitch to you today, they show up in the genome that is important to us, that does stuff, and in the portions that don't. And that tells us that there's inheritance going on that's belying a history that I will, again, get into later. It's kind of hard to do this now without having explained what all of this means, but I want you to come back to this after today and think like, okay, is that fair? Would that have done it? Because I think that would have done it. And the third thing, and this could have happened anytime from, the 1810s to now, is if we discovered a lack of order in the fossil record. Like consistently, like the rabbit and the Cambrian would be one thing, but I think you could have immediately disproven common descent if there was no discernible order at all, right? Like most people agree that there's some level of order. Creationists will come up with hypotheses to explain it, like the ecological zonation model, or they'll say it's hydrologic sorting. Everyone agrees that there's an order. The question is why. But I think you could have falsified evolution right out the gate if you were finding T-Rex and deer next to each other in the same layer, like all across the board. And what I'll say too, and I'm adding this now, I think you could also falsify a lot of our ideas of deep time if you could show that every single major fossil bed that we have was deposited catastrophically under water conditions, right? Like a global flood type thing. And the reason I say that is because there were scientists during the 1800s who tried to do just that, the diluvialists. Like Winston Whewell, there's another guy, I forget his name, the early flood geologists. So that's what I think would do it. I think all three of these right out the gate could have cut evolution and deep time off at the pass, but none of these came to be. And so the question that I am asking myself now is like, okay, well, what would do it now? Because as you said with gravity, if we found one rabbit in the Cambrian, people would be like, there's got to be a better explanation. And you're right, that's what they would say. And we would look for one. Mm-hmm. Now, if you found 10, I think we'd have a problem. Or if the one that you found, it was genuinely a rabbit in the Cambrian and no one could falsify it, I think that would put you a long way towards it as well too.
Okay. We still haven't sequenced all the genomes. And so that I see a little bit of a hole, a little bit of an opening there. If I could take it, can you help me understand what you mean? Like maybe a couple examples of discontinuity in genomes?
Yeah. So I'm saying there isn't any, right? So all organisms that we have, when we sequence their genomes, they form a nested hierarchy with one another. There's not a stop sign that exists in the genome that says all felids are exclusively clustering here, and all canids are exclusively clustering here, and all ursids are exclusively clustering here with no overlap with these other groups. In the same way that they form these nested hierarchies physically, they form these nested hierarchies genetically. And so if we're just talking about the functional portions of the genome, that can be easily explained. Organisms share characteristics because they were designed by a common designer, right? Like, of course, a lion and a bear can share a lot of their genome because the genes that allow a bear to grow its claws and allow a lion to grow its claws, both of which are made of keratin, a designer would put that gene in both of them, right? And so, of course, they share that gene. That doesn't necessarily mean they inherited that gene from a common ancestor. But that discontinuity, so like I would say that point that's a great point in favor of evolution, but it certainly doesn't preclude common design, right? Now, on the other end of that is that there's portions of the genome that don't do anything. And there is in fact, not just a nested hierarchy, the same nested hierarchy in that non-functional region shows up as the one that we see in the functional region at these larger taxonomic categories. And so because of that, we have to ask the question, why is the stuff that's not doing anything, what seems to be, from an evolutionary perspective, non-functional evolutionary leftovers to some degree, why do those show the same family tree if they don't have a function? To me, that would be like, that's like a smoking gun. Does that make sense, what I'm getting at there?
Yeah, I still don't understand, though, what a discontinuity in genomes would have looked like.
Well, for instance, it would be like there's no overlap in the genomes of like felids and canids in their functional and non-functional regions. It wouldn't be that all life shares some degree of their genome in the functional and non-functional portions. There would be discrete clusters that don't overlap, or at least mostly don't overlap. That would be evident. That would be out the gate falsify common descent, because then you would have to say, like, statistically, it would be impossible. You'd be having to argue for separate origins for life for every mammal order, and then they would all have to evolve along the same trajectory to reach where they are today, which would be ridiculous. But the fact that they have these nesting patterns in the stuff of their genome that does stuff, and also the non-functional stuff, tells us that this relationship is there because they have similar characteristics, but also that it's there because they inherited those characteristics from a common ancestor. Maybe we come back to this because I have a couple of slides that maybe will make this make more sense. Okay. I'm going to make a note. I also made a note about your mutations here. I'm going to make another note here. Back to X slide so we can come back to this and we'll check with the discontinuity stuff then. Okay. All right. So, TLDR, right? What would it take now? I think it would take a lot more to falsify evolution. I still think you could do it, but I think it would take a lot more. You'd have to come up with another reason as to why the fossil record seems ordered. Something that isn't explicable under evolution, while simultaneously coming up with a different mechanism or explaining why we have these nested hierarchies. And what I found, especially with this nested hierarchy stuff, an answer that I've seen a lot in like the past year with the creationists that I've talked to, is that the answer seems to be like God could have just done it that way in the functional and non-functional regions. And I don't understand that, because I think that makes evolution unfalsifiable. What I'm saying is that if God could have done it in a way that exactly looks like evolution but isn't, we'll never know. Like we just would never know. There'd be no way to find that out. And so I don't find that to be a satisfying answer, although the people who I've talked to about this seem to. Okay, I want to do a quick case study. Pivoting a little bit, that's my thoughts on falsifiability. I'm going to continue to think on it and continue to noodle on it, and I hope you will too. You sent me this 2 or 3 days ago by Tim Clary, who's a geologist, geology background. He's at ICR. And this is an out-of-place fossil. You mentioned this to me as this is like a fossil, like a Cambrian rabbit type of thing. And I was really intrigued by it because I love that topic. And so I started digging into it really yesterday. So I'm going to give you my bird's eye view because I don't really understand how he's making the case that he's making. Maybe you can provide some clarity for me here. So he's got this title, Rhino Fossil Requires the Impossible from Conventional Science. He says a recent study in Nature, Ecology, and Evolution claims that the impossible actually happened, not just once, but 3 different times. I already find this framing kind of weird because the authors don't describe this as impossible. This seems to be something that Clary thinks is impossible. But I read the paper, and then I read some of the press releases of the paper, and like no one seemed surprised by this. I thought that was kind of weird.
Well, they wouldn't. They wouldn't think it's impossible. So.
No, but like that language is used a lot, right? Like speaking from my own field, all the time you see headlines that are like, or even the authors being like, "We didn't expect this. Like this, we can't make sense of this." And like that's fair, but let me explain. So he argues that this is an out-of-place fossil for 3 reasons, or I should have phrased this. He argues that it's an impossible fossil for 3 reasons. The first is that the fossil's in the wrong layer. The second is that it's in the wrong place geographically. And the third is that it has proteins in its teeth. Now, we've kind of done, first of all, do you think it's a fair summary?
Yes, that's not the whole fossil though. So I just want to make sure we're going to show the whole thing or not. Okay.
No, I couldn't find an easy picture of the whole fossil.
Okay, no problem. It's a very complete fossil.
Yeah, it's like a mostly complete skeleton. Yeah. The authors say that, and it's like very diagnosable because of it. It's not like, although just to be clear, if this is all that they had, I wouldn't think any less of the paper. This is an immaculate cranium. Like this is really good. So like I wouldn't think any less of them for that. This is the picture that came up. So this is the picture that I used. Basically, the summary is the authors found this rhinocerid. It's not a rhinoceros, it's a rhinoceros relative in this location here, Devon Island, basically in the high Arctic. This is really interesting because conventionally for a long time, it wasn't thought that there was an actual means by which for animals to disperse up into this area because as you can see, they're islands. And so crossing them to get there is not easy now. You couldn't do it now. Or if you did, you would have a high mortality rate trying to do it. So the authors describe the paper and describe the fossil. They say it's a mid-Cenozoic rhinocerid dispersal via the North Atlantic. So that's kind of the summary. Fossil found in a place where rhinocereds hadn't previously been found. And most importantly, it's dated to the Miocene, early Miocene, specifically around 23 million years of age. And that's my zone. So that's fair to say. Now, 23 is barely Miocene, but it is still Miocene. Okay. The fossil's in the wrong layer. I don't think this is true. I don't think this is a fair categorization, and I don't know why he's phrasing it this way. So what Clary says is he says, according to evolutionary paleontologists, rhinos are found on multiple continents in the early Cenozoic rock layers, specifically in layers identified as Paleocene and Eocene units. But that's already not true. Like we have rhinocereds today, right? Like rhinoceros can be found from as late as like 40 to 50 million years ago, all the way to present day. And we know that because like we can see them. So rhinoceroses, as he's sort of outlined it here, can be found in all of these periods. And I included this little phylogenetic tree from Wikipedia here to show you some of the ones that can be. There's our current one, Epiaceratherium, the fossil from the paper. This is Diceratherium, and this is Ronzotherium. And Diceratherium is younger than this new fossil, and Ronzotherium is older than this new fossil. So this new fossil of a rhino is bracketed by other rhinos, and rhinos indeed persist to present day. So I don't know why he said that. I might be misunderstanding, or maybe he elaborates later, but I've read this article a couple of times and I don't get it. He says the strata were all deposited above the KPG Cretaceous Paleogene layer. So this is the mass extinction of the dinosaurs. He says boundary on top of rock layers containing fossil dinosaurs, elaborating on what that KPG layer actually is. And then I've underlined this. He says previously the genus Epiaceratherium was found in multiple locations across Europe and Asia, but only in Eocene Oligocene rocks. Therefore, it was surprising to find Epiaceratherium, this new species, which I'm not even going to try to pronounce, in rock layers that were much higher in the rock record, extending the range of Epiaceratherium into the Oligocene-Miocene boundary of the Upper Cenozoic. Evolutionary scientists say this was 20 million years higher than expected. I also don't know why he's saying this, because I couldn't find a quote of any of the scientists in the paper or in any of their press releases specifically pointing out that finding a rhinocerid, even this genus, was surprising. Instead, what they talked about was that the location was kind of weird. And I'm going to talk about why it's weird here in a second. Not impossible for reasons that, again, I don't quite understand. So I don't know why this paragraph is acting surprised that a rhino was found at all. The timing of this doesn't make sense to me.
He claims that the scientists said it was 20 million years higher than expected. Is that true? Did they say that?
I don't think they say that in the paper. I can check though.
Okay.
Let's just pull it up. I have the bioRxV right here. bioRxV mid-Cenozoic dispersal. This is the one I have access to, 20 MA. They say, this is the only, well, actually make sure that that's the only mention. 20 MA for the NALB, and then that's a citation, Yeah, so they talk here about the North Atlantic land bridge that may have been crossable for mammals for at least 20 million years. But they're not talking about the existence of rhinos. Like the whole sentence says, notably multiple dispersals occurred in the Oligomycene, suggesting that the NALB, the North Atlantic land bridge, may have been crossable for mammals for at least 20 million years longer than previously considered, consistent with emerging geological and paleoclimatological models. We thus provide insight into the importance of the North Atlantic land bridge as persistent high-latitude connectors of geographically disparate terrestrial faunas, underscoring the pivotal role of the Arctic in mammalian evolution. But what Clary said was that evolutionary scientists say this, meaning like when he's talking about this rhino, is 20 million years higher than expected. So my understanding is that he's saying that the presence of the rhino is higher than expected.
Mm-hmm.
Do you think am I getting that wrong?
Oh, I don't know. I don't have the paper, unfortunately.
I'm happy to send it to you, but this is in the section talking about the presence of the species, not the land bridge. The land bridge is a different section. So I don't understand. Maybe this is just weird wording. I'm willing to grant that. But I don't understand why the rhinoceros-ness of the animal is weird, because, again, there's rhinos younger and rhinos older, and there's rhinos now. So I don't get that.
Okay.
He also says that the fossil's in the wrong place, and this is my real sticking point. Specifically, I took a screenshot. He says, this created a big problem. According to evolutionary plate reconstructions, there was no way to cross the Atlantic Ocean at that time. The authors of the study write, and he quotes the authors here. Whereas paleontologists recognize that the North American, and it's actually, this should read North Atlantic land bridge instead of North American, played an important role in the biotic interchange during the early Cenozoic, so the Eocene. Dispersal via the land bridge more recently than the Eocene or Oligomycene has been all but impossible, assumed all but impossible, right? So that's the sentence that he pulls, and that makes his point. He goes on to say, so how did this new fossil get from Europe to Devon Island? To answer that, evolutionary paleontologists claim that the NALB somehow still existed, allowing animal dispersal from Europe to North America as late as the Miocene. So I saw that and I was like, okay, I'm just going to go to the quote. So I got the quote. I went to the paper. Here's his quote. While paleontologists recognize that the NALB played an important role in biotic interchange during the Cenozoic, dispersal via the NALB more recently than the early Eocene has been all but assumed impossible. And then they provide a reviewer here. But the next sentence says, recent geological and paleontological data, however, suggest that the NALB was not fully submerged until the Oligocene or even the early Miocene, with some portions submerged as late as the Quaternary. So I don't know why the quote was cut off there, because to me, this next sentence is answering the question, not just how they did it. They're saying that they've got data that actually support it, which I think violates this idea that they just claimed that they managed to get there. Like these authors are saying that they actually have papers. And in fact, all of these citations here, 34, 19, 37, 44, 46, and 56, all of those are like 2014 and younger, or excuse me, older. So like they're at least 10 years old by the time this paper came out. So what I'm saying is that I don't think that it's been consensus for quite some time, at least according to these authors, that this land bridge was definitely underwater. It seems like the authors are saying, no, recent geological and paleontological data are saying that it wasn't fully submerged, which would have provided a way for these rhinos to get there. So So I don't know why it was cut off there. And I also don't understand why he says or implies here and then later on that there's just not a good reason why they would suggest it, that it's just evolutionists trying to make sense of the data. Like, this discovery was last year, but all of those citations are again from 2014 and earlier. So to me, that says that we've known for quite some time, or at least suspected for quite some time, that this land bridge might have been above water. And then we find a rhino, and it's like, well, okay, it probably was. Since we have all this other stuff that's saying that it might have been, this is probably good support to say that it was. Like, I don't like this line. To justify this impossible interpretation, the research team found 6 other Miocene mammal fossils in both North America and Europe that hinted at the late dispersal over the NALB. But that's not the only reason. The next sentence specifically says that also they look to recent geological and paleontological data. So again, I'm willing to think maybe I'm missing something here, because I read his article a couple of times and I read this paper once. Mm-hmm. But I skimmed the method, so maybe I'm missing something.
So I am encouraged that that the paper actually does use the word impossible. I was a little bit bothered by that, because he was putting the word impossible in quotes as if they were saying that word. So I do feel better about that.
Yeah, they use the word impossible for sure. But what I don't like is that they say, while it was assumed impossible, the next sentence mm-hmm. says, however, which seems to suggest to me that they don't think it's impossible.
Yeah. Now, does the paper suggest that the rhino might have got there from like riding on a piece of ice?
I don't think so. Because to me, when I read this, I don't know, but when I read this sentence, recent geological and paleontological data suggests the NALB was not fully submerged. So the North American land bridge, my understanding is that it's a land bridge, not an ice bridge. And when they say that it wasn't fully submerged, that's telling me that they're saying a land bridge was not fully submerged. So this tells me that, unless I'm reading this wrong, they're saying that recent geological and paleontological data suggests that this land bridge was at least partially above the water until the Oligocene or even the early Miocene, which would have been right when this rhino is dated to, 23 million years.
Mm-hmm. Okay, yeah, I might have misheard. Again, this is the problem with me not reading the paper.
It's okay. And I don't know. I mean, to be honest with you, like, I kind of have a problem with the phrasing of this article in general. Like, articles, regardless of who's writing them, tend to be sensationalized. That's true. So, like, and I don't know Tim Clary, but like, I don't like where this quote was cut off. Mm-hmm. Right? Like, I do my best when you can notice this hopefully you notice this about my presentations here, but like, when I take quotes, I try to take pictures of them and be like, here's the full thing so that you can go read the rest for yourself. Because I wouldn't have even known that the recent paleontological and geological evidence suggested that there was a land bridge still. If I'd only read Clary's article, I wouldn't have known that. He gives no indication that there might be one.
Yeah, by the way, I consider this out of place because a rhino can't survive in the high Arctic.
I got that next.
Okay.
Next slide. He says, second impossibility is the latitude. A second impossible claim by this article is that the high latitude environment where these rhinos were supposed to be living. He says, according to conventional science, these rhinos were supposed to have lived where their fossil bones were found. I agree, on Devon Island in Canada's high Arctic. In addition, plant fossils found with the mammal bones indicate a temperate climate. The team wrote, the presence of the rhinoceratid, I always thought it was rhinocerid, rhinoceratid in the high Arctic represents a paleoecological puzzle. Though the flora of the Houghton crater where the fossils were found suggest a temperate paleoclimate, there was a near-modern light regime with months of winter darkness. That's compelling, right? He says, so how do they explain this conundrum? The authors don't. They simply appeal for the need for further research. So I went to this quote. This one was a lot better, I think. Here's the quote. The presence of the rhinoceratid in the high Arctic represents an interesting paleoecological puzzle, though the flora of the Houghton crater suggests temperate paleoclimates, blah, blah, blah. You get it. Months of winter darkness. But the sentence prior says the presence of these types of animals, rhinoceratids, in the western low Arctic during the Tertiary, so same time period, is apparent based off of enamel fragments that are not identifiable to the genus or species, but they're definitely rhinoceratid. I'm just going to keep saying rhinocerid. People can deal with it. The much higher latitude occurrence of this new specimen reported herein is represented by a near complete skeleton. So what they're saying here is that the new skeleton cinches it. Definitely, they were at these high altitudes. Agreed. And the sentence after says, oh,
maybe not. Sorry,
I'm saying they were buried there for sure.
Okay.
Fair enough?
Yeah, for sure.
Okay, because that's where we find them. I didn't mean they were living there. I'm saying they were buried. Fair enough. They go on to say analysis of paleo diet and migration patterns using tooth wear and isotopes may in the future illuminate the ecological strategies of high latitude herbivores during the period of deposition of the Houghton Formation. So to me, as somebody who, my master's degree was in primatology, and specifically was primate biology, behavior, and conservation, which included a lot of ecology. So the study of how animals live and thrive in their environments. And when I saw this, I was immediately like, actually, when I read Clary's sentence, I was like, that's a really good point. I wonder how that happened. Like, how would they have survived if that's actually where they were living? And then I thought about it for a minute, and I was like, okay, are there any animals that live in these areas today? And like, of course, I thought of seals and polar bears and penguins, but those guys aren't big herbivores like rhinocerodids are. And then I thought about reindeer, and I thought about musk oxen. And musk oxen and reindeer, these guys here, they actually migrate over the course of the year. So they go to places like Devon Island. I don't think they go to Devon Island specifically because obviously you can't get there now, but they go to areas as north as Devon Island, and they graze on the grasses that sometimes grow there, some of the foliage that grow in these high latitude places. Similar ecosystems, I should say. And then when it gets dark and there's no plants that can survive and the plants all die off for the season, they go back south again. So I thought to myself, I was like, okay, well, what did Devon Island look like? What does Devon Island look like now in the summer? And this is a picture that I got. It's covered in grass. So Devon Island right now, when it's as cold, like right now it's colder global climate-wise than it's proposed to be in the Miocene by a lot. And so it would have been, if like temperatures and the plants that grow there are consistent now as they were in the past, if the earth was a lot warmer during the Miocene, there would have been even more plants than there already are here now in the summer. That's the logic that I'm following. So then I went back and I read the sentence, and they say analysis of paleo diet and migration patterns, specifically migration, which is like, I was like, oh, duh, because they're proposing here that these animals migrated. They don't think that they stayed there during the winter darkness. Using tooth wear and isotopes may in the future illuminate the ecological strategies of high latitude herbivores during the specific period of deposition at the Houghton Formation. So they are not saying what Dr. Cleary is saying they're saying here, that it's like, oh, we'll do future research and figure out how this is possible. What they're saying via this quote is that future research is going to illuminate the specific type of strategy that they used when they were present, because they migrated, because if they could get there, then they could leave. And I think it's really telling that the fossil that's there is dated to a time when these authors are proposing there would have been a land bridge way to get there, because that means there was a way to leave too. So I don't find that to be a compelling, I don't find this to be an impossible fossil, in the same way that I don't find reindeer living in areas like reindeer for sure are dying in the Arctic now, right? Like in the summer. But if you went into the future and found a fossil of them there, it would be kind of silly, I think, to say, well, this is an impossible fossil because it would have been dark in this area for half the year, but don't live there during half the year. So I don't, and this is where my ecological background comes into play. And maybe because Claire is a geologist, like that he's not thinking in that way. I'm sure there are geology ways that he thinks that I wouldn't even begin to wrap my head around, because I'm not a geologist. But like what animals actually do does matter. And here's the slide that I was mentioning earlier. Why are the out-of-place fossils always, and I'm reusing this from my Tiktaalik or from my tetrapod presentation, but updated for this rhino, why is it always out of place from here to here? Why is there not one single out-of-place fossil that's from here to here, right? Like, and again, I want to be clear, this rhino is not an out-of-place fossil. Not by the author's conception, not by my conception reading Dr. Clary's article, like I don't think that case can be made. What do you think about that?
I got to look into it more. I can't. One of the biggest problems I have is how much speculation goes into science, unfortunately.
Yeah.
Which is, we're speculating that there was a land bridge. We're speculating that it crossed the land bridge. We're speculating that there was food for it to eat. It's just difficult for me. It's like, yeah, that's all. Like even surviving in that area in the summer to me would be difficult for an animal like that, to have months of 24 hours of sun.
But animals do it now.
Yeah, I don't know.
Right? Like reindeer and musk oxen do it now. And like, to be clear, what they were eating, Clary quotes. He says, when he's quoting the authors, that the authors said there was a temperate paleoclimate via the flora, which means temperate paleoclimate. That's analogous to where the Midwest is now. The Midwest right now has a temperate paleoclimate. Right? It's not Arctic. So what they're saying is, he's quoting the authors saying that the types of plants that are there are similar to modern day Indiana. So it's not, it wouldn't have even looked like how horrible it is for musk oxen and for reindeer. It would have been lush. And that's not speculation. Those are the plants that they find there. And like, I don't know, like I get where you're coming from on speculation, but like, I don't know that you can make a better case for what an ancient environment was like than like the plants that grow there and the animals that live there, especially if you've got modern analogs.
Well, yeah, anyway, I mean, yeah, yeah.
When ask Dr. Cleary.
Yeah, I will. I will for sure.
But I do think, yeah, I mean, I think you just think on it, because like, again, we have animals that do this in way worse conditions today. And I don't think it's, so I guess what I'm saying is,
but they're covered, right? They're like covered in fur and stuff to stay warm.
Yeah, but it would have been temperate. Right? Like we have animals that live in areas that get really, really cold now, who have very little fur or no fur at all. And with these guys, like I guess also too, there were woolly rhinoceroses. And we know that because we have their frozen remains. I think we have frozen woolly rhinos. So it's like some rhinos did have hair. And I don't know about this one, like the support that they have for this, but like, I really don't think, like, okay, let me give you an example. There are rhinos at zoos in the Midwest right now. And they don't, like, they can go out during the summer. And they can, like, a summer temperate climate is not hard on a rhino. Just think on it.
Okay, for sure. I appreciate it.
Yeah, I mean, I didn't have a lot of time to dive into it, but that's my 2 cents. Right. Okay, so today we're talking about humans. Conventional science for human evolution makes like a pretty simple case, right? Like I'm really oversimplifying here, but conventional science argues humans are physically apes or hominoids, which evolved from earlier apes over about 7 million years of time. So what should we expect to see if this is the case? And I will note that this is grossly oversimplified by the March of Progress, but I'm using it here because I think it's famous for a reason. It gets the idea across. So if that's the case, humans should be demonstrably apes now, like hominids now, physically and genetically. That means we should cluster with them more than anything else. And I would even say we should be more similar, if this is the case, if we really are related to other apes, we should show up as more closely related to apes than pairs of animals that a creationist would accept as related, right? So I think we have to keep a standard in science. I think that's fair. So I would say that like if 2 animals, like let's say somebody proposes that lions and tigers are related, or that grizzly bears and black bears are related, right? If you sequence their genomes end to end and they show up as, I don't know, 95% similar, and then you do the same thing for humans and chimps and they show up as 96% similar, you can't say that the former pair is related and the latter is not. There has to be a standard for relatedness. So humans should be apes now, physically and genetically, for human evolution to be true. And then the emergence of the traits that make humans unique from other apes, because we are unique compared to every other kind of ape now and in the past, those should show up slowly over geologic time in the fossil record from these more primitive forms. Can we agree that this would be good support for human evolution?
Um,
And if not, what would make the case better?
yeah, I'm struggling. I mentioned this last time, but I'm struggling with the extrapolation of humans being 99.9% similar and then extrapolating that out to everything else. That to me seems a leap too far.
So I hear you, but let's try this. In your opinion, are golden retrievers and German shepherds related?
Oh, man, I should have looked into this. You asked me this before. I Yeah, it's a good question. I don't know.
Like dog breeds. Do you think dog breeds are actually related?
I think some are, yes, but I don't. I haven't looked. Here's the deal. I haven't been convinced, but I haven't tried to convince myself that all dogs came from like a dire wolf.
What about this? How about cats and tigers?
Hmm.
Do you
Well,
think those are related?
when we say related, what does
Like, do they
that
have a
mean?
common ancestor?
I don't know. That's a good question. I don't know.
Do you have any two species that
Mhm.
aren't that you would accept as actually related to each other?
Hmm. That's a really good question.
Because earlier
Um,
you said falcons and hawks.
yeah, I know, and I should have asked you to clarify what we were talking about related. Uh,
Yeah.
I don't know if those are separate quote unquote kinds or not. I don't know.
How about
But
this?
I should be able to come up with something right now that I think is related for sure. I don't know why I'm drawing a blank.
What about grizzly bears and polar bears?
That's a good. I can't necessarily say yes.
Maybe African and Asian elephants?
I can't picture them, but that sounds fine.
Hmm. Okay. Well, how about this then? Is it fair to say that if we're being consistent, if someone says that two species are related, like
Hmm.
let's say, and I'm not setting you up for a gotcha, I don't have the numbers for this. If someone wants to say like black bears and polar bears share a common ancestor,
Hmm.
and if they show up as 95% similar,
Hmm.
and somebody says we know that polar bears and black bears are related to each other, whichever pair I said,
Hmm.
of the two bears, that we know they're related to each other because they share 95% of their DNA.
Hmm.
And then they turn around and they look at humans and chimps, and human chimps show up as 96% similar.
Hmm.
Is it standard to say then that humans and chimps are not related, despite the fact that they have a higher percent similarity than two bears do?
That wouldn't make sense if you are taking the position that the bears are related.
Perfect. That's all I want to put
Okay.
out there. And I
Okay.
would just, so that's all I'm saying here is I'm saying
Hmm.
that, like, okay, if any two species are related and like share a common ancestor, if any two species share a common ancestor, and if genetics plays any role in that at all, then that sets us up for a standard that we can assess other pairs of organisms. And we
Hmm.
can set a standard for like, okay, you have to be, minimally this similar to have a common ancestor from a creationist standpoint. From an
Hmm.
evolutionary standpoint, they're all related, right? But that percent similarity does tell you how close something is. So, for example, if I share 99.9% of my DNA with my sister, and then 98% with a chimp, and then 95% with a monkey, and then 90% with a cat, it's fair to say I'm most closely related to my sister, and then a chimp, and then a monkey, and then a cat. Does that
May
same
Maybe
page?
I'm not there yet.
I'm saying from an evolutionary perspective.
Oh, definitely. Yes.
Okay. It's just like those percent similarities like
For
translate to
sure.
related. Cool.
Yeah.
Okay. So, and then is it also fair to say that if human evolution is true, then we should see the traits that make humans unique in the fossil record?
Mm. Yeah.
Awesome. All right. So, You're going to agree with most of this. After your Linnaean opener, I think you're going to agree with most of this, but we got to go through it anyways. Humans should be demonstrably apes or hominoids now physically. Let's start with that. I love these two side-by-side comparisons because this left one is a drawing made by Huxley, who was Darwin's contemporary. And then on the right, we have the actual skeletons of a human, a gorilla, a chimp, an orangutan, and a gibbon, and they're all standing up. And I like this because it shows you that these guys have all of the same bones. They have femora and humeri, I guess, the sternum in the front, the same types of bones that make up their skull. Sizes and shapes are how they differ, especially between like gorillas and chimps or chimps and gibbons. You can see the size and shape of the bones changing. What are humans?
It's a little misleading that they're standing, but I'll let that slide.
I think it is, but if you want to compare the bones in an easy way, you have to put them in an anatomical position. So like if you wanted to compare the bones easily between a human and a dog, you would do the same thing. However, there is an important caveat here, which is that dogs can't walk upright habitually. They can't do it. You can train a dog like a circus dog to try, and they can do it a little bit, but like they develop pathologies, one, and two, they can't maintain it.
Right.
Chimps, orangutans, gorillas, and gibbons all walk upright in the wild on their own a lot. So while it's misleading to, no, I wouldn't say it's misleading at all. I would say that it is potentially a biased representation of these skeletons, but I wouldn't say that it's misleading because they do move like this sometimes in ways that other animals don't.
And then they also kind of made them the same size, right?
No, the ones on the right are to scale.
I mean the picture.
Yes, the one on the left is scaled. Or is not scaled. Or it is scaled. Sorry, the gibbon is huge, and they made them all look the same. The actual size of the gibbon is the right over here.
Okay. Got it.
Okay. What are humans, right? We can answer this in a lot of ways. We can say, what are they physically? We can say, what are we philosophically? We could say, what are we spiritually? But science can only answer the physical one. That doesn't make the other questions unimportant. It just means science can only weigh in on the physicality of humans. So what are we physically? This has been a question that's been on our minds for millennia, a few notable names in the West. We've obviously got Plato, who defined humans as a featherless biped. And then Diogenes came in and he was like, behold a man, and he holds up a plucked chicken, like revealing that featherless biped isn't a great definition for a human. Then Edward Tyson, prior to Linnaeus in 1650, published, like 50 years prior to Linnaeus, published The Anatomy of a Pygmy, which was a section of a chimpanzee. And what he showed, interestingly enough, which this was kind of radical for his time, but what he showed is that chimpanzees shared more with humans in all of their anatomy, including the brain regions, than they shared with monkeys, because he did a three-way dissection to show the similarities. He pitched this, and people were like, that's kind of weird. And then Linnaeus, of course, comes along with taxonomy to categorize animals. Now, despite being a creationist 100 years prior to evolutionary theory, on the basis of physical characteristics, Linnaeus groups humans and other primates together in a group called anthropomorpha. And you've heard this quote before, but Linnaeus is writing to his friend Gmelin here in 1747, and he's like, it doesn't please you that I've placed man amongst the anthropomorpha, perhaps because the term means with human form, but man learns to know himself. He says, let's not quibble over words. It'll be the same to me, whatever name we apply. But I seek from you and from the whole world a generic difference between man and simian that follows from the principles of natural history. So he's talking about a physical difference. He says, I absolutely know of none. If only someone might tell me a single one, I would have called man simian or vice versa. I would have brought together all the theologians against me. Perhaps I ought to have by virtue of the law of the discipline. So he's basically like, I don't know what you want me to do. He's like, if you don't want me to put humans with other apes, then what you're going to have to do is provide me a reason to do so. He also had some really interesting things to say about how what animals are. This is a quote that he writes in 1733, before the prior one. He says, one should not vent one's wrath on animals. Theology decrees that man has a soul and that animals are mere automaca mechanica, but I believe they would be better advised that animals have a soul and that the difference is of nobility. This is over 100 years prior to Darwin. Darwin would say the same thing 110 plus years later. And And this is notable, of course, given his creationism. All right, but what actually makes humans physically animals or mammals, primates, apes, etc.? And the reason I always start with Linnaeus is to show that like evolution didn't make people consider humans apes, right? Like evolution comes along 100 years later and tries to explain why we look like that. But for a long time, again, most systematically outlined by Linnaeus, the physical characteristics were the physical characteristics. Okay, I want to run this exercise that we've run before, but I want to run it with gibbons instead of humans. And I want to ask the question, where do chimpanzees fall? Right? So all of the animals in the box are eukaryotes, and they're eukaryotes because they have a true nucleus bound by a membrane and specialized organelles. What that means is that chimps are eukaryotes because they're in that box. Chimpanzees are composed of animal cells, and their animal cells have like organelles and a nucleus. A paramecium, as depicted down here, is also a eukaryote because even though it's single cellular, it has those traits too. Chimpanzees and all of the other animals in the box belong to the kingdom Animalia. And the reason is because they're multicellular, they form a blastocyst, at least during development, or a blastula, and they're heterotrophic, etc. So like obviously snails, fish, snakes, baboons, skunks, chimps, and gibbons are multicellular. I don't think I need a picture to explain that. But this is their blastula, or blastocyst rather, at the same stage in a mouse, a cow, a rhesus monkey, and a chimp. This is one of the characteristics that defines being an animal, because animals organize their bodies in similar ways in accordance with specific genetic cascades. And that involves forming this blastula or blastocyst really early on in development. They're also heterotrophic, which means they can't produce their own food. They have to eat it, either by grazing or by foraging or by hunting. So all of these guys are also chordates. To be a chordate means you have a notochord, you have a hollow nerve cord. By the way, a notochord is sort of like an early version of a spinal column, right? It's an early version of a backbone. They have postanal tails, they have pharyngeal slits, and they have a thyroid or an endostyle. So here is a chimpanzee's neck. This poor guy has a tumor and is having a scan here to show where that tumor is. But in this scan, you can see both the vertebral column. So there are the different vertebral bodies of its neck. And then you can see its spinal cord too. And so that tells us that chimpanzees are chordates because they have a spinal cord located within a vertebral column, which is advanced notochord. They're also, they've also got this postanal tail here. So chimpanzees have a coccyx or a tailbone that is about as long as what you see in a human. In fact, I think they actually have the same number of tailbone vertebrae as us. But during development, chimpanzees and frogs for that matter actually have a tail, like a vertebral body that starts extending beyond the anus that then, much like the limb buds in a dolphin, terminates. And it pulls back and the rest of the body develops around it, and they don't end up having that postanal tail during adulthood. Frogs don't and chimps don't. But early in frog development, you can see it in their tadpole form too. They start with a tail and then they lose it. With chimpanzees and with other apes and really tailless animals in general, that terminates early on during their development. And of course, they have a thyroid. And this is something interesting that I learned when I was reading about thyroids in general. The thyroid is derived from the endostyle. So the reason why you have thyroid/endostyle is because there are a couple of genes that turn an endostyle into a thyroid. And what I mean when I say that is that when you sequence the genes for the development of that particular organ in non-vertebrate chordates, lampreys, hagfishes, and then like jawed animals, you have endostyle, which is the primitive version in these like squishy looking chordate guys, but lampreys start their development with an endostyle that then through a genetic cascade become the thyroid. And then it's a thyroid, starts as a thyroid in gnathostomes, but the genetic setup is the same. So this is the reason why it's thyroid/endostyle, is because it's a structure that is similar in these animals, but not identical. So then these guys are obviously all mammals. Fish and snakes aren't mammals. And the reason they're not mammals is because they don't have what chimps and other mammals have, which is the ability to breastfeed their young, hair, 3 ear bones, and a dentary squamosal joint, which is a specific way in which you call it the TMJ in a lot of mammals. It's the specific way in which the hinge of the jaw meets the cranium. So then it's like, okay, well, these guys are all primates, right? They have characteristics that primates have. Skunks don't have the characteristics that primates do, which include forward-facing eyes, which you can't see in this horse or in this rabbit. A post-orbital bar, which I'm going to come back to this here in a second, which all primates have, but no non-primate has, like how primates do. Large brains for their body size, and dexterous grasping hands and feet that are capped in nails instead of claws. So these are characteristics that unite primates. If it is a mammal and it has all of these characteristics, it's a primate. And I have some little guys here to show you. Hold on just a second. What I mean, because I think it's kind of cool. I have some props. This is a big deer skull. And on this deer skull, you can see, one, that the eyes point side to side. When their eyes are actually in their sockets, they angle outward this way and they angle outward that way. And that eye socket, while it closes in the back here, it doesn't actually have that post-orbital plate development, which that's actually a bad example. So more on that in a second. The cat is probably what I should use for post-orbital bar. This is a little cat skull. And if I take my finger or this pen rather, and I go through the side of that eye socket, hopefully you can see, you can just run your pen through there, right? Like this eye socket isn't closed. And it actually is closed in the deer, which I didn't know that. Deers have a post-orbital bar. Who knew? They don't have a plate, which characterizes the next group of animals that we'll talk about later on, sort of, but like that post-orbital bar is missing in most mammals, I guess, excluding deer. I've got like so many skulls over here. None of them have it. It's classic that I picked the deer. But this is the most primitive of the primates that I have, which is a lemur. And the lemur has it too. So the lemur has that full closure. And it's got the large brain for the body size. They've got grasping hands, of course. Their eyes face forward. Same thing with this gibbon, closed orbit on the eye, right? Forward-facing eyes. Same thing with every primate skull that I've got in here, even the itty-bitty, teeny tiny little tarsier with its giant forward-facing eyes that do, in fact, close. What's cool is that the next stage is actually a post-orbital plate, which is that full closure. So in the lemur, that's the cat. In the lemur, you can take your finger and stick it all the way through. Even though there's that bar, I can take my pinky and hook it through the eye socket to the other side, like over the post-orbital bar. See that there?
Mm-hmm.
In all monkeys, and humans are included in this group, the whole socket is closed off. If you stick your finger into the eye socket, there's nowhere for your finger to go. But in tarsiers, which are proposed to bridge the gap between lemurs and lorises and higher primates, the gap is partially there. It's mostly closed, but not all the way, which is neat. Anyway, so those are the characteristics that define primates. And then you have the characteristics that define apes. Skipping past this, which are a really large brain for your body size, slow development. All apes take a long time to grow up, in part because of their big brains. A large, broad chest. So you can see that the gibbon and the chimp over here have a really wide chest, whereas this macaque monkey has a deep chest, like a dog does. And the reason is because monkeys spend most of their time on all fours. Monkeys like macaques, I should say. They have long arms for their body size. And they have a special molar pattern called a Y5, which is unique only to apes. Finally, chimpanzees are obviously chimps. And the characteristics that define chimps amongst the apes mostly involve their specific nature of knuckle walking, the types of tools that they use, or their capability as reference to their brain size, their omnivorous diet, their polygynandrous mating system, and their fission-fusion social system. And a bunch of minute linear skeletal dimensions that you could use too. So chimps are cool, right? They're members of the species Pan troglodytes. They use all kinds of tools. They hunt with spears. They termite fish. They crack nuts. They eat meat, which they hunt for in groups. They walk upright sometimes. They walk on their knuckles sometimes. And they have these really complicated social systems. So then chimps are a type of hominoid, which is a type of eukaryote, which is a type of mammal, or excuse me, which is a type of animal. Let me back up. Hold on. There we go. Chimps are a type of eukaryote, which is a type of animal, which is a type of chordate, which is a type of mammal, which is a type of primate, which is a type of hominoid, and they are members of Pan troglodytes. So the reason I did this is because by the same way, by the same set of characteristics that land chimpanzees as apes, humans end up as apes, right? Humans belong to the domain eukarya because we have the same characteristics that chimps have. We're made up of animal cells that have a nucleus. And we're members of the kingdom Animalia because we're multicellular. We form a blastocyst during development, and it took me forever to find this picture. In mammals, it's called a blastula. We also have to eat our food. We can't just absorb light. Of course, we're members of the phylum Chordata because we have a notochord. That CT scan here on the bottom left, you can see the vertebral column and then the spinal cord coming through it, just like we saw with the chimps. We have that postanal tail during development. We have a thyroid, just like every other chordate does. And we have the pharyngeal gill slits, or pharyngeal arches, if you will, as we develop. We are mammals. This is not controversial. Like we've been over this before. We obviously breastfeed our young. We're covered in hair. We have 3 inner ear bones, and we have the same type of jaw that a chimp has. Finally, we're primates, right? We have dexterous hands, binocular vision, big brains, postorbital bar that becomes a postorbital plate, and then nails instead of claws. So I'm using myself here, right? There's our forward-facing eyes. That's not my skull. There's our forward-facing eyes, again, as well as our postorbital bar/plate. There's our hands, and of course, our really big brains. And then humans are members of the ape family, or Hominidae, because we have really big brains for our body size. There's humans compared to a bunch of other apes. We take forever to grow up. We stay with our parents for a long time. We have broad chests instead of deep chests, and long arms for our body size. And we also have that Y5 molar pattern. So for the same characteristics that land chimpanzees as apes, if you take humans out of the question, that's why humans end up being hominids, or apes, rather hominoids. And then humans are unique amongst the apes as well, right? We walk upright. We have huge brains. We use really complex tools. We have little bitty canine teeth, and we grow up like extra slow. So some of the characteristics that make humans unique amongst the apes. So you roll it back, and you end up with that nested hierarchy. Same old, same old. So I would propose, like, as you're thinking about this, like, that the differences within Hominidae, physically speaking, or hominoidea rather, they're not greater than many other animal families. And the reason why I think that's important is because, and I know you make up your own decision on this, but like Answers in Genesis, they would say that Felidae, the whole cat family, is related, that they share a common ancestor in the form of a cat or cat-like thing that was created in the Garden of Eden. So it's like, when I look at the diversity in cats today, like the shapes that they take, the dimensions of their bones, blah, 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 I see that and I'm like, that is not, like, just brutally, that is not more extreme than what you see in apes. So I don't know really what to make of that. So I think it's unsurprising when you consider the entirety of the organism that humans are physically hominids or hominoids, depending on whether you want to go with the apes or great ape classification. Can we agree that physically humans have those characteristics?
Yes.
Great. All right. This is why Linnaeus categorized humans like he did. This is one of his contemporaries, Buffon, who worked sort of alongside Linnaeus a lot of the time. He says, although the works of the creator are in and of themselves equally perfect, the animal is, according to our way of perceiving, the most complete work of nature, and man is the masterpiece. So he's like, humans are animals, but we're the best of the animals. I learned this while I was researching. Apparently for one edition of Systema Natura, which is Linnaeus's book, He actually categorized chimpanzees and orangutans as members of genus Homo. He called them Homo troglodytes and Homo sylvestris. I thought that was crazy. He looked at them and he was like, they're so similar physically that we actually have to put them in the same genus. And he would walk that back because it was really unpopular. People did not like that. So he just kept them at the family level. Okay. So modern data sets confirm this. I found this really interesting. Apparently, at a lot of zoos, human doctors perform the regular, or not the regular, the unique surgeries that apes require. So, for example, in these 3 different headlines, human doctors were called in to help this baby chimpanzee who had broken her leg, to help this Siamang who had, I think it was a gastrointestinal issue, and to help this gorilla with a mastoidectomy. And a mastoidectomy had previously only been done on humans. They did the exact surgery that you do on humans, just on a gorilla, because all the bones are in the same place. And it was successful. This gorilla was fine. So Darwin would obviously take it further. By his time, the Earth is considered quite old in conventional science. And then him and Wallace successfully showed that populations of organisms change over time and species converge on common ancestors. There's a lot of change in the present, plus a lot of time in the past. So organisms were probably changing for millions of years. Humans thus weren't just the same type of animal as chimps with a common designer. These guys would argue that they actually share a common ancestor sometime in the past. That's the logic. But making this case required 2 things science didn't have. The first was lots of fossils, and the second was genetics, right? To actually make the case that this is common descent rather than common design, you need both of these. You need the record of the physical forms as they change through time, and you need the genes. So interestingly, the fossils were on their way. So Darwin, Huxley, and Lyell had all separately predicted that human fossils were going to come around eventually. When they were living, dinosaurs and marine reptiles were just coming to the forefront of the public's mind. But specifically Lyell, which is funny because Lyell would never fully accept evolution. He was like, we're going to discover extinct varieties of man. Eventually, with all the fossils that are coming to light, we're going to find something, some earlier form of humans. But before Darwin died, we actually found the very first hominin. And this is one of the members of that species, Neanderthals. This is actually the specific skull that Darwin got to see Gibraltar 1. More on that later. And as for genetics, right, fossils are on the way, but genetics is just coming to the forefront. So humans are categorized as apes physically. What about genetically? Okay, genetics refresher. This guy is responsible for the discovery of genetics. Gregor Mendel, he's an Augustinian monk, and he does his work in his abbey in the Czech Republic in between 1856 and 1863. He does it by breeding pea plants. And the entire purpose that I'm bringing this up again is because what Mendel showed, outside of just that genetics is a thing, is that inheritance is predictable. There are laws at play. Biological laws are governing how characteristics are being passed from parent to offspring. Genetics, the study of genes, genetic variation, and heredity, is going to provide the link between biological organisms that unites them into this larger scale family tree. So, over the next century, we find out that biological information that Mendel was basically recording is passed from generation to generation via DNA. The information for the making and maintenance of you, it's in your DNA. So, multicellular organisms are made up of cells. And then inside cells, there's an organelle called the nucleus. And inside the nucleus, there are a bunch of chromosomes. Those chromosomes are tightly packed bundles of DNA. And those DNA can be unstrung, and sections of it can be isolated. And those sections are where your genes live. So genes are found in DNA. DNA is wound up tightly to form your chromosomes. The chromosomes live in the nucleus. The nucleus is found in almost every type of cell that you have. And that's how multicellular organisms make and maintain themselves. So DNA is this double helix, right? And it contains the information, genetic instructions, for the making and maintenance of you, as well as the instructions for making more of itself. This is really important, right? The fact that DNA replicates is how we tell how organisms are related. More on that in a second. So the genome is all of your DNA. It's the full set of genetic instructions and information that any given organism has. And it can be broken into coding and non-coding portions. A very small fraction of the genome is coding. That means it contains the recipes for making proteins, so like making specific receptors, making actin and myosin for muscle repair, making insulin, that kind of thing. Important stuff for making you and then keeping you moving. However, some of the non-coding stuff is important for regulation, for sure. At present, though, most of the genome, like over 50%, and it's by quite a bit, has no known function.
Quick question.
Yeah, go ahead.
I'm forgetting now because it's been so long, unfortunately. But immediately, the underline there, most has no known function, seems to contradict with what I heard about a while ago with encode.
Yeah. So encode defines function as being transcribed. That definition, they actually walk that back themselves as being too liberal, because just transcribing something doesn't mean it actually has a function. They say, okay, no, that's actually not a good definition for it. That happens at about 2016. But then we have to remember that ENCODE's original estimate for the functionality of the genome, even with that most liberal definition, was that it was 80% functional. So 80% transcribed. So at the very least, there's 20% that everyone basically agrees has no known function. But later, after ENCODE, several different researchers would come out and say, okay, but what's the limit? Like, how do we know that 80% isn't actually doing anything? And through experiments and also theoretical limits of how DNA sort of replicates itself, the upper limit was proposed by Grauer and colleagues. Actually, I think it might have been Grauer himself, and then later with the co-author in 2017, was about 25%. So he would say that stuff that actually has a function, meaning it contributes to regulation or protein coding, the two main things that make and maintain you, you're not going to get above 25. And at present, we don't know of a function for anything more than, I think the number's 12. Dan is the one that did all the work on this for sources.
Okay.
But even if you go with ENCODE, 20% is non-coding. And they don't think that definition should hold.
You mean not non-coding?
Non-functional, excuse me.
Okay, got it.
Non-functional. Sorry.
Okay.
Sorry.
That's okay.
The amount of the genome that's non-coding is 98%. But non-coding DNA can still do important stuff. So non-functional, meaning it doesn't do anything that impacts development or physical characteristics or reproduction. It seems like it's just there. And part of the reason why I think they're pretty sound in that is that in some organisms, you can knock it out. Like you can just take it out and they can just reproduce, no problems. There's another reason that we'll talk about here in a second.
Okay.
So DNA across the board universally links parents and offspring, right? There's a reason why you look like your parents. And it's not just because physical characteristics manifest out of nothing, right? The reason is because you receive DNA from your parents. That's how you're made. You are a combination of your parents with a couple of tweaks that are unique to you via recombination and mutation. This is why paternity tests work. So in this little example here, you have a mom on the left and you have a child, and you're trying to figure out between two fathers who the dad actually is by looking at these bars are representing genes or sequences in a very variable portion of the human genome. Because humans in their full genome are pretty dang similar. In their portions of the genome that do stuff, we're 99.9% similar. It's very, very close. So you have to look in those areas that differ to run a paternity test. That's how it works for the courts. So they have this kid here, and the child shares a gene with father 2 here, 2 different genes with father 2, and then they don't have genes from father 1 or father 2 here, but that's okay. They might have gotten the mother's condition who lacks that characteristic, and that seems like it's the case. And then they share another characteristic with father 2, and another one with father 2, and another one with father 2. So overall, they share more genes with father 2 than they do with father 1, which means father 2 is the dad. That's how genetics works. That's how paternity tests work. So the question is why? Why does it work that way? Why is that a fair assumption to make at all? So cell replication happens trillions of times in your body every day. And when it happens in your, like trillions with a T, and when it's happening in your skin cells, that's mitosis, right? Like your skin or your muscle or your mucous membranes, your gastrointestinal system, those are body cells reproducing themselves. And they reproduce themselves with fidelity, ideally. But you also have to create your sex cells, so sperm and egg. And those are going to be haploid. That means they have half of the genome, only one copy of every chromosome instead of 2 copies, because they're going to combine with whoever you procreate with. So body cells have 2 copies of every chromosome. Sex cells have 1 copy of each chromosome. And the reason is because sex cells will combine during sexual reproduction. A haploid sperm meets a haploid egg, and they create a new organism with a novel combination. But offspring aren't just combinations of mom and dad. A kid differs from their mom and dad, even in the chromosomes they share with them, because of mutation and recombination. So recombination is the mixing of genetic information that's already there into a unique combination outside of just mom versus dad. Some of those chromosomes are going to exchange information in this crossing over event that happens during cellular replication. And then, of course, there's mutation. Mutation happens generation to generation. It's an error in the replication of the genome, and it's going to change the sequence as compared to the parent. This introduces new variation into populations every single generation, and natural selection can then drive evolution depending on how these new variants fare in any given environment. So here's how this works. And this is if you take nothing else from this talk today, I want it to be this next little demonstration here. Changes to genomes occur generation to generation in regions that everyone agrees are functional and in regions that everyone agrees are non-functional. So here's the yeah, go ahead.
Can you define everyone?
Creationists and non-creationists alike.
Okay. Okay, thanks.
Because if creationists are going to use ENCODE to say that 80% of the genome is functional, that's still 20% that isn't. So there is a portion of the genome that everyone agrees is non-functional. Are we on the same page with that?
I will take your word for it right now. Yeah, for sure.
Feel free to check me on it.
Yeah, I don't doubt you.
I could always be wrong.
No.
I could always be wrong.
Yeah, I don't doubt you. I just forgot. So I got to go back and look.
I could be wrong. I don't know. My understanding is that the way that they argue that the genome is really functional is using ENCODE, and ENCODE does not say the genome is 100% functional. So unless there's something else in the past couple of years that they've started to pull from, and maybe it's escaped me, that's my understanding.
Okay. Yeah.
So go ahead.
No, I was just going to say what comes to me instinctually, which I'm not saying anybody says this, but what comes to me instinctually is that I could imagine a creationist saying that there is the regions that we understand their function and the regions that we don't understand their function, and take a position that we just haven't figured it out yet.
I think that's true. And I don't know this, but I think that's probably how they get that rest of the functionality if they want to take the ENCODE position. But the problem with that is you can knock some of the genome out and not have any problems, right? Like, and moreover, individual humans differ in their genome size. Like your genome and my genome aren't the same size.
Oh, really? Yeah.
So the whole thing can't be functional because one of us would be having an issue. And I don't think either of us are. There's, again, another reason in a second. So bear with me here. We're going to look at this from a conventional standpoint, but it works either way. Like, no matter what, this is how inheritance works. So from a conventional standpoint, you've got the large and non-functional genome, right? Like, this is the genome in gray, and in this little bitty slip down here at the bottom, in this light gray section that I'm highlighting with my cursor here, that's the functional region of the genome. If you zoom in on that, it looks like this, right? I'm just telling you, here's the code, like the labels for the cartoon that you're about to see. You've got the large and non-functional majority of the genome and the smaller functional region of the genome. Even if you want to say that the genome is mostly functional, you're just flipping these, right? Like one is going to be bigger and the other is going to be smaller, but everyone agrees there's a portion of the genome, and to some degree, there's some level of functionality and some level of non-functionality. So let's say we have these two people. We have Mr. Blue here on the right, or excuse me, on the left, and then we have Mrs. Blue here on the right. And they have here, represented by these cartoons, the functional section of their genome, and then a zoom in of that non-functional section. And in our male here, he's got two mutations that are unique just to him, that happened just in him as he was being created from his two parents. And they're marked in orange. Whereas for our woman here, she's got two red mutations that are totally unique to her. And this is how it works. Everybody's got mutations that are totally unique to them. Most of them don't do anything. Some of them provide novel characteristics, like changes in hair color or eye color or whatever. So if these two reproduce, right, we're going to see what happens. You'll notice that when you look at these cartoons, they share 99.9% of their genomes here, but they differ just a tiny bit minimum. For simplistic sake, pretend this is reflective of actual reality, that it's just two differences, right? In these mutations that our guy has and these mutations that our girl has. He doesn't have the red mutations. She doesn't have the orange ones. So suppose that they procreate and have kids. The two children that they have are going to receive the unique characteristics that their parents had in this example here, right? Our boy here, our son, is going to receive both the mutations from mom and the mutations from dad. So in his genome, he has both of the mom characteristics and both of the dad characteristics in his own genome. And in addition, he has his own unique mutation in the form of yellow.
Question.
Yes.
Are you saying it's guaranteed he's going to get his parents' mutations?
I'm saying it's guaranteed that he is going to receive some of the unique variation from his parents.
Okay, I don't understand the distinction.
So in this example here, I'm using single mutations for simplicity's sake.
Mhm.
But there are characteristics that you receive that only exist in your parents. I'm not going to say it's the amount of it because I don't know. And it probably differs depending on which sperm you get from your dad and which egg you get from your mom. But there are going to be sequences of DNA that you receive from your parents from mutations that they've experienced that are going to end up in you that exist in nobody else. So
Okay.
for the purposes of this example, you're guaranteed to get these characteristics from mom and dad.
Okay. I apologize. I'm a little lost, and I feel like it's going to hurt us if we move on, and
Please,
I'm lost.
I'd rather us stop.
Okay. There are, I know so many parents where they have issues, which I would assume are from mutations, and their kids don't have these issues. Where's the
Right.
disconnect for me?
The disconnect is because you produce millions of sperm, and women produce many, many eggs. Not as many eggs as sperm, but a lot. And inside each of those gametes, inside of those sex cells, while they're overwhelmingly similar to each other, 99.9%, every single one of those sex cells is slightly different.
Really?
Because of the mutations that have occurred within them. That's how you get variation in a population. And so depending on which sex cell you get from which parent, certain mutations might not make it through. But because you're getting half of your material from mom and half of your material from dad, and that's combining to make your entire genome, you are going to get for sure some of their unique sequences. But it might not be a specific bad mutation.
Got it. Okay. I think it's making sense now. So it could be that dad doesn't have a mutation other than in his sperm. So he doesn't even know.
Or he might have it in, yeah, and it might be in some of them, but not all of them, depending on whether it's dominant or recessive. You see what I mean? So like that's the way that sexual reproduction and the way meiosis works.
I still have one confusion.
Yeah,
So
please.
if the child is getting something from a sperm cell, but it's only that sperm cell, that sperm cell has that unique thing, how are we tying it? How are we tying the child to the dad? How
because
could
you're
you?
because that mutation is unique to that child, not to the parent. That mutation occurred while dad was making sperm. So not all of your
Yes.
sperm are 100% identical to your body cells because they experience their own mutations in the same way
Right.
that you might experience a mutation in a skin cell and end up with cancer. But
Yeah,
you might end
sorry.
up with, yeah, go ahead.
My fault. I did not articulate that well. I get that it's coming from dad, but
Yeah.
how are we then connecting father and son if the mutation is in a sperm cell that the dad is not even aware of?
Like a characteristic, you mean?
Yeah, or just a genetic test.
So what I'm trying to say is that, like, let's say like you and your parent are going to share 99.99% of your DNA that does
Mhm.
stuff, right? Like
Mm,
mostly. And I'm being very specific here because
okay.
there's a problem later on. Um, there are areas of the genome like that are large copies of non-functional DNA that artificially will result in more differences that don't have an impact on the individual's physical form. It's messed up, and it has to do with what we're going to talk about later. That's rare from parents to offspring. So for all intents and purposes, you share 99.99% of your genome with your parent. In that 0.001% that you don't share, those are the bits that are unique to you. Those are
Okay.
mutations that have happened like upon recombination, upon mutation, and just due to the fact that you're a combination of mom and dad. And so when you run a paternity test, you're looking at areas of the genome that vary in people, but that are like they vary in people to the extent that it would be statistically nearly impossible for that specific mutation to happen between you and your offspring, like directly parent to offspring. And so what I'm trying to say is that you should, for all intents and purposes, without exception that I'm aware, share more with your parent than with a stranger, because you may
Yeah.
differ from a stranger at like 200 spots, but you might differ from your dad in only one.
Yeah, I think it just clicked for me. I think when we're looking at who's your parent, we're focused on the percentage of agreement, not
Right,
disagreement. Okay,
right.
that makes sense.
And
Okay.
especially relative agreement. So relative agreement versus someone else.
Mhm,
If you agree more with your dad than with a stranger when you line them up in these highly variable regions.
mhm.
And I apologize, this isn't my area, so I do my best with the genetic stuff, but I'm
Great.
sure there's people who explain it better.
Alright, we
So
can keep going.
for for example, right, like we've got mom and dad here. Dad contributes these orange mutations, and again, this is an oversimplification just for cartoon's sake. Mom is contributing these red mutations, and let's say both of these kids, they get the full Monty from both of those parents. They're also going to have their own unique changes because of the nature of sperm and eggs and the nature of recombination and mutation. So they've got the mom and dad pattern, but they also have their own unique mutations. In our boy here, in our son, we've got these yellow mutations. And in our daughter over here, we've got these lime green mutations. Now, both of them marry someone else. And that new person is going to have their own unique variation to add as well, right? Again, overwhelmingly, all of these people are really, really similar because humans share the majority of their genome because we're all humans. But there is little bits of variation in those areas that differ. And it is those areas that we're looking at when we make family trees. So these two pairs are going to pass on the inherited orange and red as well as their own mutations to their own offspring. So when they have kids, those kids are going to inherit the genetic markers in our example here, not just from their parents, but from their grandparents too. Those changes are maintained through the family tree. And so they've got markers from mom and dad, and then markers from grandma and grandpa, same on this side too. But if you sequence the genomes of all of these individuals here at the bottom, you're going to get this specific family tree where these two individuals that are siblings are going to share more than these two individuals that are siblings because their parents experience different mutations as compared to their grandparents. So they share a lot, but they don't share everything because of what uniquely happened along each lineage. So now they have kids. And if you compare the genomes in the functional and non-functional regions of these great-grandchildren, you're going to see that they share, all of them, the grandparent markers. And then they share a little bit less, like compared to each of these parents. And then they've got unique aspects from their own parents and then unique aspects for them specifically. So if you sequence the genomes of only these great-grandchildren, you are going to get this family tree. And it's not arbitrary. All of them share the red genes and all of them share the orange genes. These two individuals on the left share these yellow genes, again, from these specific parents. And then the green genes over here on the right. And then they have mutations that are unique just to them. Now, if you pull in a stranger, despite the fact that they share 99.9% of their genome, they're going to differ at all of these key locations. And so they're going to show up as least related to all 4 of these kids. That's how paternity tests work. Does that make sense?
Yeah, super fascinating. Thank you. It's
Yes. Sweet. I'm so glad.
It's cool. Yeah, it's great. I love it.
So to be clear, this is the schmanimals. It's just with genes instead of traits. And the reason why we trust genetics over morphology when the two are compared is because genes are recording actual procreation events, right? Like you literally can trace them through these copulations to the offspring and create this family tree. So then we have this question: if genes code for traits, why do physical trees ever disagree with genetic trees? So genetics always trumps physical similarity, even though they usually agree. And I do think that that's fair to say, although I do need to look into your exception examples. In the areas that I'm familiar with, like primates, they usually agree, and they usually agree quite well. Genetics unambiguously reflects relatedness in sexually reproducing organisms just because of how heredity works, right? So when you procreate someone else, you're combining your DNA, and then there's going to be unique aspects that end up in your kid that aren't in you or your partner, even though that kid shares the majority of that DNA with you and with your spouse. These relationships are usually reflected in physical traits, since the genome is responsible for morphology. This is why parents and kids usually look the same. In this picture on the top, these are parents compared to their kids. They look really, really similar, right? These two individuals here, same picture. They've like tried to take the same picture as they did when their parent was a kid. These two swimmers, these two, I think they're cops or army guys, and then these two youngsters. But there are some confounding factors. This is the reason why they don't always agree. The first reason is that physical traits can result from different genetic changes. So, for example, the genes that give you a specific eye color come from more than one place. So you can end up with a different eye color for completely different mutational reasons. Not always, not for every trait, but for many traits. And also, some changes are more common than others, right? The genome, not every area of the genome mutates at the same rate. So the best way to avoid these pitfalls is sequence the whole thing. If you sequence the whole genome, you get a 100% fidelity for who's related to who. And the only assumption that you're making is that heredity works the same way it's always worked, with zero exception. So for this reason, genetic similarity reflects relatedness, and it does so more accurately than morphology does in living organisms. So unique changes amongst billions of base pairs of DNA across generations can be mapped by sequencing the genome, and that reveals the relationships.
I would say that it works within species. I hate to use that term, but I think I have to right now.
I know, I love that because I agree. In fact, everyone agrees that I know of. Everyone agrees that it works within species. So why does it stop between species? What about jumping species makes this principle stop? And that's what I meant earlier when I said there's no discontinuity. There's no type of change that we've discovered. There's no level of complexity that we've dug up that we look at and we're like, we just don't know how this mutation could possibly happen.
Well, I could give 2 hypothetical examples.
Please.
Okay, I used this one already, but I want to say it again because I've been thinking more about it, and I think again, it's just an analogy, but it could be valid. Is my only point.
Yeah.
I've done a ton of, and this is in the past, so I don't remember stuff as well. I've done a
Hmm.
ton of research into New Testament manuscripts and New Testament textual criticism. I was actually invited today by a guy who's an expert in New Testament textual criticism. He actually runs that group on Facebook. He invited me today to go with him to Pakistan, coincidentally.
Ooh,
I have
that's
no
cool.
idea if I'll do that, but um, if we find a manuscript that is 99% similar to a manuscript of Matthew,
Hmm.
I feel comfortable saying that those go back to an original Matthew. No problem. And I like the manuscript analogy because they are replicated and they are copied, just like the genome is, and you get errors like typos, mutations.
Hmm.
But if we find another manuscript, which I will call John,
Hmm.
it's got a lot of similarity. There's a lot of the same stories, a lot of the same quotes. Let's say it's 80% similar. I'm not okay saying that both of those go back to a common ancestor. I think one could just go back to an original John, one goes back to an original Matthew, and that's it. So it could be in that example, I think everybody would agree that extrapolating that to another common ancestor before an original Matthew and original John would be wrong. And so I'm just suggesting, what if that's the case with genetics?
The thing about that is that those manuscripts don't have a non-violated biological through line. So, for example, there there is a way we can explain why Matthew and John differ. They had different authors, right? Like we can look at
Mhm,
them and say we know they had different authors because today different authors write different books and sometimes they share a lot of similarities, but you can very easily trace the edits of a manuscript back and tell whether or not you've got two different authors or not. The
mhm,
thing about genetics, though, is that that doesn't exist for genetics. There's
mm,
no clear authorship mark in any of the genomes. Instead, what we
mhm,
see is that when you sequence them, they bleed into one another. You can count specifically which changes have occurred in one genome versus another, like down to the base, down to the letter. And so then the question is, okay, one, is it possible that those changes can happen? And the answer to that is yes, right? Again, we don't know of any mutation that is outside of the bounds of, or I should say, we don't know of any genetic difference that is outside of the bounds of mutation. We see large scale duplication, sometimes hundreds, thousands, millions, billions of base pairs duplicated at the same time, whole genomes in plants. We see huge deletions. We see inversions where the sequence is read in the wrong way. We see point mutations, right? And these build on top of one another to create the large scale differences that we see within species and between species. So there is no difference in the types of differences that we see between cats and tigers, right? The
mm,
differences are clockable. You can write down what they are letter by letter. And you can say, okay, we find one gene maybe once in a cat, and then we find that same gene twice in a tiger. Is that possible? Yeah, duplication. That's happened in observed populations now. So, okay, then the next question is, okay, if it's possible that the mutation happened, is there enough time for it to have happened? Because as you wind the clock back, like you saw with our cartoon, individuals converge on common ancestors within species, full stop, no exceptions, right? Eventually, you're going to come to common ancestor of all people
mhm,
for Y chromosomes, for mitochondrial DNA, for the nuclear genome, whatever, right? You're going to come back to that common ancestor. Now, that doesn't mean that common ancestor was the only person on the planet at the time, but there will be a last common ancestor that everyone on the planet shares. And you can calculate that. So where does it
mm,
stop? If you haven't,
mhm,
if you have a lot of time, the convergence mathematically is just going to keep happening. And since
mhm,
all of the mutations are explicable, the burden is on the doubter of these relationships to say, here's where it stops and here's why it stops. Because for right now, we don't see any indication of that it's even possible for one of these gaps to exist, let alone that they do. Because it seems like they just keep converging. And the problem gets a lot more intense when people accept that some species are actually related. So, for example, if you were to say that cats and tigers are related, then every that they have a common ancestor. Like, let's say God creates an archetypal cat in the Garden of Eden, and all felids descend from that cat. That is what Answers in Genesis proposes. It's what many creationist organizations propose, and I have a problem with it because the types of difference that separate a tiger from a cat are no different than the types of differences that separate a human from a chimp. In fact, arguably, depending on the pair in question, the differences are way more severe than they are for humans and chimps. There's massive size differences. There's differences in coat color. There's differences in the topography of the teeth, right? The sagittal crest presence or absence. And so it's like, then you have a problem. There's a standard, right? So if you accept, like, as a creationist who thinks that common descent might not be true, if any animals are related, then you set a standard. And that becomes the baseline for comparison for all other organisms. And then what you're going to find is that you end up with a problem.
That's a great argument. I get it. It's a great argument.
Cool.
I do have one other thought. I don't want to lose it.
Please take it. Don't lose it.
I got it. So I gave you my first thought, which again is just a thought experiment. My second thought, you have to remember I'm a numbers guy. Are we talking, when you're talking about what you're talking about, such as you just did with tigers, are we talking about actual quantities or are we talking about percentages? Because that's going to be a huge sticking point for me.
I'm not using examples here with cats and tigers that I know off the top of my head. There was a publication a while back that said cats and tigers, or what did it say? It said your cat is 95% tiger, right? What the hell does that mean, right? And the answer, as somebody who's really sunk their teeth into this topic, is that it depends. And you have to dig so much deeper than you should to understand what people are actually talking about when they say something is X% similar to Y, right? There's a lot of ways to compare genomes. For example, like, let's say that I clone myself. If I clone myself perfectly, then all 3.2 billion base pairs, individual DNA letters, numeric letters, quantifiable letters that you can count, all of those should, in theory, be 100% identical between me and my clone, right? But what if I took my clone's genome and I duplicated it? 100%. So now my clone has, instead of 3.2, 6.4 billion base pair long genome. This happens in plants all the time. And in plants, it doesn't matter because they have redundancy into their genome, so it's fine. Well, my genome can be found in its entirety in my clone's genome, right? You could line my genome up to my clone's genome, and it's going to map perfectly along that first 50%. But then my clone's genome keeps going, and I don't have the extra genome to align to it. So in a genetic software, it's going to say I'm only 50% similar to it in alignment. But that's not right, because all of what makes me me is there in that clone. It's just there twice. So the answer to your question is there's only one, in my opinion, perfect way to compare genomes, and that's sequence the whole thing end to end and then produce a couple of different numbers. A number for the alignment, a number for the similarity of the aligned regions, and then a characterization of the regions that can't align. Is the reason it can't align because it's there twice in them and once in me? Or the reason it can't align because the sequence is different and it's doing something genuinely different? And geneticists are working towards getting that done in a lot of species. Long story short.
I feel comfortable now giving you this last one here, which is a little pushback on this of why doesn't it cross species? It could be that the gap is too big. Let me explain what I mean by that. Again, percentages are going to throw us off. I'm talking about base pairs, right? When you showed mother, father, child, grandchild, etc., we can count those differences. And I can't give you a number because I don't understand genetics, but we could count those. And there's going to be limits, right? There's going to be a range that a child can have. You see, sorry, you want to say something?
No, I was thinking. Sorry, I was emoting.
So
I was thinking.
what I'm saying is hypothetically we could look at humans and the differences that all humans have from each other and actually count it. So again, throw out percentages. It'll probably be 99.9%. And then we could look at like, let's say chimps, because they're our closest related ancestor, and then look at the differences between chimps and humans. It's possible, I'm suggesting it's possible that the difference is too big. And so there's your line.
That's been proposed, right? That the difference is that it's in several different numbers, but that it's effectively what you're saying. Take a raw alignment, line them up next to each other, and see how much of A can map onto B and how much of B can map onto A, and get a percent. And then do it again, but look just at the sections that could map and get a percent for that too. And then break down the actual genes that contribute to the making and maintenance of that organism and see what you get. There's two issues that I foresee with this. The first is that, as you're going to see, we've done this. We have those numbers for humans, chimps, bonobos, gorillas, orangutans, Bornean and Sumatran, and Siamangs. And then we have pretty good estimates for like mice versus rats, fruit flies, E. coli, other like model organisms. And so because of that, we can characterize the types of similarity just by brute base pair by base pair, count them up, right? And get numbers. And you're going to find that obviously there's a gap between humans and chimps, and between chimps and bonobos, and between bonobos and gorillas, because they're different species. So the question then becomes, what gaps are crossable and what are not? Because there's also a gap between cats and tigers, or between the different dog breeds, right? Like humans are more closely related to each other than a dog is to like a wolf, or than the two most distant dog breeds are to one another, if memory serves. So then the question becomes, okay, how much of a gap is too much of a gap? And the only thing that I ask is that we stay consistent with how much is too much.
Okay,
And go ahead, sir.
I'll work on that. I'll try to see if I can come up with something that makes logical sense.
Cool. Okay. So just to really hammer home this relatedness stuff, we've done actual experiments where we've taken bacteria. I think it was bacteria in this case. It might have been fruit flies. I can't remember. It was some model. No, it was a bacteriophage. Where we've taken bacteria and watched them reproduce in a lab and been like, okay, this one beget these ones, and this one beget this one, and these ones beget those ones, like marked their phylogeny. Then we sequence their genomes and see what type of family tree you get by genetic similarity. And those two map, just like it does within people, right? So that would be an example of like the literal, we watched them reproduce is recapitulated by the family tree by genetic sequencing. This is the reason why, again, we trust genetics over morphology. We've also done that, of course, with family trees. Like this is Ancestry.com's bread and butter. And it's robust enough that it holds up in court. And we've used it to catch like killers. And we use it in the AKC to like guarantee parentage amongst dog breeds. So this to me is the final experiment for evolutionary theory and common descent. Comparative genomics offers a chance, that should read chance, not change. It offers a chance to test evolutionary theory and common descent. Because if Darwin and evolutionary theory are correct, then the genetic similarities should work between species as well as within them. The gaps that you're talking about, those gaps should be either crossable or crossed as we look at organisms. And when I say crossable, I mean you need to show that those types of differences can occur in nature on their own, and you need to show that there's enough time for them to occur. So all life should theoretically form this nested hierarchy genetically in the known functional and known non-functional regions when the whole genome end-to-end is considered. And the differences between those groups, like I said, need to be biologically explicable. So the group relationships, then after you've done all of that, they need to be reflected in the fossil record as well. So awesome example of testing this. If humans and chimps are related, then when you look at the fossil record, you shouldn't find organisms that show a mix of human and bear characteristics along the time period that humans and chimpanzees were supposed to have diverged from one another, right? The relationships that are proposed need to be reflected in the fossil record. And this is what we've been talking about this entire time, right? Like these check marks that I've been doing for every one of these lectures, that's all of what we just talked about, sort of, you know, by design almost. So genetics and morphology agree when we have enough information. And I did not change this slide after your presentation. This is the slide as it was. So most evolutionary relationships argued on behalf of morphology have been confirmed by genetics. And I need to clarify this because I paid attention when you were talking about some of your specifics, specifically when you were talking about the pigs, the wild pigs. I think they're African wild pigs, yeah? I think?
Yeah, that's right.
Okay. You specifically used the word species, and I was listening very closely for that because species are a lot harder to categorize than like genus is, right? Or family is. And the reason is because of that same family tree that I showed you, right? If organisms are related by common descent, then the majority of their genome is inherited from much older relatives. So it's going to be a lot harder to classify species than it is to say, okay, all pigs are more closely related to each other than any is to like a bear. And so when I'm saying here that these relationships, as far as I know, are confirmed on the basis of genetics, I'm talking about these higher level taxonomic ranks. But then you cited a paper that said something about higher taxonomic ranks. I think it was for the rat type. No, it was for the snakes. And I don't know enough about snakes to say anything about that. To me, I heard that and I was like, well, shit. That's not what I thought. So I'm going to have to read it. I'm going to be talking about primates, though. And the ones that don't map up, at least from my understanding, are for these like reasons that we talked about, in addition to a couple more. Hold on. Oh, I remember what I was doing here. The first reason why morphology and genetics might disagree is because physical traits can result from very different genetic changes. So we talked about it with relation to people looking a lot like each other. There was a paper actually that I forgot to mention here that I thought was really cool. It's a comparison of people with their doppelgangers. So you know how like, yeah, there it is. So these people in this bottom picture aren't related. Like they're not close, they're not brothers or sisters or mothers and daughters. These are like people who have found people that look really, really similar to them, but they're not related like immediate family or even like secondary family. And so the researchers were like, that's really weird. What the heck is going on? And so they sequenced their genome and they found that they have similar but not identical mutations in the portions of their genomes that code for like face shape or hair texture or like brow ridge or whatever. And so they still are far more genetically similar to their siblings or to their parents, but in the regions that code for these characteristics that they share, the mutations are close but not identical. It's many ways to Rome, many ways to skin a cat. And in this same way between species, there are several different mutations that get dark fur color. So I talked about some of these earlier in the in this series, but all three of these mutations, CBD103, MC1R, and the agouti gene here, all three of these independently result in darker fur or like melanistic fur. There's not only one way to get a physical change, like a specific physical change. It's not one-to-one. So that would result in an incongruence between genetic and physical character or family trees if you were including fur color. And then some changes are more common than others too. I mentioned these ice fish that's supposed to be with the physical changes with different genetic changes because both of these ice fish have the antifreeze gene, like not the antifreeze gene, like there's one. They both have antifreeze blood, and that antifreeze blood is conferred by different genetic changes. That's how I should have phrased it. Many ways to Rome. And also, in addition to those other reasons why genetics and morphology might disagree, organisms that don't have very many close living relatives may appear to share an equal amount of traits with a bunch of different groups, which means unless you're sequencing them end to end, telomere to telomere, which has only just become possible, you're not going to be able to tell who their closest relative are. So historically, again, my understanding, elephants, dugongs and manatees, bats, cetaceans, colugos, and pangolins are all examples of animals that changed their placement drastically once we figured out genetic sequencing, because none of these guys have close relatives. Like all of cetaceans, what's the closest thing to cetaceans in general? Like no one knew. That's why people were like, I don't know, is it the mesonychids in the fossil record? Is it manatees? Is it hippos? And it wasn't until we sequenced the genome that we found out they share the most with hippos. Or like with elephants, it's like, what's the closest living thing to an elephant? I don't know. Like they're hard to place. Whereas when you looked at all the primates, for example, it's like, okay, all the primates share a lot of physical characteristics. They're pretty easy to group. As we went over, they share these really nice, neat, nested characteristics that group them into subsequent categories. So when you're a lone twig here, right, like these Afrotherians down here that don't have very many close living relatives because extinction doesn't equally impact all taxa, you're going to end up with these difficult to place animals down the line unless you have genetics. So primates, this is the paper that I used to say these morphology and molecules mostly confirm one another. It's 2007 paper by Pisani and colleagues titled Congruence of Morphological and Molecular Phylogenies. And I really want to look into that one paper you had with, it was a graph that had like randomness expectations versus what we actually find, because this paper says the exact opposite. This paper was like consistently across the board, we find that the phylogenetic trees that we get from morphology and from genetics outperform randomness. And it's like, it's not close. So I'm curious about that. For primates though, which again is the one I'm most familiar with, I actually had read this previously by Shoshani et al. Primate phylogeny, morphology versus molecular. They say overall, it seems that congruency between molecular and morphologic results for primate intraordinal relationships is a general rule. Minor differences in branching patterns include the position of aotus within Cebinae. And then they go on to say disagreements are minor and usually with regard to the precise relationships and classifications of hominid taxa. So they're like, morphology mostly agrees with genetics within primates. So where does genetics place humans? This is one of those big sort of final experiment moments, I think, because genetics offered a chance for us to investigate our own place in the animal kingdom. Prior to genetics, as we went over, humans are recognized as apes and as primates and as mammals, etc., because of physical similarity. This was the family tree that we created. If you go to like a textbook from the 1950s, this is going to be the phylogenetic tree that you see. Hominoidea, the apes, and amongst the apes, there are three families. They would say you have Hylobatidae, the gibbons, the little apes, and then you have Pongidae, which is the great apes, and then Hominidae, that's just humans. And the reason is because if you look at these guys, humans stand out amongst the great apes, at least. Like we're bipedal and our fur is very thin and we have little canine teeth. And so they were like, that's reasonable. Humans are apes, but they're their own type of ape and they're the odd man out. And in fact, as I mentioned with our cetacean conversation, at the AMNH, they still have this categorization. At the American Museum of Natural History, they use a classification that was outdated in 1975. So I don't know why they haven't changed it yet. But by investigating the genomes of organisms, we can check our work. We can see whether or not the relationships that we see within primates map our expectations on morphologic similarity. And we can see what that means. So the first person to investigate this, I'm super psyched to talk about this because I find this really interesting. These guys did some crazy experiments. So this guy is George Nuttall. And in 1904, prior to like genetics was invented, like rediscovered 4 years prior to this. And he decided that he wanted to assess the immune response of 230 different vertebrates to the blood of one another using a precipitin test. So the way that this works, like overly simplistically, is if you inject yourself with the blood of something else, or even a non-compatible human, right, who doesn't have your blood type, your body's going to issue an immune response. and it's going to fire off a bunch of antibodies that are going to go bind to that blood to get it out of here, right? To break it down. And interestingly, the more closely related two organisms are, or within species, the more similar their blood types. The cloudier the solution will get if you mix them up in a vial, because the more you share genetically, the more similar the genes you have for making blood, which means and for your immune system, which means that the binding sites are more similar. And what that means functionally is that when you mix them up in a tube, it gets cloudy as all this binding is occurring, and it clouds up the solution. That's what the precipitin test is. Cloudier means more closely related. So he does this, and he mixes humans with a bunch of different animals, and he finds that humans have the closest blood relationship with the great apes, so gorillas, chimps, and orangutans, followed by old world monkeys, then new world monkeys, and then things like rabbits. So people were like, that's kind of interesting, but like that's not super unexpected. So then Morris Goodman comes along decades later, and he decides to relook at Nuttall's test using modern tech for the 1960s. And he specifically is looking at the great apes, because he's like, I want to show that we see this family tree and that it maps our expectations. So he finds something really profound, though. All of the apes reacted very weakly to humans and vice versa, indicating that the blood was really similar. But the odd man out between humans, chimps, gorillas, and orangutans was orangutans. Orangutans were the ones that reacted least similar to all of the other three that showed up with a least similar blood relationship. And this was so shocking to people that it triggered like quite a bit of additional work. And Surich and Wilson are going to confirm this with albumin, which is another sort of blood factor, and they get the same results. The odd man out is the orangutan, not humans, as it should be. So people are like, there's got to be a reason for this. Like, there's got to be some reason why like humans are showing up as not the odd man out here, as more similar to gorillas and chimps than any of them is to an orangutan. But we can't really get there yet. We have to discover DNA first. DNA gets discovered in 1953 by Franklin, Watson, and Crick. And around this time, we karyotype, which is like staining and banding chromosomes. And we find out that humans have 46 chromosomes. So these are the actual stained chromosomes, like a picture of real chromosomes stained. And we find that we've got 46 chromosomes for a total of 23 pairs, while chimps, gorillas, and orangutans have 48, one additional pair. So this is like in direct opposition to the blood test, right? Like, this is weird. We don't quite know what's going on here. So if these animals are related, there's a very specific prediction that was made by conventional evolutionary science. And this is a famous evolutionary prediction. It starts in 1972, shortly after these chromosomes are sequenced by Turlot and Grouchy. And they say, okay, if all the apes are related and gorillas have 48 and chimps have 46 and chimps have 48 and gorillas have 48 and orangutans have 48 and humans have 46, then one of two things happened if these guys share a common ancestor. Either there was a chromosomal fusion in humans, or there was a fission 3 separate times in chimps, gorillas, and orangutans. And they were like, okay, probably fusion. That's the most parsimonious. That's the least amount of steps. And so they write this up in 1972, and they say, we think that what's going on here is that there was a fusion between 2 ancestral ape chromosomes to make human chromosome 2 in particular. They say, in this summary article, they say Turlot and Grouchy compared the karyotype of man to that of the chimpanzee and then that of the gorilla and the orangutan, and demonstrated that the human chromosome resulted from a fusion between 2 acrocentrics, and that human chromosome 2 is the specific one to be ID'd. So here's a comparison of ape chromosome karyotypes. The order is the same. The one on the right is the actual pictures of the chromosomes, and the one on the left is like a cartoon of them to show you what you're looking at here. And for 1, 2, 3, 4, 5, 6, 7, 8, all the way down, you have the same order in both of these pictures. It goes human, then chimpanzee, then gorilla, then orangutan, human, chimp, gorilla, and orangutan. And you'll notice that human chromosome 2 here lines up in banding pattern with chimp A and B, chimp A or gorilla A and B, and orangutan A and B. Their banding pattern seems the same. So this seemed to support what Turlot and Grouchy were saying. And I really have to pee, so I'm going to leave you on a cliffhanger, and I'll be right back. Is that cool?
Yes, for sure.
Okay, 1 second.
Not sure how to entertain everyone. Yes, I have to deal with chat now. Hey, Mr. Gibbon. Good to see you. I cannot do a flip. A dad joke. I love dad jokes, but I don't have any on the fly here. I don't read the chat.
Okay, I'm back. Peed as fast as I could. All right, so
that was fast.
Man, I was gunning it. Okay, so we have these chromosomes that are lined up. We have this expectation from an evolutionary perspective of what we should see if these guys are actually related and if a fusion is responsible for the discrepancy. But it's a little bit more specific than that. What actually should we see if human chromosome 2 is a fusion of two ancestral chromosomes that still exist in the other apes? So chromosomes are these little X structures up here. Hopefully you can see my cursor highlighted here. They're often represented this way because this is when they condense and it's easy for us to see them. The two chromosomes that make up this pair are bound in the middle by a centromere. That's just a structure, like a sequence, the name for the sequence that's pinning them together in that middle there. And then at all the ends of the chromosomes, there are telomeres. Right? You probably heard that term before. So you've got centromeres and telomeres. And then the content of these chromosomes, all of those genes and regulatory regions, that's going to be along each of these arms. So if a fusion occurred, we have a pretty straightforward expectation. If ancestral chromosome on top 1 on top here fused with ancestral chromosome 2 on the bottom here, and they stick together end to end just like this, then the resulting chromosome should have a fusion site right there in the middle. There should be telomere sequences right in the middle of human chromosome 2. And there should be a second centromere instead of just one, because you're taking two chromosomes and sticking them together. Does that make sense?
No.
Okay.
But that's okay.
It's okay. All chromosomes have a top and a bottom, right? Pretend that this is a chromosome here, highlighted by my cursor. There's a top portion in blue, and there's a bottom portion in blue. All chromosomes are like this. In the middle is the centromere. That's just the denoted middle of that chromosome, right? So if human chromosome 2 is the result of a fusion of two previous chromosomes, then instead of having one telomere section on top and one telomere section on bottom, you should also have a telomere section in the middle where those end to end, that top and bottom, stuck together.
Makes sense so far.
Makes sense? Okay. Then you should also have a second centromere, because if every chromosome has one centromere, then a fused together chromosome should have two. Does that make sense? Okay. Fortunately for us, we can tell whether or not a region, a sequence of DNA, is from a telomere or from a centromere, because telomeres and centromeres have calling cards. If you unwind these chromosomes so that they're strings of DNA, like really, really long strings of DNA, at the very ends of those chromosomes, you're going to find sequences that denote those telomeres. And you're going to find sequences in the middle that denote the centromere. So for telomeres, it's TTA, GGG, and forward, or on bottom / top, depending on which way you're looking at it. And then it's CCC, TAA in reverse. And it's like a billion of those repeats. So on every chromosome in the human body, if you take any cell and pull any chromosome out and you chop off the end of it and you sequence it, it's going to be like TTA, GGG, TTA, GGG, TTA, GGG, TTA, GGG, hundreds, thousands of times. And that's how you know that you've cut off a telomere, because all telomeres have this calling card. Likewise, All centromeres have a much longer calling card that's 171 base pairs long. And in the middle of chromosomes, this big sequence is found again and again and again. So all we need to do then is sequence human chromosome 2 and see if you can find these calling cards in the middle of the chromosome. Because in no other human chromosome is there a fusion suggested. And in no other human chromosome, spoiler alert, do we find telomere sequences anywhere other than at the ends. Likewise, centromeres are only in the middle. In addition to that, right? So here's our hypothetical sequence here. We've got a bunch of forward repeats and then a bunch of reverse repeats. The reason why you have it in forward and in reverse is because of how DNA reads. So if these telomeres are stuck end to end, this is the sequence that you should see, right? The end-to-end fusion site should be the ends of chromosomes, but instead of being at the ends, it's smack dab in the middle. Something that only is at the tips is now smack dab in the middle because two tips fuse together. Same thing with that second cryptic centromere. That's what you should see there, and that's what you should see there. It should look theoretically something like this, right? When you run those bandings together, the bands should one-to-one map onto one another, right? The sequences that make up this band should map to the same spot on the chimp when you lie them side by side. If human chromosome 2 is a fusion of these two chimpanzee chromosomes, right? So you need to have a second centromere, a fusion site, and the overall sequence should be really similar, right? If two chimp chromosomes in an ancestral ape glued together to make human chromosome 2, being overly simplistic, they should share sequence and they should have a fusion site. The problem, though, is that we can't sequence genomes in 1972. So instead, in 1975, we get this absolutely insane first of its kind paper, Evolution at 2 Levels in Humans and Chimpanzees by King and Wilson. And what these guys do is they use the nascent technology of DNA sequencing to look at the protein coding regions between humans and chimpanzees for the first time. And what they find is that humans and chimps are more similar to each other in protein coding regions than either is to anything else, which is, again, kind of shocking, but supporting both Goodman and Nuttall's earlier work that orangutans, not humans, were the odd man out amongst the great apes. However, it's important to note that they say in their article here, evolution at 2 levels in humans and chimps. And if you read this article, you find, much like this lower headline here, that the reason why humans and chimps are so different must be regulatory. They're like, humans and chimps are different enough that it's surprising that they're this similar genetically. So the things that make them different must be in how the genes are regulated, not necessarily the genes themselves. But this made humans and chimps sister genera. So humans versus the chimps / the bonobos, or chimps generally speaking. And it also suggested that whatever, like I said earlier, whatever made humans humans and whatever made chimps chimps probably wasn't in those protein coding regions. This created a new family tree, right? You can't have a group called Pongidae with chimps, gorillas, and orangutans if humans and chimps nest with each other to the exclusion of gorillas and then to the exclusion of the orangutans. Much like family trees within species, genetics told us that this is actually the similarity between these animals. So the first human genome that was mostly complete and the first chimp genome that was mostly complete were able to be compared in 2005. It's worth noting, neither were fully complete. They were mostly complete, but they were missing huge chunks of highly repetitive, difficult to sequence DNA. However, in the regions that were complete, we found that just like earlier, 99% of protein coding similarity existed between humans and chimpanzees. That should read higher than the similarity of rats and mice, which were sequenced around this same time at only 94% of their protein coding regions. When taking into account those size differences, because of multiple genes being, or multiple base pairs, I should say, being deleted at once or put in the wrong place, when you took that into account, the human chimp number fell to about 96%. And then the rat, mice, whatever, that comparison dropped to about 85%. So the researchers noted, they're like, okay, humans and chimps by these two methods are more similar than rats are to mice. What that meant is probably that humans and chimps had diverged from one another more recently in the past than rats and mice had. Because look at rats and mice, they're pretty different, right? But they're both like rodents. And so they thought, okay, well, the reason is probably because they diverged deeper in the past. So this ends up being maintained for 2 decades. People continue to try to further sequence genomes, and they succeed, but sequencing genomes is really hard and it's really expensive. So it was kind of slow going. However, after 2 decades, last year in 2025, we finally got the complete sequencing of the ape genomes. The authors describe these genomes as complete or nearly complete, missing very small portions of the genome. And I don't know which species they're talking about here, but they call them the complete sequencing, end-to-end, billions of base pairs for humans, chimpanzees, bonobos, gorillas, orangutans, Sumatran and Bornean, and Siamangs. They finished a lot of those hard-to-sequence regions, and they got the same phylogeny, the same family tree that's been maintained for 2 decades, where humans, chimps, and bonobos are more similar to one another than any is to a gorilla, but all of those guys are more similar to each other than any is to an orangutan, and all of those guys are more similar to each other than any is to a Siamang. But the thing is that it depends on the method. The actual, like this family tree is recapitulated every single time. You get the same family tree regardless of the method that you use. But you'll notice that this paper right here, or this paper, that this figure right here, which I created from the supplementary data of you et al., is the sequence identity within aligned regions. So humans and chimps are 98.64% similar to one another after the genomes have already been aligned. But this doesn't take into account those regions that couldn't align because of duplications, right? Like me versus my clone. So what happens if you take all of those into account and consider them as real differences? Well, people have been doing that since the original 2005 paper. And the number drops to about 85% of identical one-to-one, like literally map directly onto the other one. No duplications taken into account, though. No deletions taken into account. If I have 1 copy of a gene and the chimp has 10, that's only 10% similar, right? So the number of identical base pairs is about 85. If you take into, is about 85. If you take into account the types of mutations, then that number jumps substantially. And this is really important distinction. I would be remiss to not pause here and mention the creationist response to you et al. 2025. For 2 decades, and I'm very clued in on this issue, it's near and dear to my heart, which is why I'm bringing it up here. Creationists have been claiming that the human-chimp similarity is too high, that it's not actually as high as scientists are saying it is. Originally, this work comes from Jeffrey Tompkins, but his work fell out of favor after numerous continuous methodological mistakes. And the reason I can say that confidently is because I've seen that with my own eyes and in trying to recapitulate Jeff's work. But so has our mutual friend, Glenn Williamson / Ruhif, who made this really awesome table going through all of Tompkins's papers on this subject, on the human-chimpanzee genomic similarity, the algorithm that he used, the percent similarity that he got, and the flaw in his methodology. And Ruhif is the one who found out that Jeffrey Tompkins didn't weight his sequences. So if you had a sequence of DNA in a human versus a chimp that was 1 million base pairs long, and it was 100% identical, that was weighed equal to a sequence that might be 50 base pairs long and 50% identical, which is not how comparative genomics works. However, you et al. 2025 saw a resurgence in the creationist enthusiasm for this topic in particular, thanks to a manipulated figure by the intelligent design advocate Casey Luskin. He published this article first, followed by, I'm not kidding you, Will, 12 more articles in the span of, I don't know, a week. A lot of articles on the same subject. He says, bombshell, new research overturns claim that humans and chimps differ by only 1% of DNA. And he says that the new paper reveals that humans and chimps are only 85% similar. This is totally new. We had no idea, and so on and so forth. Problem, obviously, is that he cites this figure here, which shows the between species similarity via gap divergence. So taking again into account those big deletions, those big insertions between humans versus orangutans, and then, orangutans versus humans, and then the opposite, right? So the orangutan matched onto the human, and then the human matched onto the orangutan. and then the gorilla mapped onto the human, and the human mapped onto the gorilla, all the way down. And Casey says, look, this shows that for humans and chimps, and you look at the autosomes, so that's all the genomes all of the chromosomes besides the sex chromosomes, that's a 12.4% difference, or a 13.3% difference between humans and chimps. So he's like, they're way more different than we thought. There's two major problems for this. The first is that this is a manipulated figure. This is the full figure. He cut out, as you can see from the little purple hump down here at the bottom left, all of the within species comparisons. And these within species comparisons are important because the gorilla within species comparison showed a difference higher than the human and chimp difference. So if you want to say that this gap divergence, that large-scale deletions and large-scale insertions in non-functional regions of the DNA mean that humans and chimps aren't related because they're like only 87, 86% similar, then gorillas can't be related to other gorillas because they show extreme within species divergence due to, and for whatever reason, I don't know why it's happening in gorillas. The paper talks about this itself, massively rapid structural evolution. So perhaps even generation to generation, you might have a dad that has 1,000 more base pairs in a particular reason than his son because those 1,000 base pairs were deleted or duplicated in the son. And so you end up with these structural differences because of changes in the non-functional DNA. And this becomes even clearer later on in the supplement. You can kind of think of these differences as occurring in the large and non-functional regions of the genome, which, by the way, is, I think, excellent support for the fact that these regions aren't doing anything. If they can be that different within two gorilla haplotypes, to be specific/precise here, then I don't think that those regions are doing anything because gorillas all look like gorillas and they all reproduce like gorillas and they all behave like gorillas. I suspect that these differences, as the paper said, are in non-functional regions, and that's why they're able to accumulate so quickly because they're not doing anything, so selection can't weed them out. This applies. Yeah, go ahead.
I've got a couple questions. I don't remember because I haven't looked into this, but and you might say that it was just chance, but didn't Jeffrey Tompkins publish a paper and the percentage that he suggested was pretty accurate based on you et al? No.
Jeffrey Tompkins published 6 different papers across 5 years, and he got a range of 70 to 84%, all of which are still lower, or excuse me, all but 2 of which are still significantly lower than the alignment method. Now, to be kind of precise here, Tompkins also wasn't trying to get an alignment number. That's not what he was trying to do. He was trying to get a sequence identity for many of these. And in fact, like online, me and Rob Carter, who's also a creationist and who's friends with Jeffrey Tompkins, and Jeffrey Tompkins both went round and round on this subject. And even Rob Carter has a video out that talks about how Jeffrey's methodology here isn't appropriate. And he's not measuring the same thing. So yes, if you want to say 85% alignment validates Jeffrey Tompkins because he has a range of 70 to almost 90%, I don't know. I mean, you've got 30% of the genome there. That's a pretty big range.
Okay, fair enough. Someone contacted me and said that they know you and wanted to give me some information on this. And so I don't fully understand it at all. They clearly do. But they told me that there's someone named Dr. Richard Buggs, Buggs. And they said that he said for years, and nobody would listen to him, I don't believe he's a young earth creationist, that it's actually closer to 85%.
He did say for years that it was 85%, but it wasn't that no one would listen to him. He said that the 85% number is the number that we should use. And the rest of the scientific community got this same number in about 3 or 4 separate papers from 2016 to 2020. right? So Bugs got his number by taking the supplementary material that was published from the 2005 chimpanzee sequence. And he said, look, if you look at the entire genome, what can be aligned, not taking into account duplications or deletions, you can only get a 1-to-1 alignment of 85%, about, give or take. I think his was a little bit lower than that. And the rest of the scientific community, people you talk to, like Steve Schaffner, you can find their correspondence on blog posts to this day, was like, that's fine, but that's not the number that you should use when you calculate species similarity, because those large-scale duplications and large-scale deletions and large-scale insertions, which have no impact on the phenotype of the organism, are erroneously driving that number up. And what's really interesting about this is as that same number was recapitulated, about, again, 85, 90%, give or take, as an alignment number by non-creationists and creationists alike, I should say, because Richard Bugs is an intelligent design proponent, from, again, 2016 to 2018, but really it's more like 2021 too, because there are a couple of papers that I'm thinking off the top of my head. The reason became clear. Because if you use this method of raw alignment as the percent similarity, and this is what people were telling Richard Bugs and creationists in general who advocated for Richard Bugs's numbers, what you end up with is a percent similarity between two humans of 86.96%. Like, let me repeat that. If you use an alignment method, because these mutations build up so quickly in the non-functional regions of the genome, they're able to build up that quickly within species too, including humans. This isn't a father and son being compared. These are two human reference genomes. So they're going to be taking pretty opposite ends of human diversity. But humans can be as different, nearly as different as humans and chimps along this alignment region. Here's the human chimp number, 84.95%. That's the other human reference genome used in the study, human GRCH38 at 86.96%. So this is the problem, Will. And this is why I get so worked up about this topic in particular. This is why everyone told Richard Bugs that this number isn't appropriate, because you're including non-functional differences that build up within species, contribute nothing to the phenotype, and acting like they're part of what make individual species or individual humans different. But I know that two humans on the face of this planet, any two humans, when you look at the percent similarity that's shared here, that 86.96%, they are 99.9% identical base pair for base pair in that aligned region, because those are that's the portion of the genome that's responsible for the making and maintenance of the human. So this is the latest on this, right? Like, this is the most recent paper on the subject, right? And I highlighted this because I think Casey Luskin is lying here. I don't use that word very often, but he highlights this 15.5% difference, specifically referring to the human and chimp number in the supplementary material of this paper. And directly above it is that human number, which gets conveniently left out. So, like, these differences aren't reflecting functional differences, because two humans look like two humans, and they behave like two humans, and they think like two humans, and they're capable of all the same wonderful behaviors that humans are capable of, and horrible behaviors that humans are capable of. This is what I meant when I said we were going to talk about another reason why I think the genome can't be 100% functional. Because if humans can differ this much in their genome and yet physically show such little differences, I just don't think that portion's doing very much. And this isn't the only paper that discovered that, by the way. This was the first paper that included both human reference genomes in a comparison to the other great apes. But in these two papers over here from 2023 and 2021, I think, maybe 2022, within 2 or 3 years of you et al., they got the same thing. This top paper got 6% difference between two humans that were within the same population. And this lower one got a 10% difference between the human reference genome that just got completed and the Han Chinese reference genome. Like, I'm sorry, but there's just not 10% of difference. There's not 10% difference behaviorally or physically or reproductively between a person from China and a person from here. I just don't think that's reasonable. So this is the crux of it, and this is why I prefaced all that functional, non-functional stuff ahead of time. I want to be crystal clear too. I spoke with the authors on this. Like, I made sure that I'm understanding this correct. You can check my work. I can send you the paper and you can look at it for yourself. But the genome is just really complicated. And this is why when you ask how similar to humans, you say 99.9%. And I think that's appropriate to say, because I think humans don't really differ more than that in what we do and in who we are. If you want to say humans and chimps are only 85% similar, though, you're going to have to say that humans and humans are only 87% similar. And I think that's absurd. By the way,
fair argument.
Yeah. And thank you. I'll say, by the way, too, my challenge to any, Jeffrey Tompkins, Casey Luskin, Rob Carter, any of the creationists who are doing this genetics work, I have never once, not one time in all my perusal of the genetics work on this subject, seen them include an outgroup pairing. It's only ever humans and chimps in isolation. What does 85% mean? It seems a lot lower than 98%, doesn't it? Until you include human, and you realize that number doesn't mean what they're telling you it means. And that's why you have to include an outgroup, which Jeffrey Tompkins never did. He only ever did human versus chimpanzee reference. And I have my suspicions as to why that might be, but I don't know.
So I mentioned this earlier. I don't like percentages as a numbers guy. Can you help me? And if you don't know, it's okay, but can you help me quantify what we're talking about here? Like, how many actual differences, not a percentage, is there between a human and a chimp?
If you want to go by this method, 3.2-ish billion base pairs.
Mm-hmm.
The chimpanzee genome is a little bit bigger than the human genome, and most of that is due to duplications. So it's not going to map, even if they were 100% identical, you wouldn't get 100% similarity by this method. Because this is saying, okay, if you take one genome and you line it up to the other one, and you draw lines from base pair to base pair across those two genomes, what is the number of shared base pairs put into a percentage out of total base pairs of the larger versus the smaller and the smaller versus the larger? So in this comparison here, what you're literally looking at is you're looking at the alignment coverage. You're looking at the human here, or the chimp. Let's just use the chimp because that's easier. Using the chimpanzee, how much of the chimpanzee could lay directly on top of this human genome, 1-to-1, like draw a direct line across. And the number was 84.95%. So whatever that is, whatever percent or whatever number of base pairs 84.95% is out of 3.2-ish billion. That's the exact number of base pairs that are 1-to-1 identical between a human and a chimp. However, as I said earlier, if one of those genes was duplicated in a human or in a chimp, that's being counted as a difference because it can't be drawn 1-to-1. Do you see what I'm saying?
Mm-hmm.
Okay.
I just did some quick math based on what you said, and it's like 480 million. So I can look if that's accurate. Now, yeah. you are bringing up an interesting point, which I don't know how they're doing this, right? A goes with T, C goes with G. So to me, like, how are they lining these up? Like, are they saying, here's an AT, here's an AT, we cancel each other out? No, that can't be, right?
It's the order. So what I do is I take my first genome and I start at the very beginning. Let's pretend telomeres aren't a thing because the telomeres have that specific sequence. So I guess you could start it there, and it would be, remember our telomere calling card is TTAGGG. Let me double check. Hold on. I've got it right here. What is it? TTAGGG. Come on. Yeah, TTAGGG. So you start at one end of the human genome, and it says at that telomere, it starts TTAGGG, 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 hundreds, thousands of times, right? You start the alignment, and it's going to go look onto the chimp chromosome, and it'll take the first set of, and it depends on how you define the parameters in the program that you're using. In BLAST, you can say, okay, run for 300 base pairs and see if there's a mismatch, like 1-to-1 in the order. So in the human, if it goes TTAGGG 8 times, does it do that in the chimp? 8 times with no mismatches? And so it just runs. It's looking at the order 1-to-1 across the genome. And if there's a gap or a break, most comparative genomics tools will look at that gap and say, okay, there's a gap here, but does it pick up within, and you set the parameters yourself, 150 base pairs or 1,000 base pairs. And so it'll scan ahead a little bit, and if it picks back up, it'll restart the alignment. And it'll take that chunk that was misaligned and chuck that into the misaligned pile. And say, and then you can go in there. This is why, I don't know if you saw, the number of authors on this chimpanzee on this great ape paper, it's unbelievable. Those are all authors in blue, because you don't just have to sequence it. You have to look at what each of these big chunks is doing, right? Like what's each gene? Is it one of those things where if I have 2 copies for a gene, there's no difference? I just have a second redundant copy, or does the second copy amplify whatever that gene does? So they have to go characterize all of the differences and all of the similarities. It's a absolute unbelievable. It's a absolute tour de force kind of paper. But it shows just how complicated comparing genomes really is. And you're right to be suspicious of percentages for exactly that reason. Because what does a percentage mean, right? And if, would you say 400-ish million base pair difference between human and chimp? It's close to that for the human by this method.
Oh, really?
So like, because see, if take what is 86.96% of 3.2 billion? If you run that math.
I'm doing it. Sorry, a little slow.
No, it's okay. It's okay.
417 million.
So that's going to be the human difference from this method. So it's like, that's the problem. That's why comparative genomics is difficult. We'll see what ends up happening, but like, to me, I don't know. That's my like hobby horse subject. Okay, are we good on that? Are we good to move on?
Yeah, for sure.
Okay, cool. All right. So obviously, because we have the complete genomes, we also have human chromosome 2, completely sequenced, and chimp chromosome 2/12 and 13 completely sequenced. In fact, we had them mostly sequenced in 2005, but it's only gotten better since then. So, spoiler, the expectations that we laid out, that Berlot and Grouchy laid out, are met precisely by this sequence. So this is the paper that was published in 2005-ish, and they say down here, sequence blocks that closely flank the inverted arrays of degenerate telomere repeats marking the fusion site are duplicated at many primarily subtelomeric locations. In addition, there is a large centromere proximal block duplicated at these specific coordinates and so on and so forth, along with 98.99 average sequence identity. So what does that mean? That means that these 2 chromosomes, human chromosome 2 versus chimpanzee chromosome 2A and 2B, once you aligned them, they shared their sequence at about a 99% rate. So in the aligned portion, and we actually have the amount that is aligned, it's about 87% from you et al. Of the 87% that can be aligned, they're 99% similar. Same thing with the human, with an additional 2%, because humans are more similar to other humans than they are to chimps, obviously. In addition, we found the fusion site, and this is the sequence, and here's where it's located. So there is a head-to-head telomere fusion site in the middle of human chromosome 2. This signal exists nowhere else in the human genome. And the only place that you find these telomeric sequences are in the ends of the chromosomes. But you don't find this head-to-head anywhere because it's 2 ends stuck together. Whereas in normal chromosomes, they read forward on top and backward on bottom in simplistic terms. Likewise, they found the second degraded centromere, and it's that big sequence that I told you about repeated 185 times. So you have a cryptic centromere, and you have the fusion site, and the chromosomes map onto one another. Meaning, if it's not a fusion, that's what it looks like. If it's not a fusion, it's made with a fake fusion site in it, because there's nothing here. I should mention too, there's a gene that flanks this fusion site, and it's only in every other member of its gene family. It's only found in telomeres, and here it is only once in human chromosome 2. So it's a big deal. It's a huge confirmed prediction from evolutionary theory that this fusion site would be found, and then 25 years later it was found. All right, does that make sense, what I'm saying there, why that's important?
Yeah, this is brand new to me, so I don't understand how chromosomes can fuse. I don't know if we've seen this happen before. I don't know how this becomes fixed over the entire population. A ton of things are running through my mind right now.
Those are all great questions. I can send you some papers on it, because we've also seen this happen, or we document this, I should say, in pigs and in horses versus zebras. So it's pretty cool. And differing chromosome numbers is like not uncommon within animals that are like pretty similar. So like all gibbons, like across all gibbons, they range in their chromosome number by like, I think they can be as low as 38 chromosomes and as high as 50 chromosomes. And they're all gibbons. So it's like, this kind of stuff can change pretty rapidly.
Okay.
They're not the only calling card. ERVs are another great example. Have you heard of ERVs before?
I have, but I don't know a lot about them.
So ERVs are sites of ancient viral infection passed down through generations. So the way that this works, as outlined by this clip that I took from Stated Clearly, which is a great channel, I love John Perry, he animated this to show how this works. So retroviruses are RNA viruses, and what they do, as you can see in this little cartoon here, is they attach to their host, and then using reverse transcription, they turn their RNA into DNA and insert it into the host genome. And then when the host divides, every time it divides that inserted chunk of ancient virus genome with it. So you literally end up with basically a scar on your genome from an ancient virus, or from a virus that, if it's passed down, ends up in your kids, and it ends up in your kids' kids and their kids, and so on and so forth, because it's now a part of the genome. This is what it looks like. It's got a long tandem repeat, followed by the GAG gene, the POL gene, and the ENV gene, and then another long tandem repeat. These are what they all look like. When an ERV, or when you've got the site of an ERV, this is the sequence of elements every single time that we're aware of.
What elements?
It's like a repeat, like a buffer, and then the genome of the virus, and then another buffer, and it sticks right inside the DNA. So you'll have your DNA here, and then because this virus has infected you, it cuts your DNA open, inserts its genome, which is very short, but inserts it in its entirety into your genome, and then stitches it back up. So every time your cell divides, it replicates that virus's genome too.
Okay, so this is not A's, T's, C's, and G's. It's something different.
It's A's, T's, G's, and C's, but these are the genes. So A's, T's, G's, and C's code for genes. These are the names of the genes. So GAG is the name of a gene. I don't know what the sequence would be. SO's POL, SO's ENV, and it's some string of hundreds of A's, T's, G's, and C's. And so you end up with an ERV sequence that's like 7,000 base pairs to 12,000 base pairs long in the middle of your genome.
Okay, if it's the same letters, same base pairs, how do we know it's a virus?
Because the order. That order only occurs. That's like asking, how can you tell two proteins apart? Because the order, right? Like, one protein is a muscle protein and another is, a milk protein, because even though they share the same 4 letters, the order of those letters determines the protein. So it's the same. This sequence only exists in viruses, retroviruses.
Okay.
So they slam themselves into genes, and if it happens in your sex cells, these can be passed on to your offspring. And so you end up with a virus that is passed from generation to generation. And because it's a part of your genome, which is replicated trillions of times every day, it's possible that this thing mutates because your genome is like your DNA is responsible for replicating it too. And so when your genome replicates the ERV that's inside it, it can make mistakes, just like it can with the stuff that's yours, and render this retrovirus functionless. And so then all it is just a site of ancient infection. And so what ends up happening is that these ERVs work the same way as mutations. If I'm infected with an ERV right now, which at the time would just be a regular retrovirus, I get a retrovirus and it happens in my egg cells, right? I'm going to pass that retrovirus on to my offspring. If my offspring experiences a mutation in that region, which this happens a lot, and I'll tell you why we know that here in a second. It's because it's happening right now in different sections of the human population. Like viruses are in the middle of becoming fixed within human populations. Then my kid will pass on that same mutated retrovirus to her kids, and so on and so forth. And so just like mutations, they end up as markers or receipts of shared ancestry. Because if I get this ERV and pass it on to my kids, that doesn't impact my sister or my cousin. They didn't get that ERV. It didn't insert itself in their genome. So it marks my lineage specifically. And the thing is that it's not just any retrovirus. There's a bunch of different families of them. I don't even know very much about this topic. There's like dozens of families of these retroviruses. And within each family, there are different retroviruses. And so when they insert themselves into a genome, they're going to insert themselves in one of 10 million places, according to the literature. And so if you have a retrovirus, the presence of the virus, the location of the virus, and the status of the virus, whether or not it's experienced mutations, all play a role in helping reconstruct the family tree. It's crazy.
Okay, so I want to make sure I'm understanding. So these aren't fixed. These have not achieved fixation then. Am I hearing you right?
Well, that's the thing. We have over 100,000 of them, and those are all fixed. But we have another several that are floating around in our population right now that exist in some populations and not others. So it's like there are a lot of ERVs, like a lot of them. And that's how we know that these are ancient viruses, because they're in the process of fixing in some populations as we speak, and in non-human animals like koalas.
Okay, so I've got two confusions here, and this is really going to show my ignorance, but I don't mind.
No, it's okay. I might not know the answer.
When I hear the word virus, I don't hear good, I don't hear positive, I don't hear beneficial. So why are they in the process of fixing? Are they giving a survival advantage? I don't understand.
So the reason why the reason why they fix is usually, well, in most reasons, as you'll see here in a second, it's just drift. If they're rendered functionless, which is looking at where they're happening right now, is common, right? So when they insert themselves into your genome, that's bad. But if they experience a single mutation, that's going to break, because their genomes are so little, that's going to break their ability to create more of themselves using your genome, which is how most viruses work, right? They hijack your cells' reproductive capabilities to make more of them. But in these retroviruses, because they insert themselves into your genome, the way that they do that, if they experience a mutation, it breaks their ability to reproduce. And so then they're just a receipt of a virus that used to be able to make more of itself. And now it's functionless DNA in your genome. And so it doesn't confer a survival advantage. In fact, this is exactly why there are so many ERVs that are in the process of fixation in populations right now. Some of them won't fix. In fact, probably most of them won't fix because they don't confer an advantage. They're just no longer viruses, but they're functionless pieces of DNA that are not subject to selection. But what they do is they record lineages, right? So if it becomes functionless in my kid, that doesn't matter. My kid's kid is going to inherit it all the same, and her kid's kid will inherit it, and so on and so forth. And you'll be able to trace it back to me as the one who originally had it. They're crazy. And so,
okay, so To make
Yeah,
sure I
yeah.
understand, so you said the order is what tells us it's a virus. So am I hearing you right? We've like sequenced the genomes of viruses and we know what their order is, and then we find that order in the human genome?
Yes, and those genes specifically. It's the order of the order of those base pairs determines what gene you're looking at, right? Or what gene they're
Mhm.
coding for, rather, to put it more precisely. And those genes only exist in these retroviruses. That order is exclusive to them. And it's easy to sequence them because the human genome is, 3.2 billion base pairs long. These retrovirus genomes are like 7 to 12,000 base pairs long. They're really little
Hmm.
because viruses are really little. And so
Mhm.
they're really easy to clock. I don't know. They kind of blow my mind. And as somebody who's not a geneticist, that's you're going to see my pool get shallow quick here. But my understanding is that these things are they're just receipts of ancestry. You really want them to get functionless fast, though.
Okay.
So here's a good opportunity, though, right? Because if ERVs are this common, if we have over 100,000 ERVs or ERV chunks, like just in humans, then like we can compare those ERVs to other species. And if they have those ERVs too, and if they're in the same place, then that's going to tell us that that virus either infected out of the, in a one in a gazillion chance, infected both us and that species in the same place at the same time, or we inherited it from a common ancestor. So that's the test, right? Is to take all of the ERVs in humans and look at them and see if they exist in other animals. And what they find is, one, there's a shit ton of ERVs. So this paper only went and looked at one group of ERVs, the HERVW group, which is a specific family of ERVs. That we call them HERVs in humans because it means human endogenous retroviruses. And they were like, can we find them in other animals? And so they looked at the primates in this paper here. And in presence and location, so in being there and being in the right place, of the 211 ERVs from this family, again, small amount out of the 100,000 or whatever ERVs we have, but of this family that they were able to look at, 131 of them were shared by all catarrhine primates. So all monkeys and apes have 131 of these ERVs. 62% of the human ERVs are in everybody. Same place, same type of ERV. We would expect that there would be even more than that shared just amongst the apes then. And that is in fact the case. Amongst all of the apes, 190 members of this family are shared, or 90%. And amongst just the great apes, 97% of these endogenous retroviruses are the same and in the same place. This is like a pretty solid support of common ancestry because, to our knowledge, these can't be anything else other than viruses. And to our knowledge, viruses aren't very good. I'll add that this corroborates this paper here that I've cited by Grandi et al., highlighted by my cursor. This paper that we just went over corroborated earlier work from 1999, one of the first looks at ERVs, where they say constructing primate phylogenies from ancient retrovirus sequences. And these authors talk about how you get the same family trees just from ERV presence or absence that you do from the earliest draft versions of the primate genomes that they had. And I suspect now that we have the complete genomes, this should probably get run again. Because I don't think that we're working with very many complete anything in 1999. The human genome's not even complete yet to see if this holds. But this paper is working, this one with just the apes is working with pretty complete stuff. I think this is from 2016. So not complete, but pretty good.
Quick question.
Yeah.
Is there a reason maybe it's just cut off? There's no bonobos on there?
Yes. I believe this paper came out before we had the draft bonobo sequence. So the draft bonobo sequence wasn't until like 2018. We didn't have any bonobo genome until then. Like regions, but there wasn't like a good consensus sequence until then.
So it should probably be in like the 205 range?
It should be, yeah.
Okay.
It should be. And I mean, I would even say that sets up a pretty interesting prediction on its own. Like what's cool is that these genomes are we have the complete genomes. So you could theoretically go right now with a genetics search tool like BLAST, and you could look for these specific ERVs in this paper, and you could just search them across all of the complete genomes right this second. I don't know how to do it, but you could definitely do it. Okay. Could ERVs be created? I don't think so. I say no here pretty solidly, and I feel pretty strongly about that because, as I said, we're observing ERVs right now in the process of fixing in humans and other animals. So here's unfixed endogenous retroviral insertions in the human population, pretty straightforward, and then koala retrovirus, a genome invasion in real time. So we're watching this happen. And what's really cool is that in this Marchi paper, this human ERV one, they looked at a bunch of different ERVs in the process of fixing in human populations or not fixing. And in some of the populations, the ERV was present in like 2% of the population. And in others, a different ERV was present in like 95%. So it depends on the virus, and it depends on whether it's functional or not, whether it's hurting you or not, or even rarely, ERVs can gain a beneficial function because once they're deactivated, they act as non-functional DNA, which means just like what we saw with the ice fish, where they got an antifreeze protein out of non-coding DNA, you can get a functional protein from an ERV if it's been dead for long enough. I also think, this is the main reason actually, not the main reason, but a main reason why ERVs couldn't be created differences, because we can reverse engineer ERVs back into functional retroviruses. So in these papers, they took ERVs and they like reactivated them. Like they looked at the mutations that had deactivated them, because remember, they're only like 7 to 12,000 base pairs long. So that's not a lot to search. And they were like, okay, what deactivated this family of retroviruses? And they compared it to the other families, to its closest similar retroviruses, turned them back on, and got a functional retrovirus. Like, I think they're viruses. So most ERVs, and I got this from Dan, who you know, so you can ask him for more information here. It's not just that they don't do anything, like they're just inactivated, they can't do anything. And it has something to do with which genes are turned off out of the 3 that they have. But out of the 100,000 retroviruses ERVs that humans have, about 20 of them do stuff. And those 20 are like found in other animals too. There are a couple of them, I think there's 1 or 2 that are unique to humans, but I don't think they do anything. Don't quote me on that though, because again, this isn't my area. Okay, so how are we feeling?
Full of information, to be positive.
Good. We're going to speed run paleontology here, the first couple steps of paleontology. We're done with the genetics. I feel like I've sufficiently shown that humans are physically and genetically apes insofar as one can tell that. How do you feel about that?
Yeah, it's a really good question. When you bring up the chromosome thing, could that be a way of pulling humans out of apes? Like, what if I said,
Fusion?
huh?
Like the fusion?
No, not the fusion, just the difference in the number, right? So what if we get to apes and we say apes have 48 chromosomes, and we add that as one characteristic that they all share that we don't? Would that pull us out?
I don't think so, and here's why. Because if you want to create the most sort of true similarity tree using genetics, which is the only reliable way that we know you have a ancestor-descendant relationship within organisms, the best way to do that, to compare genomes, is to include the whole thing, not some of it, but all of it. And if you do that, right, that human chromosome 2 is more similar between humans and chimpanzees and bonobos than it is between chimpanzees and bonobos and gorillas. So despite the fact that it's fused in humans, that humans have 46 and the other apes have 48, the content of those 46 chromosomes is more similar between the 46 having human and the 48 having chimp than it is between the 48 having chimp and the 48 having gorilla. So that, I think that'd be a hard sell.
Okay. Fair enough. That's just where my mind went.
You wouldn't be the first person who was like, there's got to be a way to pull us out of here. It's happened before. All right, we're going to do paleontology quick. We're going to do Paleo 101. Obviously, becoming a fossil is rare. Fossils aren't very common. Step one is you got to die, and then you want to get buried quickly. Because if you aren't buried quickly, then you're going to get ripped apart by scavengers, right? Again, this is a picture that our mutual friend John took of a fish decomposing on a beach. And part of it had already been scavenged away by like either birds or crabs or whatever. But if that remaining half was to be buried under sand, like right now, it would stand a much better chance at becoming a fossil. Then you want to get buried even more, right? Because if this little fish gets under the sediment by like 3 inches, then something might come along and dig it up, a dog or a larger fish once the tide comes in. You want to get buried even more. Ideally, you want to be sequestered as far away from everything else as possible. Then you need the areas around you to become rock. Then you need it to lithify. And then you need your body, which is lithified in that rock, to become mineralized or paramineralized, and for sediment to fill that cast, basically the fossilization process. And then you need to be found. But there are many ways to preserve, as we talked about last time. You can become a fossil by falling into a tar pit, like we see with this Smilodon fossil, the stuff at La Brea. You can become a fossil, or be preserved rather, by getting trapped in amber, like resin, tree resin, like with this ant. You can fossilize if you end up caught in a mudslide, like these dinosaurs. Or you can get fossilized if you sink to the bottom of an anoxic lake, because anoxic lakes have really low oxygen content, which means at the very bottom of these lakes, bacteria that require oxygen to survive can't live. It's a hostile environment for decomposers. And so if you die and fall into an anoxic lake and you sink to the bottom, there's nothing to decompose your body. So you just end up laying there in this basically like a preservative, essentially, until sediment buries you, like with Darwinius massilae here. Or you can end up desiccated in a desert. It's easy to fossilize if there's nothing to tear you apart. Some interesting fossils. Obviously, we have fossils that form in a myriad of different ways. This fish fossil was specifically formed in a limestone mudslide. We went over this a couple of lectures ago. These dueling dinosaurs in a desert environment. This little mammoth was obviously frozen solid. This ichthyosaur was caught in a submarine landslide while giving birth. And this little, this little tetrapod and this little mammal were in a burrow when the burrow was flooded, filled with water, and they were turned to stone over time. So the thing about fossilization and about burying quickly, and the thing about rocks in general, is that you can use like processes in the present to tell what happened in the past by looking at the mechanics behind like geomorphology, the grain size, how they're sorted, how they've moved, the types of microfractures in the rocks, whatever. Because catastrophism happens. Local catastrophes happen today all the time, and they leave clues of themselves when they do, right? So like for these fossils, how do I know that this was a local flood for these two little guys up here and not a global one? Or how do I know that this submarine landslide only occurred where these ichthyosaurs live instead of on the whole ocean? Or how do I know that these dueling dinosaurs lived in a desert, right? We can use the current rock formation and current catastrophes to apply back to the past and see if those same features are present. So this is a submarine landslide that occurred off the coast of New England. We caught it like on GPS or whatever, and they 3D modeled it. And this is a fossilized landslide. Like it's a landslide recorded in the rock. And you can see it, they have these same features of a mishmash of these slumping sediments. We've even found fossils like that ichthyosaur caught in submarine landslides just like this. So that's how we know that was a submarine landslide and not like a tumultuous global event, because there are limits to where this landslide begins and ends. Or you can use volcanic ash, like in this preserved group of, I think they're rhinoceros here, from the Miocene. Uniform processes versus local catastrophe can be checked, right? Both of these rocks have these gentle ripple marks on them. I got this from John, by the way. And these ripple marks show up today on beaches in this not even fully lithified rock. But we also find them in the geologic column in ancient oceans, ancient shallow seas. So the present is still the key to the past, right? Like modern catastrophes do happen, and they let us see how they impact the world. But because modern catastrophes happen, we know what to look for in the rock record if such catastrophes happened in the past. Big floods happen now, so we know what we would expect to see in the past at every formation if a big flood buried them. Same thing with a big asteroid impact, or with a big volcanic eruption, or a big sandstorm, or what have you. Modern geology is not rejecting catastrophism. It just says that modern general uniformitarianism is combined with local catastrophes to tell the true story of geology, because it's factual that catastrophes happen. It's also factual that they don't happen all the time everywhere. So how do we age fossils, right? We've got relative dating and we've got absolute dating. Relative dating gives us an order. Absolute dating gives us a numeric age. Relative dating relies on the law of superposition. So as you've seen this before, the law of superposition by Steno in the 1800s basically was like, okay, layers that are lower down are older than layers that are up top, assuming that the sediment hasn't been disturbed. So in this sand over here, which hasn't been disturbed other than people walking on it, hasn't been shaken up or anything like that, we can see that first someone dragged their cooler towards the beach. Then someone drove a Jeep or a side-by-side over that cooler drag. And then some person walked over that tire track. So we know that the order was first someone dragged the cooler, then someone drove the Jeep, and then someone walked across. The problem is that's if rocks haven't been disturbed, and rocks can be disturbed. We know that, right? Here's a cartoon that shows some disturbances, regular old laying down versus what if you have a river that carves through and gouges out a big chunk of the middle? Or what if you get tilting, right? A big magmatic upwelling that punches some of these layers up and makes them diagonal, or an intrusion. Those happen. Undisturbed, tilted, right? Like, that's just a thing. So how do we know when they've been disturbed? They leave traces of themselves. And we know that these are traces that are indicative of these types of change because some of these changes are happening very, very, very slowly right now in the present. Rocks can fold under immense heat and pressure. But if they fold like that, they leave microfractures and macrofractures that you can see at these bend points of the rock. Likewise, fractures cut all the way through these multiple different strata and offset them, which lets you know that that's not how they were laid down. Geologists basically have criteria that tell you when a layer has been disturbed, when a formation has been disturbed. And they base those criteria on areas today which are currently undergoing these processes versus areas that aren't. So areas that are undergoing like this big crunching right now, like mountain building orogenies, are places like the Himalayas, the Zagros Mountains, and the Southern Alps. And places that aren't are places like Indiana. Then, of course, radiometric dating. We all know this. This gives us the numeric age. It works by the ratios of unstable isotopes to stable isotopes within rocks using known half-lives. And they assume only, radiometric dating assumes only the constancy of the laws of physics. So the way that this might work for dating fossils, since you can really only date, not only, but by and large, we date igneous rocks and crystals. So igneous meaning volcanic, rocks and crystals. But we can use them to bracket specimens. So maybe we can't always date a fossil itself, but we can say, okay, if I've got this geologic column over here on the right, and I want to know the ages of these two bones that are in the geologic column, if this is an ash layer that's 2.6 million years old by radiometric dating, and then here's another ash layer that's 2 million years old by radiometric dating, and and then here's a third ash layer that's 1.8 million years old by radiometric dating, then we know that this bone is 2 to 2.6 million years old, and this bone is 1.8 to 2 million years old, assuming that there aren't any signs that these strata have been disturbed. Again, we know what to expect if they have been. But how the hell do you date them when they're on the surface? Like, what do you do with this cetacean down here, or with this dinosaur up top? The cycles of geology guarantee that the surface of the Earth is constantly changing. So areas subject to intense erosion over time are going to expose these fossils that are down underneath to the surface as the surface of the Earth is scoured away by wind and water. What this means is that bracketing layers, which are datable, are gone, as are your relatively younger strata. So, a tale of two field sites. This is Hell Creek, which is where Will's done some paleontology, and this is Koru, which is where I've done some paleontology. Koru's in Kenya, Hell Creek is here in the United States, but they have a lot of similarities. You'll notice that a lot of them involve collecting fossils on the surface, and a lot of them too, or both of them, I should say, both of them involve collecting fossils on the surface or digging down into the sediment if you see something poking up. You'll also notice that in both of these areas, they're really hilly. You can see these hills in the Badlands here of Montana or North/South Dakota, and you can see these hills here in Koru covered in grass, which makes my job very, very hard. Surface fossils in one place are not surface fossils everywhere. This is the first way in which we get a general age for fossils on the surface. You look at an area like this and say, okay, we have this type of shell on top, and we also have this type of shell on top over here, but this whole strata and all of the creatures within it are missing in location 2, and several strata are missing in location 3. Then we have another cut here through section C, and another cut here through section B. So let's say we wanted to know the age of this C collection of fossils in location 2. It's on the surface, so we're up the creek here without a paddle. But we know that everywhere else where these types of fossils are found, they are overlaid by this orange type of fossil in location 1 and in location 4. So you can go to these areas and radiometrically date ash layers or datable material in these larger geologic columns, and then apply that to this unknown or surface level find in location 2. That's one way in which surface fossils are dated. Another way is that many of the surface finds are on the surface because they've eroded out of hills or out of cliffs. And you can track the sediment flow from where they are to where they were. So in this hill in Koru that you can see highlighted by my cursor, or with me with this big dumb grin on my face, both of these have exposed hills, and you can find fossils at the base of these hills. But they didn't start there. And you know that because they're usually parts of skeletons. And you can find bits of the skeleton as you move up the hill, and occasionally find the rest of it in its original resting place at the top. Finally, occasionally, when you're really lucky, you can either date the actual fossil itself. I didn't know you could do this, but John told me about this study, Will, where they dated the actual eggshells of dinosaur bone, or of dinosaur eggs in this site in Yunyang, and got a really old age. You also can date sediment that's within fossils sometimes. So bones aren't usually solid, right? They have a honeycomb structure inside the spongy bone. Sediment can get trapped in there, and when paramineralization is occurring, minerals can get trapped in there. Elements, isotopes can get trapped in there. So occasionally you can rescue sediment from inside a fossil and get a general age for the fossil, because we know that that sediment was not in this ape's head when it was alive. It wasn't until it died and was buried that that sediment got in there. So these are how you can date surface level fossils. As a side note, I just wanted to do this really quick. Neither of these field sites could result from a global flood. So at Koru, which is where I work, this is my friend Venanzio. He does work on fossil leaves. Koru is a site that is primarily volcanic in preservation. So it's all these layers of periodic volcanic eruption over time. And you can still find the volcanoes that erupted 23 million years ago in the surrounding area. And part of why we know this is because we find these root traces burrowing through some of these smaller ashfall layers as plants tried to recover from the ash blanketing them. They grew upward and then were essentially choked out by that ash. In addition, the paleosols or the soils that are recorded in these areas are like dry conditioned soils. They don't form underwater. And you have weathering in between. So these layers couldn't have been deposited all at the same time, because you'll have a bunch of layers and then a carve out by a river that cuts through, and then it's filled in by more layers. But I think most strikingly for what I study is that nothing that lives here is anything other than Miocene fauna. There are no modern mammals and there are no dinosaurs. It's all stuff that's close to modern things, like this big tragulate or this anthracothere or this member of proconsul. I also researched into Hell Creek a little, and I don't think this could form under a big flood either, namely because I found a bunch of reported seasonal signals. So the Hell Creek spans 3 states, lots of different environments. It's got forests, it's got swamps, it's got coastlines. And in all of these areas of Hell Creek, you can find within their respective biomes seasonal signals. So I found this paper that records wet, dry seasons recorded in the types of plant growth, in the recorded leaves and the recorded plants that are fossilized in Hell Creek. This paper looks at groundwater fluctuation, which occurs summer versus winter. And this one looks at seasonal wildfires. That's a big one for me. I don't know how, if Hell Creek is a flood formation, I don't know how you're getting like wildfires in the middle multiple times. Like you've got multiple instances of charcoal formation through it. And it spans about 2 million years if memory serves. When I was researching about this too, what's crazy is that Tanis, which is a site in Hell Creek, is a great example of stuff that was buried by a flood. Right? Like we know what it looks like when stuff is buried by a flood. Tanis is a great example, which was like right the day of the big asteroid impact. It's an unbelievable site that's very understudied. And of course, all of the fossils that we find in these non-coastal sections of Hell Creek, they're non-marine, right? Like the water species that you find there are like, they're like river species. They're river dwellers, at least when we find them today. Or they're land animals. Like I would expect there would be like at least one like marine shark or something in the middle of there, like specifically like a marine shark. Okay, so this leads up into primates. Let me see really quick here. We're at, oh good Lord. Okay.
Yeah, I'm starting to fade.
I know. All right. Let me see. I think we can do this pretty quick. Actually, let me see how many super chats we have. I like this back and forth, but it also like
yeah,
exactly. we got a lot of super chats. Okay. How about this? I'm gonna punt this to next time. Does that sound okay?
Yeah, for sure.
I have basically all the I've got like that slide. Let me tell you what slide that is. That's 127 out of 155. So we still have like quite a few to go. God, and we like cut it too. Okay, so we'll group early primate evolution to next time. I think I've given you enough to mull over anyways.
Yes, thank you.
I guess it ended up being ape genetics mostly this time, but I guess it is what it is. How are you doing?
I can knock these super chats out and then I gotta go to bed.
All right, let's do it. All right, we got from Michael Ellott for $9.99. Think you both watched every minute but won't be able to watch live tonight. Question for Will: what is the affirmative case for Earth being young without reference to the Bible?
Yeah, really good question. I don't know, if there's I don't know. Here's what I'll say. There seems to be things that make sense if the Earth is young. But I also think there's things that make sense if the Earth is old. If you haven't heard me say this before, I actually think that every single young Earth creation ministry is wrong when they say 6,000 years. So I won't make any friends saying that. I do have a goal, which I don't know if I've ever stated publicly, which is just a pie in the sky thing of trying to get a big Young Earth Creation Ministry to drop that number. But I feel like it might be too late to do that. But I will tell you that, I've mentioned this in the past. My top three things for the younger side, and again, I see the compelling nature of the things on the older side as well. My top three on the younger side are the faint young sun paradox. I forgot. I think John actually emailed me about that way back in the day, and I never responded. That's number one. Number two would be coastal erosion, which I read about from a geologist who he actually converted from old Earth to young Earth because of his knowledge of geology and coastal erosion, and then dinosaur soft tissue. So those are kind of the three. At this point, I'm willing to say I don't know how old the Earth is, and I'm fine with that. I don't know if I want to spend my time on that per se, but I did think of something interesting, which I wanted to ask you, Erica, and I want to ask a lot of people this actually. Are you guys predicting, meaning everybody other than young Earth creationists, are you guys predicting that there will never be DNA found in dinosaurs?
Man, I have no idea. If you had asked me, if you had asked me, if you had asked me 5 years ago, I would have said no. But I think now that we're finding so much biological material in so many fossils, I wouldn't put it out of the question to get some unbelievably degraded material that is not in everything. I think I wouldn't put it outside the realm of possibility. And the reason is because we know that degradation impacts, like, not degradation, that conditions impact degradation, right? Like something rots faster in the sun than it does in the fridge, like in the cold.
Mhm.
So I would expect that it's possible. I don't even know what it would look like, but I would suspect that it's possible that there is some bone out there that you might be able to get some scraps of DNA from. I don't know though. I would love that. I hope that happens.
Yeah. Let me also remind you that someone sent a $300 super chat before we started.
I have it pulled
Oh,
up already.
okay,
Yeah,
good. I just
I
wanna make sure we
also
don't miss it.
remembered that. Yeah, I had to like, for whatever reason, because it was last night, it like kicked it from the list because the stream
Okay.
hadn't started yet. But I have it pulled up.
Oh, you know what? I wanna respond to something.
Hmm.
I think, and by the way, you guys have heard me tell young Earth creationists arguments that they need to stop making, right?
Hmm.
Like they need to stop saying evolution is a theory. And I can't remember them, but I say them. And so I'm gonna give you guys an argument that I think you all need to stop using. It's really bad. You need to stop talking about the fact that Mary Schweitzer, who originally found dinosaur soft tissue back in 2005, is not a young Earth creationist, and that she didn't conclude the Earth is young with her findings. Here's why that's a completely illogical, invalid argument. I'm going to give you two reasons for you to understand this. And my goal here is to help you understand what young Earth creationists hear and feel when you say that, because it makes zero sense. Had Mary Schweitzer, who was a young Earth creationist in the past, found the dinosaur soft tissue, did everything identical that she did, published her same papers, but concluded that actually I'm going to go back to being a young Earth creationist. None of you would be a young Earth creationist. Zero of you would. You would disagree with her conclusion and you would say, we disagree, but thank you for finding it. We're going to go figure out how this material lasted as long as it did. Okay. That right there should prevent you from ever making that argument again. The second argument that I'll use is this. When a young Earth creationist finds original biological material in dinosaur bones and publishes it in the peer-reviewed journals and concludes that it means the Earth is young, you all disagree with him. Okay? I'm speaking of Mark Armitage. And so you're doing the exact thing that you're telling us not to do. And so you can see that, and I'm not saying it's invalid. Please disagree with him. It's valid. It's okay to disagree with somebody's conclusion with their paper. So I'll leave it at that.
We have, for the sake of time, We have the $300 super chat from Alfredo Jones, unbelievably generous. Again, all the super chat money does go to charity here at the end, to our respective causes. Alfredo Jones says, don't know if I'll be able to watch this one live. I wish I could make this on the screen, but I can't because it's not in StreamYard because the stream hasn't started yet. I don't know if I'll be able to watch this one live, so preemptive support. Colorado cornfields are notoriously lacking cell service to even make calls from. Been missing the premieres of the last few, trying to make up for it. Thank you very much, Alfredo Jones. You really did not have to do it. It's extremely kind. But you don't have to do that. For an unbelievably generous $50 from Rebecca at Examining Origins. Great series, loving this. Erica, a few months ago you mentioned it. Yes, I know. Mentioned to Talk with Will and Fuzz Run on soft tissue and dinosaurs and other fossils. When do we get to see that? It is on my list because I don't like to just put them out there without an intro, like giving my thoughts on what happened. It's the same reason I haven't like posted a clean version of like my talk with Peter Line or my talk with Fuzz Rana just on his channel, which I'd love to put on mine. I have it. It is definitely coming. You'll see it. If I become evil overnight, Will also has access to the stream. So if you want to expose me ever, just be like, Will, give me the video and I'll post it myself. But it's coming, Rebecca. I'm sorry. I really have been dragging my feet with it. Thank you. Also, guys, I'm only reading the $15 ones. I accidentally slipped. You get a pass. Michael A. Lot 708 because I forgot we're only doing 15. But I do have to skip some of these. Thank you very much for your support. I'm sorry. If you want a refund, ask me and I'll do my best. We have from Danny Isenga for 22 euros. Thank you, Erica, for highly informative lessons. Question for Will. When physical evidence contradicts multiple eyewitness accounts about some past events, which should we believe and why?
I wish there was an example here so I could process what they're asking, but I don't understand an example of what they're talking about. I think to try to give a good answer here, you have to take everything into consideration, right? So if eyewitnesses are lying, then that would be an example where physical evidence would contradict eyewitness accounts, and you would hopefully figure out they're lying, and you should believe the physical evidence. Now, you could be interpreting the physical evidence wrong, which has happened all throughout history. And therefore, the eyewitness accounts are more accurate because you're interpreting the physical evidence wrong. So hopefully that answers the question. I apologize. I don't know exactly what you're asking there.
We have from Andrew 7626 for $15. Former YC, now theistic evolutionist and vet student. Erica, great job teaching. Will, kudos for doing this. I'd like to email you. Please read it, but don't feel the need to respond because I know you're super busy.
Yeah. So could someone put my email in the chat? That always fails when I try to do it. By the way, please email me. But I want to brag for a second if I can. After last session, there was a point in time where I responded to every email that I received after last session. That's like a huge victory for me because I really do try. So anyway, yeah, please reach out and I'll do my best to respond.
I know you do because oftentimes people that email you CC me. And so I always get notified when Will responds. I'm like not in the conversation. Sometimes I try to like give my 2 cents, but it's not, not always about me. From Jackie Kong for $20. Math starts with definitions and derives consequences. All empirical sciences start with the data and derive definitions. That's why of the class tetrapoda is not a circular definition. It's defined that way from the data. This is what I was getting at when I pulled that slide back up to like give the genetic or developmental characteristics.
Yeah, I don't understand what they're saying here, unfortunately. I stand by the fact that it's definitely circular. I don't think you could just go to anybody on the street who's never heard of clades and tell them a tetrapod is someone of the class Tetrapida and ask them to tell you then what that means. They won't be able to. So I apologize. I don't know what you're saying here about math and science. I wish I did, but I don't.
Do you think it's fair to say, do you think it's more fair to be like, okay, tetrapoda is defined by, limbs and digits or the developmental presence of limbs and digits? Or genes for them even? Yeah,
possibly. I'd have to think about that because the good thing you're doing there is you're actually putting a line in the sand, because it could be that we find something genetically that doesn't have that.
Right. Or
And then,
developmental.
huh?
Yeah, I said or developmentally, like either.
Y-
Either or.
Yeah, exactly. And then I think if that happens, it would just be immediately accommodated. So,
We have from Simon Gordon 8182 for 1999. Language is as messy as is biology terminology. When we say fish, that word is overloaded. When we say we're evolved from fish, we do not mean the same as when we talk about fish today. Fish evolved into fish and non-fish. You're
yeah,
making it
yeah.
worse, Simon.
Yeah, I don't mention the fish one too often anymore. The ones that got me were the anapsid diapsid and the one that we just talked about with tetrapods. So
Yeah.
the anapsid diapsid one is super interesting to me because it's literally a physical characteristic that puts you in that clade, and they don't have it. And so now it's like, okay, well, we think they had it, we think they lost it. There really isn't any evidence of that either. And so anyway, it's just a little bit, it's fine, by the way. I'm not complaining. I'm just saying the more fluid and flexible and accommodating this is, the harder it is for me to, feel like this is something solid that should be accepted.
I wonder if we found that all turtles or tortoises had the genes, genetic remnants, whatever, for forming that fenestra,
Mm.
would that then be good support for including them? I don't know if there is
I,
or isn't.
I
I just thought about looking for it.
yeah. That would be compelling to me. But by the way, I didn't mention this, but if what you said about ERVs is true, it's very compelling. Very compelling.
Please, by all means, check my work. And also, if you want, you should reach out to Dan on that because he studied viruses. He knows them
Mhm.
better than I do. I got most of my references from videos that he's made that I then read myself, and like I had to relearn. Like no shame when you were asking questions about viruses because I odds are I probably learned it like 2 weeks ago. Like I've been reading about them
Ah.
and like how little I realized I knew about viruses. So please, by all means, check my work on that.
Okay.
Okay, we have from the Salubri Outcast for 15 Canadian dollars. Morphology is a highly faulty thing. If something works well, it keeps showing up again and again. That's how it works. Think of how humans all around the world made beer, bread, and swords.
Hmm. Yeah, I think I understand what they're saying. It
Yeah.
doesn't doesn't do much for me, especially the beer, the bread, and the swords.
It's like,
uh,
yeah, comparison to with the design stuff I know can be kind of tricky because like humans make those.
yeah,
But I will say that like,
exactly.
yeah, yeah, go ahead.
It sounds teleological to me, and I know you don't want me to go there, so
It's okay. It's okay. And
okay,
I know you don't want me to go there talking about convergence, so we can live and let live on this.
okay.
Okay, we have God, I really wish I could read these, you guys. Maybe what I can do is I can put them somewhere and maybe we can do them sometime because there are some really good ones in here, but they're not 15 or more. So, and that's what we said, and Will has to get to bed. Again, it's not because we're trying to make money. I don't want to keep him here all night. And I knew this was going to be long and we didn't even finish it. So we're all compromising. Okay, we have from the same person from the Slubry Outcast for 15 Canadian dollars. As a cradle Catholic turned pagan, fundamentalist claims on the age of the universe utterly confuses me. It feels heretical claiming that you know the mind of the big guy.
Hmm.
I think that's for you.
Yeah, man, I wish I understood it. So here's what I'll say. Knowing the mind of God is going to be very difficult, and I would say impossible most of the time, unless God reveals his mind somehow to you or to us. In terms of the age of the universe, though, I don't think you necessarily have to know the mind of God to know the age of the universe. Like they're not at odds with each other logically. Um, so yeah, I mean, if I make a candle and and I light it and walk away, you might be able to, with the wax that's melted and the rate, be able to figure out how long the candle's been burning without knowing my mind. Hopefully that helps.
We have from J. Lou Bell for 1999. Linnaeus did a great job with what he knew at the time. That's me praising a creationist, getting you in there, the earlier one. The more we learn, the more we adjust. But we've never had to throw his taxonomy out and start over, including when genetics surprises us. That's what I was trying to get at the earlier levels. Like, I agree. Like, I don't think we know what to do with octopuses. But Linnaeus also didn't categorize all the species of octopuses. Instead, he was like, cephalopods are more closely related. They share more physical characteristics with, snails than they do, or snails, and it's snails, it's clams, and it's, what's the other one? What's the other mollusk? Snails, clams, cephalopods. I don't know what I'm doing here. I'll remember it. Whatever. He grouped them on the basis of physical characteristics. He wasn't like getting at the species of octopus, which is why I really want to dig into your papers. Maybe you can forward me the emails and I can like comb through them.
Sure.
We have from the Slewberry Outcast for 15 Canadian dollars, comb jellies and sea jellies. Do tell me, go on, tell me they're the same kind. Comb jellies and sea
Okay.
jellies.
Oh man, is it a comb jelly? I think it is. Okay, so comb jellies are amazing. I'm just Googling it real quick because I don't want to mess this up. Yes, okay. I ran into a comb jelly recently, like within the last 6 months. I didn't know they existed. I didn't even know what they were. I was out in the middle of the ocean with water so deep I couldn't see anything other than blue. I couldn't see the bottom. I was with a bunch of people spearfishing. And all of a sudden, there was a comb jelly right by my face. And it was the coolest thing ever. I can't even tell you guys how neat it was. It has rainbow colors that were moving up and down in it. And I couldn't stop looking at it. I took a video, but the video is not that great. I had never heard of it before. So if there's a really... Yeah. If there's a really good video you can find on the internet, go watch it because it's like a professional light show underwater. But I have no idea if they're related to sea jellies. All I know is that I was so excited that I got to see this creature. And again, I had to wait till we went back to shore, got back home, and I could like look it up to figure out what in the world was this creature that I came face to face with. It was really neat.
These guys are gorgeous. I'm looking at these like pictures of them, and you can see some of the rainbow color. I bet that was nice.
It's the movement that's stunning. So the pictures won't do it justice. Try to find a video because the colors move up and down. It's unreal.
Wow, those are awesome. We have from 1999 from Joshua Stink 5636. Will said the beauty of birds can't be explained by evolution, but birds find birds more beautiful than you find them, I hope. This is sexual selection in action and evidence for evolution. That's for you.
Yeah, so yeah, this is a tough one. This is going to be a much longer conversation, unfortunately. I don't even understand attraction according to evolution. So that to me seems like something pre-programmed by God. Like, why are we only attracted to our own species? So anyway, yeah, I appreciate what you're saying. It's just difficult because, like, when I look at a macaw, right, it's insane. Like, all the inside feathers are one color that's so bright. All the outside feathers are another color that's so bright. And I just don't see how if one feather changed to a different color, how like all the rest are going to stop reproducing and they're all going to be like, nope, I only want that one that has that other color. And then they reproduce and now there's two with another color. Like, it just, I struggle to wrap my mind around that.
My like one sentence answer to that is I think that animals that undergo sexual selection, they have preferences. Just like my favorite quote from Linnaeus is not the one where he says humans are apes. My favorite quote from Linnaeus is the one where he says that humans aren't that animals too have souls and that the difference is of nobility. And I think you see this show up time and time again with these early naturalists, where the more they study animals, the more they're like, these things are awesome. And they're like they're much deeper than we give them credit for. At least that's what we gave them credit for at the time. So I think attraction isn't as mechanical maybe as you're presenting it. I don't think it's any more complex necessarily than, me preferring brown hair. I like it because I like it. And sometimes those characteristics are coupled with health, right? Like in Wakaris, which we didn't get to primates, but this is a type of primate with a really red face. There was a study that linked the redder Wakari faces with greater health in the males that had them, like a stronger immune system. So it's like maybe the females like the red. They don't have to know that that's good for their immune system. Anyways, we have from Corey Markham, 1822, for 25 Australian dollars, science welcomes honest inquiry and challenges to existing models. The issue, Will, is that many YEC scientists wear two hats, genuine in science settings, but YEC circles, they are ideology-driven rather than evidence-led.
So that's a really interesting claim, and it might be true, but if they're not admitting that, like I don't see how you can state that with certainty. So let me turn the tables really quick and say, what if I told you that atheist scientists wear two hats and they are ideology driven rather than evidence led? Like, would you just believe me? Would that be convincing? It wouldn't. And so how you feel when I tell you that is how I feel when you tell me this. Hopefully that helps.
I want to add my one sentence to this, which will not be one sentence. I would agree with you on this, Will, but I think that it's not, in actuality, it's not atheist versus creationist, right? It's creationist versus evolutionary theory in deep time, which has people of all walks of life, of all faiths that what's the word I'm looking for, that further the field and find no problem in it. Whereas, at least in my experience of the creationists that I know, they're all either Christian or Muslim. I don't know any atheists who believe that the earth is young, or any Buddhists or any Hindus. I would think that at least once somebody would come to that conclusion independently.
Yeah.
We have from, I got a $5 here. Thank you, but I can't read it. From Taylor Gallia, 698 for $15. I'm pretty sure the ability to see red, an objectively beneficial trait, comes from a mutation of a gene that controls green sensing cones. That is true. It's actually a duplication of an existing cone that's then shifted slightly by a point mutation, I think.
Hold on a second. Sorry, I got to go back to that. Who was not able to see red?
So there's an entire group of primates that they're not trichromats. So they can't see, like lemurs and lorises, they don't have the ability to see all the colors that monkeys and apes do. Monkeys and apes can see more colors than the lemurs can. There's a mutation that is basically a duplication of one of the cones, because the reason why we are trichromats, why we can see all of the colors that we can see, is because of 3 cones instead of 2, right? Like dogs and cats have 2 cones, and so they can see blues and they can see yellows, but they can't see reds or derivatives of reds, which means they can't really see violets or oranges very well.
Okay, sorry. I misread this. I thought it meant in the human lineage there were humans that couldn't see red. Okay.
No. All humans, other than the colorblind. This happened earlier.
Okay.
Hopefully I didn't butcher that explanation. It's been a while since I looked at that. From the Salubriacast for 15 Canadian dollars, lactose intolerance is the non-mutated thing. Milk drinkers are the mutants, and it gave them a survival advantage, more food you can consume.
Yeah, I misspoke. I meant to say lactose tolerance. But again, like, yeah, it's if this is a survival advantage, then that means everyone should have it eventually, right? It should reach fixation. That's my first issue. I will predict right now it won't reach fixation. I don't know why. It just doesn't make any sense to me. But the second point, which was the most important point I was making, is that this doesn't change our body plan.
We have from $20. I know. East Point Pictures for $20. Hey, Gutsick, any chance you can do the interview for my doc this month? You told me to poke you post-porn, so poke. I know, Travis. You emailed me, and I owe you a response. You actually owe like 4 or 5 very important people with pressing matters emails back, but I've been working on this, so I did not get to it. So I'm sorry. I owe you an email. $10, $5. Okay, from Blackmore, $278 for $20. I'm late. What does redshift discrepancy mean to YEC? The math for redshift only works if galaxies are far, 10 raised 23, miles. Wow, that's a lot. That's far. Taking light billions of years to travel, speed equals distance divided by time. The YC argument should be no redshift at all. I don't know anything about this, so this
That's
is fun.
okay. I'm not your guy either, but what I will tell you is this. Jason Lyle's predictions were based on the fact that he believes God created the universe all at once. The secular model, the Big Bang cosmology model, states that the galaxies that are the furthest away require time for that light to travel to us. And so when we see distant galaxies, we're actually seeing them in the past because of the time it has taken for the light to reach us. So that's the issue there. And so that's why Jason Lyle went against the predictions being made by essentially everyone on the redshift and was correct. So
I don't know enough about this to speak on it without talking, out of my ear here. So I'm gonna defer until I can look into it.
okay.
We have for $20 from Ergomeister. If a cave were discovered where everything falls to the ceiling, we wouldn't discard all the evidence for a theory of gravity. It would be an amazing anomaly worth investigating. Same with out of place fossils and evolution. I think we came to an understanding on this. I think both of us are like, yeah, one observation isn't going to do it for any major theory. We have from iBiopoxel. Thank you so much, Gutsy Gibbon. Never enjoyed a class so much. I'm very glad to hear that. Thank you very much for your donation. From GK Pseudo for $20. Valkai Special. When a creationist quotes a scientist, read the next sentence. Here's my thing. I just don't know Cleary well enough to say anything on this. Like I've read one article by him, and it was the one that I posted here, at least that I remember. So I don't know what happened there. Maybe I'm misunderstanding something. I'm trying to be open on this because Will knows Clary, and Will like Tim Clary, right? Tim.
Yeah. Yes, that's right.
Will knows Tim, and Will likes Tim and trusts him. And so, like, I don't know him. I can't say anything about his intentions. I was just giving you my interpretation while I was reading it and explaining what does and doesn't make sense to me. We have from Jesse Tompus Wadlington for $15. Please tell me someone looked up the three-toed skink after my super chat last time, or I'll cry. Okay, thanks. Make good decisions. Be safe. I love you. I love three-toed skinks, and I did look it up. Actually, I think I looked up while you super chatted it. Will is looking it up now. From Scientific Origins for $15. Do you think giving him knowledge will cause Will's science denial to dissipate? I thought it might first, but now I don't think it will be enough. Thank you for trying. Here's hoping it does work. I can't like change Will's mind for him. I can't help him decide what he finds compelling. And the thing I always come back to is that Stephen Jay Gould, one of the greatest paleontologists, or I should say one of the biggest names in paleontology, influential names in paleontology ever, at least in the past 100 years, had a creationist student, Kurt Wise, and didn't convince him. That doesn't mean Kurt Wise is stupid, and that doesn't mean that Stephen Jay Gould is incompetent. People can't decide what they find convincing. I'm doing my best, but where Will comes out at the end is where he comes out at the end.
So I don't know who you are, sir, I assume, sir. But I have a question that I almost never can get an answer to, which is, what is science denial? So if you could tell me what a science denier is, that would be great.
We have from Design Song for $20. Testing the influence of previously well-hidden mechanisms that reliably formulate previous observations better than other tested mechanisms serve to enhance, disprove, established theory. Anything to suggest for testing? Okay, let me try to break this down. Testing the influence of previously well-hidden mechanisms that reliably formulate previous observations better. Then, yeah, okay, I get what they're saying. So what they're saying here is that it's like, because this is that thing when we were emailing, because we make so many reliable predictions, accurate predictions with evolutionary theory and or deep time as an assumption, as a presupposition, because of previous work, in part to overturn it, you would need to test the influence of a previously well-hidden mechanism. So something that we haven't discovered so far that reliably explains everything else we've already observed better than the mechanisms that we currently use to explain them. I don't know how you would test. That's what I was saying. I don't know how you would test that. I think you'd have to show there's a flaw in the current setup first.
Yeah, I'm good. I don't even know what they're asking. So,
Okay, from Carson Reed Davis for $20. Hey, Will, I work with NASA. Your interpretation of Dr. Lyle's predictions is misleading and does not imply what you're suggesting. I'll email you and we can set up a talk with astrophysicists who actually work with JWST. If that's
okay.
true, if you're actually from NASA, that's crazy. That's so cool.
Yeah. So please email me. Actually, he might have already emailed me. And I don't think there's anything misleading. So yeah, please do. But by the way, I met with Dr. Jason Lyle in person on Sunday. So I am very, very well prepared for this conversation.
I'm happy to host it if you guys want to host it somewhere, but I would love to hear how it goes all the same.
That would be great.
From Anita Pupe for $19.99. I thought our direct ancestors were mostly small, that hugeness isn't a part of the base model. Life doesn't go from rhino to rat size often. Is this false? Also, Drew Berry's molecular animation. Please tell me how they evolved. I don't know about this second part, but I can say hugeness, size is usually a function of environment, which is why you have things like island dwarfism or island gigantism, and why domestication tends to work how it works. We have from BBDMTG for $20. Will, I'm a lifelong atheist. Despite our differences in worldview, I have mountains of respect for you and Erica too. What did you learn from this series that you're most excited to tell your family?
Well, hopefully my family's following. Let me think about that. That's a really good question. Man, I spend so much time in research. I feel like I don't have time to talk to my family. I kid you not. No, it's fine. I spent about 12 hours alone on history of taxonomy. 12 hours.
You know what, though? I'm so glad because, well, obviously I'm biased, but you were like, I'm down with taxonomy. Like, you're down with taxonomy. The next question is how does genetics fit in and what do you do when they disagree? Like, I'm so happy with how that went. I'm really psyched about that. I'm actually so psyched.
I will answer this with a non-answer that I think will get everybody excited. So I think this is a true win. Because of this series and my research, based on what Erica teaches me, I have stumbled on something that I cannot reconcile with young Earth creation. And I have
got to tell us eventually, Will. You got to get so
I promised you I would.
Okay.
And I'm going to commit to that. It'll probably be in our final session. But I've been slowly testing the argument with young Earth creationists, asking them if they have an answer, and so far no one does. So there you go.
That does get me excited because I know you've talked with a lot of them. I know you're like actually putting this to some big names. So I'm like, God, that's so tantalizing. I want to know so badly. Okay, from J. Lou Bell for $19.99. I love seeing Will's affectionate facial expressions watching Erica go to town with her skulls. It's the same way we all feel. Skull Erica is my favorite archive. I just love them. They're so cool and I never get to use them. And I wish so badly. Like, I have so many more that I didn't even use, but I kept looking at the time and being like, we don't have time for this. I don't have time to show this. So it's getting punted to next time. A lot of these that I already got ready. We have from Tortoiseshell 710 for $20. Can't wait to watch this later. I'm at work, so I'll just drop in a super chat while I have the chance. Thank you both as always. This podcast has given me so much dopamine. Seriously, thanks. Our pleasure. I'm so glad. We have from Paleo Artemis for $19.99. Important to note about evolution, all living species are equally modern and no living species is more primitive than another. Traits can be more primitive or derived, e.g. cat paws have more derived features than human hands. So this is true. And one of the examples that I'm going to talk about with this is hand dimensions in apes. So the human condition with the long thumb and short fingers is actually found in some of the first apes we have. The longer finger, short thumb is an adaptation for knuckle walking that shows up in gorillas and chimps like way late. It's weird.
What if those hands are human?
I think when you see them, you won't think that they are.
Okay.
They're like that big. They belong to a very small animal.
Okay.
And their growth plates are complete. So they're like done growing.
Okay, gotcha.
Unless it was a human that was this tall. And they were found with an ape skull. So there's that. I'll show you. You didn't even get to meet Akembo Heslin. I should have talked faster. Okay. We have $20 from Transbluency. Thank you both. Thank you for your donation and for being here. We have from Words and Witness 1 for $19.99, coming in late and just got home after leading a Bible study on Revelation 6:7. 6:7. I don't believe you. I got so excited to see this was live tonight. Look forward to hearing the whole thing. Proud of Eric Cudwell for doing this. As always, our pleasure. Okay, I got to skip a couple because we're doing $20 or more. A lot of people just did send $20 super chats. From Where is Waldo for 20 Canadian dollars. Sorry, Erica. Mike PhD 42 in the chat has raised suspicion that Will's chosen charity is affiliated with sexual predator Tim Ballard and that the charity is actually a harmful front. Please clarify if the charity is vetted.
Okay,
I'll let you respond, but I have something to say about this too.
Yeah, so this reminds me of what flat earthers did to me. It's the best word I have for it is insufferable. When I donated a lot of money to this charity, Exodus Road, the flat earthers came out and said they pretty much just made up lies about it to try to make me look bad. So where is Waldo? You are doing the exact same thing. Number one, you shouldn't call people sexual predators without solid evidence that that's the case, like they've been charged or there's been police reports filed. I don't know Tim Ballard. I would almost guarantee that he's not been charged and that there are no police reports filed. But number two, the worst one here is a simple Google search would tell you that Tim Ballard has nothing to do with the Exodus Road. So this right here is despicable behavior, and I hope you can see it.
I'm trying to do some Googling in the background, but I'm happy to like look into anything, right? Like this is a pretty big claim, so I'm gonna look into it. However, the last time I looked into Exodus Road, I tried my best to do my due diligence to make sure that it was like, a highly ranked charity, and I couldn't find anything negative about it when I checked then. So I don't know what to say about this. Where's Waldo? I'll look into it, but I just don't, and I don't know. I haven't had a chance to look at anything other than what I've seen before and like quick Googling while Will was speaking. That's a big claim though. So, I don't want you to be right, but that's a big claim. I'm going to look though. We have from Dazen 13 for $19.99. I've learned about fetal milestones like tail webbing, eye-ear placement, seemingly experiencing evolutionary history in brief. Recently, I learned my child had fur lanugo. Does this also relate to a protofur, or is there another reason? I don't know about lanugo. I do know that newborn babies are really cool for two reasons. One, they have a grasping reflex. So do you know about this, Will, the grasping reflex?
Mm.
This is really cool. So if you take a broom handle, and I don't know how they figured this out, if you take a broom handle and you put it in a baby's hand, because they're opening and closing their hand a lot, you put it in their little hand, they'll reflexively hold on to it to the degree, and their grip strength is so good at that age, and that reflex is so strong that they can actually hold their body weight. I don't know how they figured that out. I hope someone was sitting underneath with a basket. But what we would have talked about today, at least in brief, is that this reflex exists in all primates. And the reason it exists in all primates is because primates ride on their moms' backs. So we don't know why humans have it, human babies have it, but the evolutionary theory would say that the reason they have it is because it's a leftover reflex. Also, all babies can instinctively hold their breath. It's called the mammalian dive reflex. I also don't know how they figured this out, that babies, like, if they fall in the water, they hold their breath instinctively. Someone is just like had a rough upbringing. We have from, I'm so sorry, Toy Fist, but that's $10. From 16 pounds from username Occupatum. Engineer here. Evolution is the best design system I've ever seen. Great argument for God, and a system that adapts and heals itself. Far greater design than hand placing every species one of two. And the second one is I had a full semester class on evolution in engineering college. It's often used for complex problems. When we can't design it, we simulate evolution from a few simple rules. It works great. I didn't know that you guys did that in school, but that's really awesome.
This is a great comment, and I agree. So I've said this before, but one of the things I'm most certain about is God's existence. And so if evolution is true, I'm looking at it through the lens of God is the one that created it, designed it, etc. So this is definitely on the table.
We have from Sadu for $15. Please watch Project Tierra artificial life documentary from 1990s and try out Boxcar 2D HTML5. Great examples of visual evolution in simulated systems. People talk about that a lot. Like the evolution simulators. I've never really played around with it, but Luke and I always talk about trying. Okay, we have from oops, that's $5.99. Okay, wait, what is this? From let me see how much this is. I don't think this passes the buck. Because some of these I have to convert. Luke gave me a list. Okay, I don't think that passes. I think that does, but that doesn't. Okay, next. From Douglas Boylan 3477 for $20. Well, this question may have already been asked already, but if it was, I missed it. Over the course of these lessons, have you encountered anything that is better explained by evolution than creationism? Much love.
This is a weird one that I haven't really mentioned to people yet because I don't know what people mean by creationism. Like, I don't know if creationism is synonymous with young earth creationism, because what's an old earth creationist, right? Are they creationist as well? So it's a weird one for me. What should I say here? I think faunal succession is the best answer I can give. It doesn't make a lot of sense. How about this? If faunal succession is true and it's as it's presented, it's exactly what evolution would predict. And I don't see why creationism would ever predict that.
That's a great answer, and I was not expecting that. We have from Stoops for $20. Not a question, but I want to say Will's example of triangles and cons at the start was very successful at producing an emotional feeling in me that's probably similar to what he has with cladistics, so I understand him a bit more.
That was the goal.
From Bill Branham for $20. Awesome presentations and dialogue by both of you tonight. Thank you. It's our pleasure. We have from Black, and thank you very much, from Blackmore 278 for $20. Speaking of repetitive DNA, many genetic diseases are caused by DNA repeat expansions, including Huntington's. It's crazy that both parents might only have 40 CAG repeats in a gene, and the child has over 200.
Wow.
That's crazy. But then that's another example. Like, and if you sequence their genomes, those would show up as differences, even though it's just repetition of what's already there. Okay, we have from Squilliam Fancy Son for $15. This is a far-fetched scenario, but if we found aliens that aren't descended from our Luca, but their genomes were similar to ours, even non-functional, could that falsify evolution? That's a great question. I'm gonna have to think on that. I don't know. This is a cop-out. It would be a point towards it for sure, but like I'd have to think on like if it would be a good like single observation that would do it. That's a good one. We got from Neurompedits, $15. This is the stream that eventually ends. Yes, it goes on and on, my friends. We started watching it, knowing what it was, and we'll continue to watch it for a long time because it is the stream that never ends. Yeah. We have from Words and Witness 1 for $19.99. The ERV evidence, along with Erica's fossil evidence of human evolution, was crucial to my transition from old Earth progressive creationism to theistic evolution. Very compelling. God is so amazing as a creator. And I haven't even gotten to my fossils yet. I'm so sad. It's going to be, Will, this is my promise to you. I want as little genetics moving forward for the next 2 at least. We have from Cognitive Trifurcation for $15. On the topic of needing better models to throw out old ones, we know the Copenhagen interpretation of quantum superposition is wrong, but we use it anyways because we don't have a better model. I did not know that, and I don't know what that is.
This is a really good point. I'm an expert in the Copenhagen interpretation of quantum superposition. Just kidding. I've never heard of it. This is a really good point. And here's why. He might be making an observation that's true. I'm not doubting him. He might be onto something bigger, which I've been thinking about in our conversation, which is it might be, if we go back and look through history, that models don't get thrown out until there's a replacement. That makes total sense to me. Like why? Like maybe we'll see a history, including this, example where it's like, this is the best we have. We know it's not perfect, but we're just going to use it and keep it until there's something new. That's a good point. I'm going to look into that.
That is a good point. I wonder too if I bet you can plot like usually models that don't work are maintained to some degree in the new model. So like relativity includes aspects of Newtonian physics. So I wonder what it would look like for evolutionary theory. I don't know. We have another for $15 from Blackmore 278. Another benefit for viral inserts is that they can add promoters for already existing genes. In humans, the genes EDNRB and APOC1 have promoters added by viruses. That's true because they get, yeah, promoters is another entire thing that I really can't wrap my head around very well because genetics is not my area. But I did come across this while I was reading. Okay, we have from Charlie Stanley 6303 for $49.99. In the ERV study you shared, I knew this was coming. This is a great question. Gorillas shared 207 ERVs with humans, while orangutans and chimps only shared 205. Wouldn't we expect humans and chimps to share more ERVs than with gorillas? Did the paper authors address that? Yes. I thought that was strange too. And one thing that I noticed, you can see this in the figure that I include, the Y chromosome has some aspects of it that aren't included, which is sort of expected because chimpanzees have like a weirdly small Y owing to the dissimilarity between the human and chimp Y chromosome, and the Y chromosome between chimps and hominoids in general. It would not surprise me if that's where those ERVs are, the missing two, since the Y isn't included and chimps have a small Y, and then it would put all the great apes at 207, which I would expect that. But I don't know if that paper's from like 2016, 2014, something like that. So I don't know if they've done anything since. That's a great question, and I wondered it myself. From Control-Alt-Shift for 20 Canadian dollars. I love Will's presentation today. Great questions and challenges with a lot of citations too. I can't wait for the final comparing notes stream. I'll miss the months new streams when this is done. Me too.
Yeah.
Maybe we can like touch base every 6 months or something just for fun.
Yeah. Love it.
We have from David V3M8I for $15. You should use comparative genomics to compare gene order between any species with a reference genome. Makes really cool figures and no genetic skills necessary. Yeah, you can also play around with the Ensemble browser, and it's pretty cool too for that. From Mr. Sandman for $19.99. How many human men does it take to beat up an alpha male silverback gorilla? It depends on what you mean by beat up. I think, and it depends on what they have. Like just fists, I think you need at least 5 guys. If they have weapons, 2 guys could do it. Silverback gorillas are really, really strong, but humans are really, really smart and very like intuitive. That'd be my guess. We have from Chan 2421. Oh, sorry, $5. Sorry, Chan. From J. Lou Bell for $19.99, best class yet. Will, last time you made the brave admission that you thought humans or animals might make, I think they mean God mad here, because I remember when you said that. How are you doing with that? Does schmanimals help? Do you think flat earthers have the same fear?
I have no idea about flat earthers in terms of humans and animals. That topic doesn't come up. I definitely am okay with classifying things based on phenotype, which is phonetics. We're good there. I don't look at us as animals. We're too different. I will say something super interesting. One of my favorite YouTube channels is called Closer to Truth. It is the coolest YouTube channel ever. And they have like not a lot of subscribers and they don't get a lot of views. They might have a lot of subscribers, but they don't get a lot of views. I love that channel. The guy that runs it does not agree with me at all, by the way. But I just watched a video this week from that channel and I was blown away. But I think the guy on there was arguing that animals are not conscious. And I was like, what? Now, to be clear, I'm not talking about a conscience. I'm saying just conscious.
That's crazy.
I know, right? And so I'm like, no. Do I literally have to look into this now? And like everybody in the comments were talking about, and he was talking about a mammal not being conscious.
This is where I like look at my own experience with with my dogs, with horses, with apes, with everything, and I'm like, I don't believe that. Like, I don't
Right.
know how you would make that. Send me the video. I'd be really interested to see it.
Okay. Actually, you know what?
Also,
He's famous. Sorry. You guys all know who he is. It's Michael Roos.
I don't know who that is.
I'm
Michael Roos.
pretty sure I'm
Actor
getting it.
Michael Roos.
Yeah, Michael Roos. He's wildly famous.
I know this guy. He acts in all sorts of stuff.
Yeah. He's big. So he's the one that said it. So anyway,
That's
I'll send it to you.
crazy. I'll have to see. Yeah, I'll have to see that. That's bonkers. Also, before chat freaks out, we've established many times now that the problem is the name. We could call animals metazoans, and I think we would solve the problem. Like, I think we could just change the name and it would be okay. Like, the traits are what matter. I'm okay with that. Animal means different things in science and colloquially. That's fine. You guys got to be as okay with that as I am. I promise you, it bugs me more. And I like Will. From Ratsicle fan. Wait, what is this? Is this? I don't know what currency this is, and I can't see. I don't see you on Luke's notes.
I don't know.
Sorry, I don't see you here. You're here though. That's Australian dollars. Okay, from Corey Markham, 1822. Will, I want to say well done for doing this. I swing between admiration for your willingness to challenge your own views and work your cognitive dissonance and frustration when you seem to retreat into defensive posture. It can't be easy.
Yeah, I appreciate it. It's not easy what I'm doing, and again, no one's going to be perfect, but I'm trying my absolute best to let the evidence speak for itself and just follow it to where it leads.
I don't see. Wait, I don't. I feel like intuitively this is right, so I'm going to check the currency on this. But first, we'll do this one from Xandor for $20. Just here to boost Metazoa, a free Wordle-like daily game that teaches phylogenetic trees. Guess an animal, and the game tells you where your guess splits off from the answer. I've learned so much. Check it out. That sounds awesome. We have from Misty3252 for $20. Eric, if you had $1 billion to fund conservation work at any place in the world for any species, if you want to specify, where would you spend that money? It's not even close. I'd do Borneo or Sumatra. These are areas that are under such rapid deforestation. And I understand why, because the government in many ways has failed the people of Indonesia and also Malaysia. But at the same time, like I think with $1 billion, I could crowdfund some awesome conservation work, make a lot of jobs over there, and really help restore some of those habitats. I think it's I think Malaysia and Indonesia are one of the most incredible places in the world. I feel so unbelievably lucky to have gotten to visit. It almost brings a tear to my eye just even remembering. It's awesome. That's what I would do. And it's not close. I would say, like, I love Kenya as well, but like Kenya's doing okay conservation wise. From John Darwin 7 for $50, super sticker. And I'm gonna check this yen to USD. Oh, I don't think this is gonna make it. Oh no, it just misses the cut. I'm so sorry. And it was such a good chat too. Sorry, Kevin Moses. Wait. No, sorry, Kevin Moses. Okay, folks, that's it.
Hold
that's
on.
all I got.
There's one more I want to take. It's Where is Waldo sent another one.
Oh, I missed that. From Where's
Okay.
Waldo for 20 Canadian dollars. Will, put me in the middle of this. Why did you suggest? I was accusing you of anything. I tagged the chatter I was quoting and used quotation marks. I'm sorry, Where's Waldo? I didn't know you were quoting. I read that like that was you. That's my mistake. I wanted to clarify for anyone concerned about where their super chats go. That's all. I didn't know that was all.
I want to respond to this. So you guys probably felt my defensiveness, and there's a reason why. So I just want to explain. I have been falsely accused of a crime. We're talking completely made up, zero evidence, never happened, falsely accused of a crime that led to an arrest, that led to jail time. And so I know exactly what it feels like when there's an accusation that's false. And so people need to realize when there's accusations, you can't draw conclusions. Okay. I'm going to give two more examples that hit close to home. Because I live in Colorado, back in the day, I'm a sports guy. Okay. Back in the day, there was a 19-year-old female who accused Kobe Bryant of rape. And so what do? Is Kobe Bryant a bad person? Based on the comments I'm seeing in the chat, yes, he's a horrible human being. Well, he fought it and I believe won, and there wasn't evidence of this. What people need to realize is that people will make accusations against people, and they sometimes will lie, and they sometimes will do it because they're trying to get a settlement or they're trying to get money. Another example happened very recently. A female accused 5 NHL hockey players of sexually abusing her. And those 5 NHL hockey players were all not signed. Their contracts were not renewed with their respective teams. They were all essentially out of the NHL. So are they all horrible people? Well, based on what I'm reading, it was all not true. And she was just going after money. And they all now most of them have lost their NHL careers. And so when I see comments that are like, well, people made accusations about Tim Ballard. Okay, that's just an accusation, right? There were comments that there were lawsuits. Okay, so people sue people because they're trying to get money or they're trying to get a settlement. Just because there's a lawsuit doesn't mean that somebody has done something wrong. So anyway, it hit me personally and it hit me close to home because I've experienced it. It's horrible. It was honestly the worst time of my entire life, and you never want this to happen to you. So when there's allegations or lawsuits or complaints or whatever, you got to just wait and see how those things pan out. And so again, it's very difficult to try to determine what is really happening or what's going on. But again, the other one too is that, like, suggesting that the charity that I am recommending is associated with somebody who might be, a really bad person also hurts because he's not associated with it. And that's really easy from a Google search to figure out. So anyway, where is Waldo? My apologies for that. I kind of jumped down your throat, and now I'm realizing that you were just actually passing on. You were just the messenger, so to speak. But I did want to be a little vulnerable here and let you guys know why this hit me personally and why I reacted like I did. So hopefully that helps.
So I'm going to provide my 2 cents to this because this is a serious topic. I know, everybody knows plenty of people, but I've known a lot of people in my life, and I've known a lot of women who have been the victims of sexual assault and rape, and they were, insofar as I can tell, 100% telling the truth. And some of them were successful in making their case against their accusers, and others were not. And specifically the one I'm thinking of who wasn't was subject to intense amounts of pressure and harassment from the accuser, which there's not even words to describe how much that is inappropriate and sucks. I also know people who I know one person who has been inappropriately accused of, while not sexual assault, actions against their ex in a relationship. And I know all of the people involved in that, and I know the story behind that, and I know the legal proceedings behind that, and I know that the person was making those accusations falsely. I know far more true accusations than I know false accusations. That doesn't change the fact that I know of false accusations that have happened. All of that to say, cases like sexual assault need to be taken gravely seriously and deserve the attention with the authorities, and if need be, legally, 10 times out of 10. I don't think anybody disagrees with that. I don't know anything about Tim Ballard, and everything I looked up with Exodus Road seems great. I Googled while Will was talking just then. I can't, a very 5-second Google search for what it's worth, I can't find an association with the guy and Exodus Road. I will look regardless. I can understand if there's a sentiment that there's an association with something that heinous, I can understand being upset about that. And I can also understand if there isn't association, Will being upset about being accused that there is. All I can do as the person who's trying to orchestrate this is my best and try to figure out what's going on here, so I can promise you my due diligence. And that's all I got. I love my audience. I love the conversations that I have here with Will. I'm doing my best. Go ahead, Will.
Yeah, I would like to also add too, I was explaining my personal story and my reaction, I support the death penalty for rape. So that's how serious I take rape. Is I absolutely support the death penalty. Obviously, that's not going to happen in this country, but that's where I stand with it. And yes, it absolutely should be taken serious. I'm just want to encourage people to, unless you're intimately involved in the situation, not jump to conclusions and then let the system do its thing.
I think, and I hope we're all on the same page. I've said my piece. So on that happy note, wait, actually, that's not true. I have two short ones that were given for whatever reason. They didn't show up on they didn't show up because they were sequential. So somebody super chatted $9.99 and then they boosted it with a 5. The first one of that, there's only two. The first one is from Blanche, first for $10, then for 5, who says Newton's theory of gravity was falsified before relativity was proposed due to inaccuracies in the orbit of Mercury, but we didn't throw it away until Einstein. And Newton's model is still pretty good. That's a perfect point. I was trying to remember why relativity came about, and you've struck my memory here. I do remember now something about Mercury. And we also have from Uncle Iro for $10 and then for $10 again for a total of 20 for one super chat that runs through both. Will, you should look into the Sundance and Morrison formations. The Sundance is a marine deposit and the Morrison was terrestrial with surviving trackways on top of the Sundance. My challenge for finding a working YEC model that explains this geologic sequence. How do you have multi-ton sauropods deposited on top of buoyant marine reptiles like ichthyosaurs? That's a great question.
Yep. Thank you.
We will touch on that again. We have a lot of stuff that we still need to touch on. Folks, that's it. Thank you for being here. Been here for a long time. Didn't even finish the PowerPoint, still here for a long time. Will, thanks for being here and discussing with me as usual. We usually get off these calls and immediately schedule the next one. It's my fault that these links don't end up until the day before. I'll try to do better about that. I'll do my best. In the meantime, guys, thank you for the support for the channel, for being here, for giving your super chats, for giving your 2 cents. We really appreciate it, and we'll see you real soon next time. Ending the stream.